Good morning, everyone. Welcome to the third Biotechnology Conference, organized and hosted by the College of Engineering and Computing Sciences at New York Institute of Technology. My name is Babak Beheshti, Dean of the College. The theme of this year's conference is regenerative medicine. We have an amazing lineup of speakers today. As you can imagine, organizing such an event takes a lot of planning and effort. I would like to thank several colleagues who, without their countless hours of amazing effort, this event would not have been possible. First, I'd like to acknowledge Mr. George Licorezos, Partner and Intellectual Property Counsel at Carter, DeLuca, and Farrell LLP, also a member of the College of Engineering's Dean's Executive Advisory Board. George has been the brain and the architect behind this event from its inception. I would also like to convey my immense gratitude to the awesome staff of the College of Engineering and Computing Sciences that has delivered this remarkable event. There are many others, including the speakers, panel moderators, and panelists, but in the interest of time, I thank them collectively. New York Tech's College of Engineering is well positioned to be a hub of innovation in biotech, as it forges partnerships and collaboration with the biotech industry leaders and globally leading research institutes. With a BS degree in bioengineering, a well-respected MS program in bioengineering, and our expanding PhD programs in engineering with bioengineering track, expansion of multiple biotech-focused research labs, and a robust research program underway, we are on the right track. Our faculty are involved in active research projects funded by NIH, NSF, the Air Force Research Lab, and the Navy Research Lab, to name a few. We are making great strides in establishing New York Tech as a biotech innovation center. We hope that we can establish this event as an annual signature event for many years to come. Also, I would like to acknowledge and thank our financial sponsors, who will be mentioned several times during the summit. It is the generosity of our sponsors and donations of our alumni and the professional community that makes the creation of programs like this conference possible. Scholarships for our students in need, diversity and equity initiatives, and many other worthy initiatives also are made possible by these sponsorships. Before I pass the podium to our first speaker, I would like to take a moment to present this plaque of recognition to Mr. George Licorezos. Due to a conflict, George is joining remotely, so I'm presenting this to him via this medium. And let me read the citation for you. I think that it captures the sentiments for this recognition. In recognition of your outstanding efforts in organizing the New York Institute of Technology's Biotechnology Conference for the past 3 years, your exceptional leadership and dedication have brought together experts in the biotech industry, facilitating discussions and inspiring new ideas and collaborations. March 16, 2023. Let's have a round of applause for George, please. Thank you. It is now my pleasure to introduce Dr. Hank Foley, New York Tech's president and a big supporter of the College of Engineering's achievements, to give his remarks. President Foley, the floor is yours.
Good morning, everyone. It's good to see you all here. Dean Beheshti, thank you for putting this conference together today. I'd also like to thank our guest speakers who are leading experts in the field of regenerative medicine. They include Scott Rodeo from the Hospital for Special Surgery, Dr. Hina Chowdhury from the Icahn School of Medicine at Mount Sinai, and Chad Boughton, who is at the Feinstein Institute at Northwell. Welcome to all of you. Thank you for being here. Examples of regenerative medicine research in recent years at New York Tech are plentiful. For example, in 2023, Professor Nikos Solanias from NYT published research that could improve understanding of finger and toe regeneration. In 2019, undergraduates working with Assistant Professor Azar Ilyas from the College of Engineering and Computer Science undertook 3D printing of human organs, specifically the kidney, which I think is really quite astonishing for undergraduates to be doing. Similarly, in 2017, Professor Michael Haji Ajayu from the College of Arts and Science has been doing research on osteocytes, which of course are bone cells, and has become quite an expert at templating bone cells and understanding their growth, which is critical if we're going to understand the eventual replacement of bones and joints. It was recently reported that the market for regenerative medicine has been valued at over $50 billion. It's now projected to grow at an annual rate of over 18.4%, which is really quite astonishing. So it couldn't be more timely for us to be having this conference, and I'm very, very appreciative of it, and I look forward to hearing much more about regenerative medicine from today's speakers. Thank you.
Thank you, President Foley. Next, it is my pleasure to invite our keynote speaker to the podium. Our keynote speaker is Dr. Hina Chaudhry, Professor of Medicine and Cardiology and Director of the Cardiovascular Regenerative Medicine at Icahn School of Medicine at Mount Sinai. The topic of Dr. Chaudhry's talk is Human Heart Regeneration is an Imminent Reality. Dr. Chaudhry.
I'd like to thank Dean Beheshti, George Lacourses, and all the organizers who've done an amazing job putting this together. And I have to say, I've never spoken at a conference where they wanted my slides 2 weeks in advance, so the organization is incredible. Thank you very much, and thank you for giving me the opportunity to discuss our data with you. I'm going to be talking about heart regeneration. I'm a clinical cardiologist, and I've had further training after my clinical cardiology training in developmental biology and genetics. I've continued to have a clinical presence at Mount Sinai, and I attend in the Echo Lab with non-invasive imaging for about a day and a half a week, and I've also attended in the intensive care units up until, I'd say, the last couple of years. So I have I guess, a unique sort of way to bridge both the worlds of clinical medicine and basic science, and some of the research is going to really reflect that. And I think what you'll gain an understanding of is why we need more physician scientists in the world. Of all the MDs now, only 1.5% are physician scientists, and it's really hurting the scientific enterprise and development of novel therapies. So just if we can try to remember that and increase their numbers and nurture them. I hope I know how to. Okay, so here is my first slide, and I want to sort of tell you a little bit about this slide. This looks like a Valentine's heart, but it's not. It's an actual cardiomyocyte as depicted under the confocal microscope. And the red staining you see is a mitotic marker, which signifies a very, very specific event in mitosis, the phosphorylation of histone H3. And it is an M-phase specific event. And the blue staining you see is a nuclear stain called DAPI. And we were the first in my postdoctoral fellowship at Columbia. My postdoc paper was really the first to show that mitosis could be achieved in post-mitotic, what are thought to be post-mitotic cardiomyocytes. As you know, the heart is supposed to be a terminally differentiated organ, and further studies that have become very elegant using carbon-14 dating have shown that indeed the turnover in the adult heart is almost non-existent. There's slow turnover up until the time we're 20, but after that there's really no mitotic activity in human hearts, and in mice that sort of stops much more quickly. So this is an actual heart that was staring back at us looking like a Valentine's heart. It made the cover of Circulation Research on one of my earlier papers on this topic back in 2007 when I was still sort of dabbling with mice. We still still continue to dabble with mice, but we're really trying to move on to humans now. And as you know, cardiac regeneration seems to have been a hot topic for the past 20 years. There's been a lot of press focus, especially since some of the earlier papers on cell therapy in 2001 by Piero and Versa's group. And I was basically growing up in this field while all that was going on, as I was a postdoctoral fellow at the time pursuing my basic science training. And at the time, it took the world by storm that maybe bone marrow stem cells could actually become heart cells, which, as you know, has been disproven. The problem is doing rigorous work in a field that there was just so much controversy. I mean, as a very junior person going to attend these meetings at the American Heart, I could basically look at this plenary session slide and say, well, that's not what mitosis looks like. But when you're young, you are afraid to question these things. And I encourage all the young people in the audience, you see something, even at a national, international meeting, that just is not right. It goes against high school biology 101. Just speak up. I mean, young people in my day, we were afraid to speak. And then the field went on like that for years until something like 45 papers were retracted on the topic. It made it harder for my group and myself to publish in the field. Now, every paper is subjected to a greater deal of scrutiny, which it should be, but I'm just telling you, the field became a lot more challenging because of earlier works that were sort of not presenting the data in a very truthful way. So we strive to be extremely meticulous and rigorous, and we don't even publish our results until we know that we're right. And we've tested every experiment 5-6 different ways and 5-6 different times. And so I just want to emphasize to you, this is just so important to get it right. And if it takes longer to publish, as long as you're right, that's the most important thing. Don't be rushing to publish something that just isn't really, really rigorous. So there are both cell-free approaches and cell-based approaches, and my lab studies both. In terms of cell-free approaches, we are looking particularly at cardiomyocyte proliferation. That's what I started with as a postdoctoral fellow in Deborah Wolgamuth's lab studying cell cycle biology. And we're looking at cell-based approaches because we've discovered a novel kind of multipotent cell that can be harvested from term human placentas. And so I'm going to first talk about our gene therapy approaches, and not just necessarily focusing on gene therapy, but the process of cardiomyocyte proliferation. And then I'm going to get into our projects on the novel kind of stem cell. And that's where you're really going to see that it helps to have both the clinical side of the story as well as the basic science side of the story. Now, as we know, lower vertebrates can regenerate their hearts. But I'll tell you, this is a North American red-spotted newt, and it's known for its ability to regenerate its heart, its limbs, its spinal cord, its iris, and its retina. Now, when you take the newt heart, and going back to newt regeneration papers, back to even 1960s, I had to go to the library archives because those are not on PubMed. If you pull all the papers on newt regeneration going back decades, nobody was able to show that you can damage, say, 50% of the newt heart and it'll grow back. No, the greatest degree of damage you see is 20%. So 20% of the apex, basically, is excised or damaged. It'll grow that back. So even the newt has a limited regenerative ability, not an unlimited ability. So if you take the newt heart and put the apex, mince the cells and put them in culture, they will re-enter the cell cycle, divide in vitro, and they will reaggregate into a beating syncytium. That has never previously been shown to be possible with mammalian cardiomyocytes. And so I'm going to show you, we were able to do that with a mouse heart by introducing a transgene for cyclin A2, which is the key mitotic regulator, as I had shown this in some of my earlier postdoc work. I'm not going to go through all those papers, but just kind of highlight the key points and then get to our large animal and human data. Now, this is a zebrafish, and the zebrafish is also known to be able to regenerate its heart. This is a seminal paper from Mark Keating's lab in 2002 in Science, which Ken Poss was a postdoc, and he's continued that work in his laboratory at Duke on zebrafish regeneration. But I'll point out to you, the same thing. You can only damage 20% of the heart, and then it will grow back. And one of the key takeaways from this paper, which even the original authors have not followed up on, is that in a temperature-sensitive mutation mutation of the zebrafish, which is a temperature-sensitive mutation in the MPS1 checkpoint kinase, which is a key mitotic checkpoint kinase. Those zebrafish cannot regenerate their hearts. Instead, they form a fibrotic scar just like you or I would if we had a heart attack. So that one gene, the mutation in that one gene is limiting the regenerative process, or is actually inhibiting the regenerative process. And so the entire healing process is going towards fibrosis and not mitosis. So when you have an absence of mitosis, you're naturally going to get fibrosis. All right? So keep that in mind, because you're going to see new approaches being out there in the press where they're trying to inhibit fibrosis. And guess what? I'm telling you now, it's not going to work. Because if you inhibit fibrosis in the absence of mitosis, you're just going to get even more of a mess. All right? You're going to get a sicker, more dilated heart. And so you cannot just block fibrosis without activating mitosis. That's a really important point, because now I'm hearing about all these new anti-fibrotic therapies. So both these models, they can regenerate their hearts, but in a limited way, only when 20% damage is induced. An important lesson from the zebrafishes, it takes just one gene being mutated to go towards a fibrotic pathway. This was really exciting to me at the time I was a postdoc. I was studying the A-type cyclins in Deborah Wolgamuth's lab. As you can see here, cyclin A. Cyclin A2 is a somatic form of cyclin A. Cyclin A1 was discovered by my mentor, Deborah Wolgamuth herself, but is a germ cell restricted cyclin. The one that's active in the heart is cyclin A2. So when she discovered cyclin A1, cyclin A was officially renamed cyclin A2. So as you can see, cyclin A2 is the only cell cycle regulator that's active at the two transitions of the cell cycle, both the G1S and the G2M. So G1S and G2M are the two transitions of the cell cycle. And the cyclin A is the only cell cycle regulator active at these two. So when I noticed that in the cell cycle, I thought this is going to be an important molecule in both heart development and heart disease. And if you knock out cyclin A2, you have an embryonic lethal phenotype at day 5.5 in the mouse heart, because, you know, we can only really do knockouts in mice. So I thought this is a key gene to study, and it's silenced in the postnatal heart. It undergoes complete silencing. There's a complete absence of transcript. Back then, I was doing northern blots. My students and postdocs now are doing very fancy RNA sequencing and proving the same results that I got way back when in the dark ages using those radioactive northern blots. So the data holds even 20 years later. That's really exciting. So we know that cyclin A2 transcript is gone in the postnatal heart. And other cyclins are not silenced. And so in my earlier work, when I found that we reactivated it, we had reactivation of cardiomyocyte mitosis all the way into several weeks of postnatal development. And then the hearts were hyperplastic, not hypertrophic. And so this was a key finding, because this suggested, that suggested that the mice would probably regenerate their hearts if we induce damage. So I'm skipping over a number of small animal papers to get to this large animal paper. We had shown in the mice that if you damage transgenic mice that are expressing cyclin A2, they will repair the heart, they will increase their ejection fraction as compared to non-transgenic littermates. Then we basically, for a translational strategy, took cyclin A2, put it into an adenoviral vector, and we gave it to infarcted wild-type rats. And we showed that it also worked when you administered it exogenously as a gene therapy. And then we get to this. At that point, I was recruited to Mount Sinai as an associate professor back then to run this new program in cardiovascular regenerative medicine. And one of the reasons I chose to go to Mount Sinai is they had this large animal testing program using pigs that was all set up. And I was looking to test this in pigs. And pigs are an important animal to test cardiovascular therapies in, because their hearts are structurally and physiologically almost identical to ours. And the FDA wants to see pig data before they'll let you do any kind of clinical testing. I think they're kind of doing away with that recently, but I'm not sure that how that's going to go. So in the pigs, the overview is right here of how we did the study. Whoops. So we did angiography, 2D echo, 3D echo at time 0. And because I'm in imaging as a clinician, I was able to utilize every kind of imaging modality we use in humans to image the pig hearts. So that was our baseline. And then we created the MI. We basically put a MRI compatible platinum coil in the proximal LED to occlude flow and induced a complete transmural infarction. Then 1 week afterwards, we repeated all the imaging scans. All right, and then we opened up the left chest and injected the virus at 10 equidistant spots around the infarct zone. This is the first author, Scott Shapiro, who was a cardiology fellow at the time, doing this injection. You can see the whitish part is this is the border zone of the infarct, because you can distinguish it because the infarcted part is white and the surrounding healthy tissue is red. So he went 10 equidistant spots around, just like we had done in our rat studies. And then we waited 6 weeks. So from time 0, it was 7 weeks later, we performed all the imaging scans, sacrificed the pigs, and performed histologic measurements. Now, we had shown with every single imaging study we used, LV angio, 2D echo, 3D echo, and MRI, that we had a significant improvement in ejection fraction in the pigs that had received cyclin A2. And in the pigs that had received the control, the null virus, the same viral backbone, but missing the cyclin A2 transgene, we saw a reduction in heart failure, in ejection fraction, and there were actually a couple deaths in the control group. We did not have any deaths in the experimental group. Now, in terms of cardiomyocyte numbers here, like I said, it was injected in the peri-infarct zone. When you inject adenovirus, it basically diffuses to involve at least 60% of the entire left ventricle. And we showed that in the peri-infarct zone, the cardiomyocyte number actually increased by 55% in the cyclin A2 injected pigs and really did not budge in the null control. So this is the most important finding in this study, that there's a 55% increase in the numbers of cardiomyocytes in the peri-infarct zone, which we counted very carefully in cells per square area. And then what we had also shown in this paper, which was published back in 2014, was we had developed novel imaging tools in the laboratory to light up the living sarcomere. So we had isolated adult porcine cardiomyocytes, transfused them with this sarcomere, this alpha-actinin that was tagged with a fluorophore, and then given the same cyclin A2 adenoviruses in vitro and recorded cytokinesis events. So keep that in mind when you see current studies being published, when people are claiming there's an increase in cardiomyocyte proliferation, but if they're not showing you in vitro cytokinesis, then it's not happening, because it's impossible to measure cytokinesis in vivo. They're using all these markers like Aurora B, which they're saying is a cytokinetic marker, but it's actually not. So there's a lot of confusion in the area on this topic, but it's really important to be very, very rigorous about this. So now what we did was, after we had shown that the pig heart myocytes, but the pig cardiomyocytes were undergoing in vitro cytokinesis, and we turned our attention to human, and we had adjusted the virus. We had basically modified it so that we put the human cyclin A2 in. It's driven by a troponin promoter, so it's cardiomyocyte specific. So we were preparing that for clinical trials. And then we took adult human cardiomyocytes isolated from, these cardiomyocytes came from a 55-year-old man who died of non-cardiac causes. We have the method of rapidly isolating them from the cadaver, and they're frozen and shipped to us. And then we thaw them, and we develop culture methods to keep them alive in culture for up to a month. And here you can see this, you can see, oops, I started the movie. Anyway, so you can see the movie. That red staining is the alpha-actinin that's tagged with cherry. And these myocytes have been given cyclin A2. You can see the first division right there. You can see the daughter cell here dividing a second time. So it's inducing robust cytokinesis in vitro. So this is what I mean. You've got to go to this length to show that cytokinesis is happening in order to believe the in vivo data as well. Because one of the things we've shown, and that's not published, is in transcriptomic analysis, going back to our transgenic cyclin A2 mice versus non-transgenic mice, is that cyclin A2 is also interacting with a wide variety of contractile contractile ion channels and contractile proteins. And so this is something actively under study in my lab right now, is that how does cyclin A2 affect contractility? Because as you can imagine, that would be of great applicability to the disease of chronic heart failure, because most of testing I've done in small and large animals has been in the post-MI period, and I'm very interested in developing a drug for chronic heart failure as well. So now this is some new data that has emerged from our laboratory that cyclin A2 appears to regulate contractility as well, which would make it a very multi-use molecule. And as you know, there are very few genes in nature that don't have redundant functions, and no one has actually proven that a cell cycle protein interacts with ion channels. We hope to be, the first to publish that. Now I'm going to turn my attention to cell therapy. And this came up because as a clinician, I was rounding on the Heart Institute many years ago, and we would do 1 or 2 months a year of rounding, like teaching rounds when I was still at Columbia. And then we had patients, one month I had 2 patients with peripartum cardiomyopathy. As you probably know, peripartum cardiomyopathy is a kind of heart failure that only affects pregnant women, either in the last trimester or the first 6 months after birth. And the heart function can literally go to like really bad, like EFs of 10 to 15%. And 50% of these women spontaneously recover, and nobody knows why. And this was the highest rate of recovery of any kind of any etiology of heart failure. And so I was talking to my residents on rounds thinking, we're thinking, could there be a fetal contribution to the recovery seen in these women? And it's not a topic I generally study. We have many wonderful women's heart disease experts at Mount Sinai. That is not my area of expertise. But I was very interested in finding out if there was some regenerative signal or some kind of cell that was being sort of homing to the heart from the placenta, from the fetus maybe, that was mediating this phenomenon. Because I thought if we could find such a cell, we might be able to utilize it to repair everybody's hearts, not just pregnant females. I mean, non-pregnant females, we could put the cells in males and see if that would repair the heart. And so that's how this started. And what we did was I went back to the lab and spoke to a graduate student of mine, Rena Caro, who's the first author on this initial paper we published back in 2012. And we basically modeled experimental heart injury in a pregnant female mouse. And we had mated the wild type females with males that express transgene green fluorescent marker in all of their cells. So the pups were basically labeled with green fluorescent protein, so they glowed green. So we damaged the mother's heart mid-gestation, and we tracked the green cells coming from the fetuses. And lo and behold, we found that there were green cells ending up in the infarcted mother's heart at the site of injury, not in a non-infarcted RV and not in non-infarcted organs, which was really exciting. And then of this population of mixed fetal cells that were homing to the mother's heart, we found that CDX2 was a prevalent marker in the cells. So this was on the cover of Circulation Research with this really funny-looking picture, which I didn't think would ever get selected for the cover. So we had submitted an alternate picture as well, a beautiful immunofluorescent staining picture, but they went with a really funny-looking picture. So anyway, and then that led us to the discovery of the cell type that we are now harvesting from human placentas. Just to go over a little bit about what CDX2 is and how we found these cells. As you know, this is a blastocyst, and this is the inner cell mass, and that's the inner cell mass in the blastocyst stage. That's where embryonic stem cells are derived from. And CDX2 is a marker of these cells out here that basically are the trophoblasts, or the outer shell of the blastocyst. And these are the cells that mediate implantation into the uterine lining when this blastocyst goes and embeds itself into the uterine lining. And so CDX2 was historically thought to be only mediating placentagenesis. It also has some postnatal intestinal development involvement, and it influences renewal of these cells. Now, we thought, that's funny where they're ending up in the heart, and the people who had discovered CDX2 cells, when I would tell them at meetings I'm finding them in the heart, they're like, no, it's impossible. You're not going to find them in the heart. I'm finding them in the heart. So that's another message for young people. Don't be scared to question dogma, because without you questioning dogma, things will never progress. You will never innovate, and you can't be scared of these big shots who tell you something is impossible. It's not. It's you just we found them in the heart. We have found them in the heart over and over and over again. In mice, we're finding these cells. In the human, we were told we would not find them in the term placenta. They're only expressed in the early gestation placenta. Well, we've not only found them in the term placenta, we're able to grow them and show that they're becoming beautifully beating cardiomyocytes. So I think we've disproven that whole hypothesis. This is the work. The work I'm going to talk to you about is largely the work of Dr. Sangeetha Vadake Madatil. Sangeetha came to my laboratory as a postdoctoral fellow, and she basically was fascinated by this project. And then what she did was, in order to study the function of CDX2, she set up a mouse model in which only the CDX2 was tagged with green fluorescent protein. So this is the mouse model where you've got from the placenta, the green cells that are isolated from the placenta are all specifically now CDX2s. It's a different mouse model. All right. And so she's able to isolate them because they're tagged. I didn't go into the details. It took a year to set up this. It was not easy to set up this mouse. So she was able to take the placental CDX2 cells, put them in petri dishes and culture them, and they would form these beautifully striated cardiomyocytes. So as you can see with the red alpha-actinin staining and the green nucleus signifying that it's coming from one of these CDX2 cells. And this is an unmistakable marker of a mature differentiated cardiomyocyte. And she also showed that they're basically forming blood vessels, because markers of endothelial cells and smooth muscle actin. So she had shown that they are differentiating in vitro to both cardiomyocytes, and these were beating. I'll get to that in a minute. They're embedding inside the heart as well, and forming mature cardiomyocytes in the heart. She had given 1 million of these into the tail veins of male mice. So think about this. She's taking them from female placentas, and we use male mice in this experiment to show that they could also repair male hearts. So we would take wild-type males, infarct them, and then a week later, we would give 1 million cells injected in the vein, so intravenous injection, and they're homing right to the injured heart and repairing the heart. And she had done this experiment so rigorously. She used 2 kinds of controls. She used CDX2 cells, okay, and showed this increase in EF post-treatment at 1 month and post-treatment at 3 months. She used placenta cells that were CDX2 negative, and they did not home. She did not get any improvement in ejection fraction, and she used a PBS control, which there was no significant difference in the ejection fraction. And the ejection fractions were measured by MRI, which is the gold standard. I find it very hard to make sense of data in mice that's collected through echocardiography, because I feel like it's subject to a lot of artifact, but when you use MRI, it's incredibly concise and accurate. So this work was published in 2019, showing that these cells could be taken from female placentas given to male mice, restore heart function, and create new cardiomyocytes, both in vitro and in vivo. Okay, so this is just a better picture. We had feeder myocytes in the petri dishes, and as you can see, there's no green nucleus. The green nucleus signifies that it is derived from a CDX2 cell. Okay, and this is a clonal experiment of 1 CDX2 cell. She did 99 well plates and had 1 cell dropped into each well, and these are so many green fluorescent nuclei originating from that 1 cell, and they're all beating in syncytium. All right, so that shows the power of these cells. And this is still the mouse work. Now, obviously, being a clinician and finding these findings in mice, I'm very anxious to develop a human therapy for what will hopefully be an allogeneic cell therapy that you can take the cells from one person's placenta, store them, and give them to other people with heart disease or heart failure. And the nice thing in the mice was that we showed that these cells are immune modulatory, so they do not evoke an immune response in the host. Now, I've been criticized by some people saying that that could just be because you were taking mouse cells and giving them to mice. So now we're working out the immune modulation in human cells, and our transcriptomic analysis shows that they match the immune modulatory properties of the mice cells in that they are very, very primitive. And so they don't have the immune markers that it would take to evoke an immune response in the host cell. So what do you need for an allogeneic cell therapy? You would like for it to be immune modulatory so that it does not evoke an immune response. You would like for it to be clonal so that you can expand it ex vivo, and I'll show you we're able to do that with these cells. Thirdly, ideally, it would be nice to be able to give it intravenously and have it home to the site of injury, and that's what the mouse cells were able to do, and that is what we're now showing with the human human CDX2 cells. So now this is a much bigger project, much more involved. We have the involvement of OBGYN, the involvement of pathology, and these three women from my lab are all involved with the study. Sangeeta, Dr. Jess Wang, who is a postdoctoral fellow in my laboratory, Michaela, who is a graduate student at Columbia University, and she is our clinical research coordinator who's responsible for enrolling patients for the study, because we're also collecting clinical characteristics on the women whose placentas we are isolating cells from. So there's, so far, a prospective enrolled cohort, also de-identified placenta samples from our biorepository. So the studies that I'm going to describe to you have been from 84 donors from with healthy placentas that have been utilized in this study. And so we've shown that CDX2 cells are enriched in the chorionic region of the term placenta. Now, placenta is divided into a maternal contribution and a fetal contribution. We're actually only able to find these cells in the fetal portion of the placenta. But remember, this is an adult organ that is extruded at the time of delivery. It's an almost limitless source because their babies are born every minute. And even the Vatican would be happy because you're not killing embryos. You're using placentas which are routinely discarded in labor and delivery rooms around the world. So we find them in the chorion, and you can see the staining. It's you know, we find a good number of these cells in the chorion. They're not present in the amnion as shown here. And then we used a new lentivirus that the group made that has a CDX2 promoter that drives mCherry to isolate the cells. We recognize this is not what we're going to do when we use this treatment for humans, but this is just to collect the cells for experimental purposes. And now what we've done is we've identified unique cell surface markers on the surfaces of these cells, and we will be using those for FACS sorting when we try to do, you know, hopefully a clinical trial. By the end of the year is when we hope to start. So that's why we had gone ahead and identified unique cell surface markers. But first, we had to use this lentivirus. So that took a couple of tries to get it right. And now we showed that when we do isolate them, they are basically exhibiting the markers of CDX2. So it's not just the lentivirus. We've confirmed with sequencing, and we've confirmed with antibody staining that these cells indeed are CDX2 positive. They also show other markers of CDX2 cells, cytokeratin. This is human CDX2 antibody, cytokeratin 7. So we've sort of, as I said, the rigor is there. We've shown many ways that these are indeed CDX2 cells. And we show through RNA-seq that they exhibit genes that are enriched for cardiovascular commitment and vascular commitment. So we can see sort of some highly expressed genes involved in cardiogenesis and in vascular development here. So within day 5, they're performing beating organoids, and by day 21, they're actually beating in syncytium with other cardiomyocytes. This is spontaneous beating. This is not provoked or stimulated or anything like that. Okay, so that's by day 5. So this is even more robust than what we see in mouse. And so by day 21, they're forming these networks of beating cells. And these are all human CDX2-derived cardiomyocytes. Okay, and then you see the beautiful actinin staining when Sangeetha stains them for the actinin, the railroad tracking. That's there in the GFP positive, I'm sorry, the lentiviral M-cherry positive cells. And she's using CDX2 human antibody as well to confirm that. And she has a very high percentage of CDX2 M-cherry derived cardiomyocytes at 4 weeks. And she has shown they differentiate into endothelial cells using the CD31 marker. And not only has she shown that, they actually display LDL uptake using LDL uptake assays. So they're functional endothelial cells. There's another movie here where she puts them in Matrigel and they form these beautiful endothelial networks. All right, and then she's shown that they're clonal in that she can isolate 50,000 cells and put them in a clonal assay, and after 6 passages, she gets over 1 million cells. So they're expandable as well. So conclusions, this is initial paper characterizing the human CDX2 cells is now in submission. They're enriched in the term chorionic region. They contain key gene signatures for cardiac mesoderm commitment, and they're spontaneously contracting in organoid clusters as early as 5 days and form cardiomyocytes with a developed sarcomeric structure. They're also angiogenic and form functional endothelial cells. So we believe this is a novel multipotent cell source that is easily accessible, and I say that now, but it was challenging to isolate the first couple patients, but now we're sort of on a roll. And they're ethically feasible to design new therapeutic strategies for heart repair. And this is the overall game plan. What we're planning to do first is to give this to congenital heart disease patients that are born at Mount Sinai and maybe other institutions, because what we will like to do firstly is do an autologous cell therapy clinical trial where we take them from the same placenta and give them to the same babies such as born with single ventricle. So that's sort of probably the earliest clinical trial we're planning to get underway, hopefully with support from the NIH. And then we would like to develop an allogeneic strategy for adult human heart repair as well. So I'd like to thank my laboratory. We have a really hardworking and very cohesive group, I have to say. Past members that have contributed to this work also, including several authors on the key papers, Amarish Ranjan, Scott Shapiro, Richard Chang, Paula Bully, Jerry Saunders, and Jesse Yoon. And I want to thank the collaborators at Mount Sinai, particularly Rhoda Sperling, who is a dean of one of the associate deans at Sinai, and she is also the vice chair for research of OBGYN. We're learning a lot of immunology from Nina Bardwaj. And then my collaborators, another maternal fetal medicine, Sherry Gelber, pathology, as well as other cardiologists who've helped us with the imaging. And we have a collaboration with Cedars-Sinai to do the proteomics, and here are my funding sources. I want to lastly thank my key mentors through my career, Dr. Judy Swain. She introduced transgenesis to cardiovascular medicine, and then Dr. Deborah Wolgemuth, who taught me all the science that I know. Thank you very much. I hope I didn't go too long.
Thank you very much, Dr. Chaudhry.
Thank you.
Thank you. We're off to a great start. Next, I would like to invite Dr. Kyle Kaniecki, Vice President of Life Sciences and Healthcare at New York City Economic Development Corporation. The title of Kyle's talk is Life Sci NYC, an investment in New York City's life sciences. Kyle?
Good morning, everyone. Thank you so much for that introduction. As mentioned, my name is Kyle Kaniecki, and a little bit about my background is that I am a scientist by training. I did my undergrad in zoology at UW-Madison. I have a master's in drug discovery from Johns Hopkins and a PhD in genetics and development from Columbia University. I actually know Deborah Volgemuth very well. We've had many conversations, so small world. And I've also spent many years in biotech R&D, tech transfer at Columbia Tech Ventures, and was an entrepreneur for many years in VC-backed clinical stage pharmaceutical companies here in New York City. And so a question that I get really often is, what is a scientist like me doing working for the local government? And what I can say is that I love this city. I'm in love with New York City. I don't come from here. I'm actually from a tiny little speck of a town in rural Wisconsin, more cows than people kind of place. And I always dreamed of growing up and living in the proverbial capital of the world. And so when I had an option to do my PhD, obviously Columbia University in the city of New York was very much at the top of my list to be able to fulfill that goal. And so I worked in Dr. Eric Green's lab, did a lot of publishing, and after graduating, I knew that I wanted to work a little more on the translational side of science. And I ran into a problem that I think a lot of people here, a lot of graduate students, postdocs, and faculty run into here in New York City back, a few years ago, is that we really didn't have very much of a thriving biotech ecosystem here. We have a ton of basic science, but when it came to the commercial side, it was really, San Francisco or Boston that really captured most of the market. And so I really wanted to work to change something like that. And I took a job at New York City Economic Development Corporation to really turn that around and offer jobs to the fantastic basic scientists here in New York City. So, this is me. All right, so with that, I'll start by saying a little bit about New York City Economic Development Corporation. For those of you who may not be familiar with us, we are a nonprofit quasi-governmental organization. We are the city's primary engine for economic development. We implement policy and initiatives, programmatic initiatives that address current issues that are faced by industry to promote good jobs, entrepreneurship, and economic diversification across the 5 boroughs. We manage approximately 200 buildings across the city that amounts to about 60 million square feet of real estate, as well as non-traditional assets, including developing the Cornell Tech campus on Roosevelt Island, the High Line, and the New York City ferry system. We provide the physical infrastructure, the facilities, programming, and amenities that strengthen the city's competitive position and provide discretionary financial benefits to private businesses and nonprofits chosen to support investment, job creation, and equitable growth across the 5 boroughs. So, if you can't already tell, I kind of love telling stories, so let me tell another story here. I want to take you back in a time machine to the recent global crisis. You might be asking which one, and I'm talking about the 2008 global financial crisis. That's when a lot of jobs were lost here in New York City, and all of a sudden, many of the office high-rises here cleared out and they were empty. And so we fortunately had a very smart local government that realized that diversification of New York City assets would actually make this incredible city that I and so many other people love so much more resilient to future economic shocks. And so while a number of initiatives were really investigated to see where we should really be intelligently placing our investments, obviously life sciences really bubbled to the top, mainly because we have all the necessary ingredients for a thriving biotech ecosystem already. We have 7,000 graduate students, postdocs, coming from an extensive and prestigious network of academic and medical institutions. New York Institute of Technology is absolutely one of them. We have over 500,000 healthcare workers, the highest concentration of Nobel laureates, the most NIH funding for any US metro, and we have investment institutions like venture capital, investment banks, and Wall Street, All of these things would lend themselves to creating a thriving biotech ecosystem. However, we never actually had it. And the reason really why was because we didn't have the infrastructure necessary. And so when a company would spin out of a place like New York Institute of Technology, out of Columbia, NYU, Rockefeller, Mount Sinai, all of these wonderful institutions, they wouldn't stay here in New York. They would go to Boston. They would go to San Francisco. And so in 2016, the mayor's office pledged $500 million to be able to grow the life sciences industry here. And places like the Alexandria Center for Life Sciences, which we're going to hear about later today, the Harlem Biospace, Biobad, and some initial talent programming started to come online and really lay that strong foundational groundwork. But of course, people probably know what happened next. There was a global crisis. Again, which one? This time I'm actually talking about, unfortunately, the global pandemic that had us locked in our homes for many years to be able to protect our family, our friends, and our neighbors. All of a sudden, work from home was a thing, and once again, our office buildings cleared out. So once again, we had really brilliant people in our local government that realized this issue. And not only can you, and they realized that you can't really do wet lab work from home. At least it's not really advised, I would say. OSHA or EHS probably would have something to say about doing wet lab experiments in your living room. And so the city once again doubled down on their investment and pledged another $500 million. So we now have a $1 billion initiative to be able to grow the life sciences industry here in New York City. And with our partners at ESD, Empire State Development, which is the state counterpart to New York City Economic Development Corporation, they pledged $600 million. We now have more than $1.6 million available to really put in place the infrastructure necessary. Sorry, yes, billion with a B. It's always fun to say. So with these investments, and very much like the story of the regeneration of the human heart that Dr. Chowdhury just told us about, we are working to actually regenerate the beating heart of this city. So what are we doing with all that money? It's a lot of money. And so we had to set some pretty lofty goals for ourselves. And so with a vision of harnessing the full potential of New York City's innovative science, diverse talent, and cross-sector strengths, we aim to solve some of humanity's most pressing biological issues. And to do this, what we're doing is we want to establish a hub and spokes model of infrastructure that will span 10 million square feet of real estate. We will grow the life sciences workforce to 40,000 high-quality, good-paying jobs. And we want to create a pipeline of 1,000 biotech companies in all stages of the life cycle. And we plan to do all of this by 2030, which is not that far away. So we have quite a bit of work that we need to do. I apologize for this slide because it's probably a little bit hard to see. But what I really want to tell you about is the graph on the left is the total VC funding for life sciences in the US, which has been steadily growing up until just this last year when we experienced, honestly, what I would have considered a healthy correction to a little bit of an over-exuberant market. So in fact, VC funding in New York City, I don't know why it is doing this, sorry. In VC funding in New York City in 2011 was...
Just close this maybe to keep the space bar maybe.
Thank you. In 2011, VC funding for life science companies here in New York City was $100 million. In 2021, it was $1.4 billion. That's over a 30% compound annual growth rate, which is pretty outstanding. When it comes to our goal of offering 10 million square feet of wet lab and office space for commercial endeavors, we've nearly tripled what we have available from 1.1 million square feet in 2016 to over 3 million square feet now just this last year, with a vision to about 7 million by 2028 in planned projects. And there are a number of key tenants who are beginning to take these funds and taking advantage of this increase in infrastructure and growing lab space, and including just a few of them that are listed here. So this is another slide that might be a little bit hard to read, but I absolutely refuse to apologize for it because I'm actually really proud of what we've been doing so far with this initiative. So Mayor Adams announced in his State of the City a couple of years ago that we were establishing a hub and spokes model. And for better or worse, New York City is very much a highly developed urban area. We don't have acres upon acres like maybe North Carolina or Texas do. So we are starting to see clusters emerge in various neighborhoods. Places like Kips Bay, Hudson Square, West Harlem, and even right here in Midtown West are starting to not only just emerge, but really thrive in the biotech ecosystem. And while Manhattan offers proximity to a lot of our premier academic and medical institutions to be able to cater to a lot of the earliest stages of the ventures, the outer boroughs are also finding their place in the pipeline so that when a company succeeds at an incubator in Manhattan, they can then grow into a lot more space in places like Morris Park, the Brooklyn Navy Yard, or Long Island City. So really, New York City can offer the solutions to the entire life cycle of a successful biotech company from start to end. So out of the 4 tactical drivers that are listed here, I wanted to actually highlight a couple of the initiatives that we are working on. We are ramping up our support for translational R&D to be able to better leverage innovations from the prestigious academic medical institutions here in New York City. We recently announced a $20 million investment into the Brooklyn Navy Yard to develop a first-of-its-kind incubator specializing in biosustainability that could provide novel solutions to climate change. We are looking to expand our network of STEAM centers to educate high school students in life sciences to enable them to take advantage of these investments in an equitable way. We are hosting internships that give college students, even here at New York Institute of Technology, the necessary skills to be able to succeed in the sector. And we do things like this, like what I'm doing right now. We speak to rooms full of people, we publish articles, we travel to conferences, and we all promote New York City as the place to be for biotech. So personally, I always love to have a couple of concrete examples to describe work. And so I wanted to actually highlight a couple of very recent examples, including one that's just happening a couple of blocks away on West 57th Street, where we invested $11.6 million with Mount Sinai to develop the Comprehensive Center for Surgical Innovation. It's really exciting work. I'm very excited. The construction has already started, in fact. With Empire State Development, Janus Properties, and leadership from City College of New York, we invested $15 million in the City Innovation Collaborative, or SYNC, an incubator in West Harlem where engineering and life sciences will cross-pollinate to create the next wave of deep technology innovations. And one of our most successful programs to date is the LifeSci NYC internship program that has placed more than 550 students across 120 companies since 2017. And with 57% of those students being female, more than half being TAP or Pell recipients, and 70% identifying as minorities, we are really aiming to unlock the New York City biotech sector to anyone, no matter your background. We want equitable development for everyone. And so with that, I want to thank everyone for your attention. I want to thank New York Institute of Technology for having me, and thank you to Carter, DeLuca, and Farrell for hosting, as well as to the sponsors of today's conference. If you want more information, I'd encourage you to visit our website, lifesci.nyc. Definitely sign up for our mailing list. We won't spam you. It's once a quarter, but it will provide you with all the key events here in New York City. Or if you are an entrepreneur looking for business incentives, I would encourage you to go to that unnecessarily long link. Or you can just come and find me at the networking sessions afterwards. Thank you so much.
Thank you so much, Kyle, for that talk. Kyle, we would love to take some of that $1.5 billion off of your hands. We will talk after this session today.
You'd love to give it away.
And also I wanted to mention, I should have briefly, Martin, please, because I just wanted to mention something I should have mentioned in my opening remarks. for everybody's information, this event is actually being live streamed at the same time to 100 or so other attendees that are joining us remotely. So even though we have a full room in here, we have many, many more attendees joining us remotely. So it is time now for our next speaker. So it is my great pleasure to invite Dr. Martin Gerdes, professor and chair of the Department of Biomedical Sciences at our own NYIT College of Osteopathic Medicine. The title of Martin's talk is Cardiac Regeneration, Replicating Fundamental Properties of Adult Mammalian talking to an engineer in here, I'm sorry. Myocardium will be changing. What are we up against? So, Martin, floor is yours. Thank you.
Thank you. I'm glad to be here. I really enjoyed Dr. Chaudhry's talk, and mine will be somewhat of an expanded introduction to her talk. She did mention Dr. Piero Anversa, and I think I would like, I'm going to read this abstract out loud because this really rocked the field. This was the worst scandal in medical research history. The retraction of over 30 falsified studies by Anversa has had a disheartening impact on the cardiac cell therapeutic field. The premise of heart muscle regeneration by the transdifferentiation of bone marrow cells or putative adult resident cardiac progenitors has been largely disproven. Over the past 18 years, a generation of physicians and scientists have lost years chasing those studies, and patients have placed at risk with very little scientific grounding. Funding agencies invested hundreds of millions of dollars in irreproducible work, and both academic institutions and the scientific community ignored troubling signals over the decade of questionable work. So this is a really interesting paper that outlines the whole story. I was actually the first one to speak out about Piero Anversa's quality of data. And at one scientific meeting, cardiology meeting, it was a point-counterpoint between Anversa and me, arguing the point of whether adult heart muscle has the potential for proliferating, and he didn't show up, which is interesting. So what I'm going to do is just kind of go over some of the things, the obstacles we have to do to overcome to reproduce, an injured heart with regenerative medicine. So before birth, cardiac myocytes are dividing only. There's not any enlargement. They're all about the same size, around 1,500 cubic microns in size. In adult hearts, the myocytes are about 30,000 cubic microns, and there's a pretty considerable range in their size from about 5,000 to 80,000 cubic microns. But the mean myocyte size and shape is virtually identical in all adult mammals. If I were to put a drop of isolated myocytes from a human heart next to one from a mouse heart, you would not be able to tell the difference.
So have you,
the average size is about the same.
I saw your question on
so there are some subtle
chat.
differences largely related to metabolism.
Are you
so this means
still
that
trying to figure out
rat
how
heart has
you
about
can present
25
from your
million
computer?
myocytes, and a human heart has about 4.5 billion myocytes.
Okay.
So the myocytes are pre-programmed
ultimately,
for
they're
a
going to
certain
give
number of cell
us an
cycles based
option
on the eventual size of the animal.
share
That's
screen.
important to realize because
so
what
if
that
you
really
look
means
on
is
your
there's a limit. The heart knows how many.
Zoom
Somehow
down
the
at
heart
the
knows how
bottom
many
in the middle, there
myocytes
should be a green
that
button
it
that
should
says
have
share screen.
based on the
I know
the genetics of
I
the
sent
kid.
also
So
slides.
elephant heart's going to have even more, of course.
Yeah,
So
that I think is
those
how they're going
myocytes
to
transition
plan
from
on doing it
hyperplasia
is
to hypertrophy.
when you go, you've got to share your
after
screen,
that
you'll do the
transition,
presentation, then
myocytes
you'll
increase
stop your
about
share,
20-fold,
and then I'll go and do
this is
that.
proportional to the heart
At least that's
growth.
what it looks like to me.
After that point, the increase of the heart mass is due
I hope
to
I'm helpful.
growth of blood vessels too, of course, but all
Regardless, pleasure
proportional
meeting
to
you.
myocyte size.
Pleasure meeting you as well.
So this is a paper that we published in 1996, and I would like to point out, so basically we looked at, my PhD student, Fai-Chien Li, was able to isolate heart muscle cells from newborn mouse heart, cannulating the aortas. That was pretty tricky at the time. And he looked at several different time points, 1 day after birth up until 12 days after birth. And as you can see, the myocyte volume was the same before this time point. There's something happening between day 3 and day 4. And you can see all the myocytes are pretty much mononucleated. They have one nuclei at this point. And in the the number of myocytes is pretty much doubles from about birth till about day 3. The number of cardiac myocytes in the heart doubles. After that point, there's no more proliferation of myocytes. If you follow that all the way through to adulthood, you would have the same number of myocytes in those hearts. So clearly there's some trigger that goes on here from day 3 to day 4 that triggers the cessation of cell proliferation, but not nuclear division. So the idea here is if you look before this point and after this point, you could look at the molecular basis of cell proliferation, and then you could look at the molecular basis of cytokinesis, the cessation of cytokinesis and multinucleation. Then you get up to this point right here, and this is the adult binucleation pattern at 90% in mouse. In rats and mice are about 90%. Humans are about 50% have 2 nuclei. So this is the adult nucleation pattern. So again, you could actually then look at this point from here to here and determine what is shutting down the nuclear proliferation, what factors. So it took quite a few years before people followed up on that. Here's on the left, you see myocytes from 2-day-old newborn. These are isolated myocytes. And you can see they have a bulging at the center where the nucleus is. They're really tiny, but they have a full complement of cross striations, the sarcomeres. And if you look at the right, these are isolated myocytes from an adult heart. And this is about the size difference. And you can see the adult myocytes are massively larger, and they are much more complicated, but they all have cross striations similarly. So it would be pretty difficult for a cell that big to divide, because what happens is this shows the difference between day 2 and day 4. If you can see here, at day 2, this is when myocytes are in the hyperplastic mode. You can see there's the cleavage furrow forming here, and this cell is going to divide. And you notice that there's no myofibrils, and that myofibrils had de-differentiated. And if you look at day 4, which is after myocytes have lost their ability to divide, you'll see this is a binucleated cell that just divided, but the one right above it, you see the cleavage furrow trying to form, but the sarcomeres do not de-differentiate, and the sarcomeres actually prevent the cell from dividing, and this becomes a binucleated cell. So it took a while for someone to follow up on this, but Eric Olson published a really interesting paper in Science. It's actually about 15 years later. It took that long for the technology to catch up. What he basically did was he took advantage of looking at injury to the heart before and after that transition point. So he looked at myocytes here at 1 day of age. They cut off the apex of the heart, and then they followed those animals up to day 21. And by the time you got to day 21, you couldn't even tell any difference, that there was any injury. The hearts were able to completely replace all the lost myocytes from as long as it happened before that transition point at day 1, and there was no overgrowth. So somehow this heart knew how to replace exactly the right number of myocytes is what it did. So it didn't overgrow. Then if you did this at day 7, after the heart has lost its ability for hyperplasia, you see something quite different. At the end of that period of time, you see that there's a large fibrous scar. So there was no myocyte proliferation if you did this after that transition point. So to summarize, before the transition from myocyte hyperplasia to hypertrophy, injury leads to cardiac repair, which includes myocyte regeneration. But after the transition, the injured area is replaced by scar tissue. There's no myocyte proliferation. So the heart has a memory. Injured area replaced precisely without overgrowth. Investigation of the transition point should be able to, this has been, the reason why this paper is cited about 1,000 times now, is this gave the molecular people a tool to differentiate just a couple of days difference. They could look at the molecular signaling going on before and after those processes take place. And I think you could see that the adult myocyte, it would be very difficult for that cell for all the sarcomeres to dissolve, dedifferentiate, and actually get a cleavage further. It's impossible. So you only have myocyte proliferation in these tiny cells. You don't, the larger cells, it's just too difficult for them to actually perform that. So some type of stem cell would be necessary. So we found, huh? Right. That was a really fantastic talk, by the way. So myocyte shape matters. We looked at myocyte shape very comprehensively in my early career before remodeling became very popular, and just about every kind of model of heart failure I could get my hands on. We were the first lab in the world to isolate high-quality human myocytes from the heart transplant program. And what we found basically, this is interesting because all adult mammalian hearts, even though there's a big difference in the range of size, the shape is, the length-width ratio is virtually identical in all adult cardiac, in all mammals. The average length-width ratio is around 7 or 8. With dilated heart failure, the myocytes elongate, but they don't increase their diameter. They add more series sarcomeres, and this is what underlies the change that happens in the ventricle. With the ventricle in dilated heart failure, you don't get a change in wall thickness, but you get a dilatation because the myocytes are lengthening. And what this does is it increases the wall stress. And why is that bad? Well, wall stress increase, particularly in diastole, is bad because the heart gets all of its blood supply during the relaxation phase. So if you increase the stress during relaxation, you're going to impede the blood flow. So this is a bad thing. So I guess the question is myocyte shape relevant in regeneration? So a few years later, I got a call from Kit Parker from Harvard. He was really excited because I proved your theory. No, and he told me what he did. I said, no, you didn't prove my theory. You just expanded in a very interesting direction. So Kit developed a way to grow heart muscle cells in culture in all different kinds of shapes. One of his papers, he had star-shaped myocytes. And then he was looking at the calcium waveform, the calcium handling, the contractile properties. And when he cultured them at the normal adult myocyte shape, the normal adult myocyte shape of around 7 or 8 to 1, and compared those to myocytes that had the heart failure phenotype, over 10 or so is the heart failure phenotype, really much higher length-width ratio. And you looked at the calcium handling activities here, and you can see if the myocytes were in that normal range here, they're over here, the blue line, but if they had that heart failure phenotype, there was a very big transformation in the calcium waveform. And this is interesting to me because we know that enlarged hearts are more prone to arrhythmias, and certainly dilated heart failure hearts are much more prone to arrhythmias. And what Kit had actually shown is that with that shape change, you have a pro-arrhythmic heart. The myocytes themselves are able to promote more arrhythmias, just the shape. So I guess the point there, as I'm making, is that the shape does matter. If we're going to try to replicate this, the shape is probably an important feature. And another feature is the heart muscle is really highly vascularized. This is actually a cross-section of rat heart, but it could be human heart too. And the myocytes and the capillaries are running out of the screen towards you. They're cylinders, they're cut in cross-section here. And if heart muscle is fixed properly, it's very difficult to see the boundaries of the heart muscle cells. Oftentimes you see the boundaries very easily because of tissue shrinkage artifact. That's really typical in almost all the histology books. But you can see the myocytes are the darker colored structures here, and it's very difficult to see the boundaries. Some places you can. And the capillaries are interspersed there. But in the adult heart, there's about 1 capillary for each myocyte. So if you're trying to regenerate the heart, you can see if you're trying to add layer after layer after layer, you really need to make sure that you have the capillaries growing along with it. So that's a pretty important challenge to make sure the vascular supply, if you're going to get any kind of wall thickening at all with regeneration, you need to make sure the vessels are there. So in conclusions, it's unlikely that myocytes can revert from cell growth to cell proliferation mode. The number of myocytes in the heart is genetically predetermined. Adult myocytes are similar in size in all mammals. implanted cells will need to couple with existing myocytes for optimum function. That's another thing, is when you get myocytes to grow in there, they really need to couple with the existing myocytes in the native heart so that they can all be electrically coupled. And we do know that length-width ratio of the myocytes is important because it affects calcium handling and can promote arrhythmias if it's adverse. So the question, will induced myocyte proliferation lead to tumor growth? That's another question. And there's actually some studies that kind of show that. So if you're trying to stimulate hyperplasia of heart muscle cells, some of the things that you do might actually cause cancerous growth. So that's another issue you've got to deal with too in this field. And certainly the blood supply is an issue. But I don't think we should lose hope. The previous talk was really, really encouraging that there's a lot more important things on the horizon. And I'm really excited about this field. So I thought what I'd do is based on what I've said and some of the background that Dr. Chaudhry gave too, let's pretend that a paper was just published in the New York Times, and you want to try to determine whether that paper does it make sense? Could you believe it or not? Because there's, as you know, from Dr. Anversa's work, there's a lot of stuff out there that's not good. Say this is a report in the New York Times, and they insert a plug of adult pig heart into a damaged myocardial area of a patient. And they're claiming that there's a dramatic improvement 1 month later in heart function. Some of the questions you might ask, well, you realize that myocyte size and shape of an implanted pig heart, adult pig heart, would probably be about identical to the human heart. So we've got that right. So that might be feasible. And if you look at a biopsy of that implanted heart tissue later on, if you see, the myocytes have a full complement of contractile filaments. And the next point here, you don't see a lot evidence of rejection. That would be white blood cells. If you just look at it in the microscope, you see it's loaded with white blood cells. That means it's having an acute rejection. One point I should make that I think is really exciting is there is a lot of progress being made on xenotransplantation of pig hearts into humans. They actually had tried that. The patient, I think, lived for about a month or so. But if we can get past the rejection component, we have an unlimited supply of pig hearts to transplant into people, but we've got to get past the cardiac rejection component. So that's, to me, is another exciting area. So, you look at myocytes and see if they're normal or they're degenerating. Oftentimes you'll see a dramatic loss of contractile filaments. So that indicates that the myocytes are not very healthy. Again, you want to look for evidence of revascularization. And how could that be done? Well, maybe, you read the methods, and it said they connected the pig's arteries and veins to the human's arteries and veins before they sewed it into that area. That's feasible. We have microsurgeons that can do that. So that's the kind of thing you would look for when you're trying to evaluate the paper. And is there evidence that the implanted myocytes are coupled with native myocytes, allowing synchronous electrical conduction and contraction? So anyway, I thought I'd just give you, I realize this audience is not cardiovascular, so hopefully this additional information, an expanded introduction of Dr. Chaudhry will be helpful for you to understand the field. Thank you.
We've had 3 wonderful talks so far. Next, we have a panel, and the panel's topic is on startups. So as you could imagine, there are some considerations that are universal to any startup, to any tech startup. But there is specific considerations and issues to be aware of when it comes to biotech startups. So today we have 4 panelists, one of which is here with us in person, and 3 are joining us remotely. So I ask our panelist who is in person, David, to please come to the podium and take one of the seats. And I will, in a minute, introduce our 3 panelists. Since we have really, if you could, you want to take a seat, or you want to? And since we have 4 panelists with, that they have a lot to offer within the limited time frame that we have, rather than doing it the Q&A, a traditional Q&A style of a panel, I'm going to ask each of the speakers to take over, present their thoughts, and then we move on to the next and to the next until our time slot is finished. So let me make it look like a panel now. So I sit in here. All right. So I will introduce the four panelists, and then in the order that they're being introduced, they will present their thoughts. Our first panelist is Mr. David Lewandowski. He's the Director of Business Development, Cell and Gene Therapy at Azenta Life Sciences. And David is joining us from Boston. And that was to Kyle's point. So, hopefully we will have startups, staying here in New York with us. Our next panelist is Mr. Philippos Touloumousis. He's the founder of Biological Lattice Industries. We also have on our panel Mr. Ross O. Youngs, CEO and founder of BioSortia Microbiomics. And our fourth panelist, who's also joining us remotely, is Mr. Paul Marshall, Chief Executive Officer and co-founder of Rapid Fluidics Limited. So without any further ado, I pass the floor over to David for his presentation. There we go.
Okay, so first off, I'd like to just say thank you to Dr. Beheshti, President Foley, who was just over here, George, Jill. They also helped out with many others at New York Institute of Technology for this special day. As mentioned, my name is Dave Lewandowski. I work with Azenta Life Sciences. I'm not a scientist, so we're going to step out of the very technical discussions for just a 10-minute presentation. We'll have just three areas: the introduction, and I'll talk a little bit about drug delivery today, and opportunities in cryogenic infrastructure to help deliver the future of therapies of tomorrow. So this is kind of my background in, I've been in life sciences for 25 years. I've worked with many of these products that you see up here. But just for background, right, when we think about small molecule drugs, pills that we take, right, those are pretty easy to manufacture once you get the right recipe to manufacture them and distribute them, because everything's at room temperature. It's very easy. In about 2002, I worked with this product called Xolair. It's a monoclonal antibody, and it was called omalizumab, and it was used for severe asthmatics. And when I would go to pulmonology offices and the allergy offices, it was quite amazing when we switched modalities, new treatment modalities, the time it takes to get an office to adopt and uptake and really try to identify those patients that can benefit from such a therapy. In this case, it just needed to be stored at 2 to 8, but it was a repeated, ongoing monthly therapy. And so there was an involved administration, more so than just giving a prescription for pills. I think everyone is quite aware of what happened when COVID happened, and we heard about the distribution challenges with the COVID vaccine. We also see that gene therapies are in this area of -20 to -80. So if you think about it, a home freezer is about -20, dry ice is -80. And so those temperatures, and they're really critical to maintain shelf life and integrity of those products. But we take it a step further, and we start talking about cell therapies or organs and distribution of organs. And we have to really ask ourselves, how are we going to get those products to the patients? The rest of my presentation will really zone in on this cryobiology and the cryobiology necessary for delivery. So Emily Whitehead, pictured here, is patient 0 for CAR T therapy. She just celebrated 10 years of being cancer-free. It's really amazing. This is Dr. Grupp, who was the actual physician on site at CHOP that squirted the cells into Emily. I was traveling with both of them last summer, and we attended a panel presentation. It was a company that was making a TIL therapy. And Dr. Grupp very bluntly said to them, the worst thing that could happen to you right now is you get an approval without the infrastructure to distribute it. And what he was saying was that you have this great therapy that's going to maybe go and take out solid tumors. And there's going to be a lot of people that want that, right? But clinical trials and cell therapy start out very small, maybe 10 patients in a preclinical or a phase 1 trial, and they might grow to 40, 50, 60 patients in a phase 3, phase 4. Once you get a BLA, you have to scale to thousands, and that's where the challenge occurs. So what do I mean by that? It's been studied a lot in cryobiology and hematology that, once you get cells frozen, it's great. You can maintain that shelf life pretty much indefinitely. But when, and as a matter of fact, about 80% of the current INDs in this space are using some sort of frozen or cryopreserved cells. whether it be in the research, discovery phase, or the actual scale-up of manufacturing. Not paying close attention to that, or having cells that go through freeze-thaw cycling or thermal cycling, can cause great damage to the cells and probably impact the potency of your therapy. Can be great side effects as well. So when we think about cryopreservation considerations, really there are just 5 steps, which might be modified slightly depending on the cell type, the volume of cells, and the container that they're going into. But cryopreservation is really important for intermediate materials, final product for distribution. It's allowing people to do the manufacturing so that you get the scheduling flexibility. And there's also a number of other ways that cryobiology is really, really important. So as we think about it, and we think about all of the different considerations for cryopreservation that are listed here on the screen, we have to ask ourselves, is this scalable? Is it scalable to have the human intervention at many points across the manufacturing and distribution chain? And is that opportunity for more variables to be introduced to an already very challenging manufacturing and distribution process? So how can we make it easier? Standards in secondary packaging, I would highly suggest, and I'm a huge advocate for it. This is currently, these canisters, these metal cassettes that you see would be typical for what CAR T therapies like Kite or Novartis might be distributing a CAR T therapy in. But today you can see that there's no standard dimension. This really causes a challenge for the cryobiology aspects of it, but also the distribution, because the end customer is actually the hospital that's going to receive this and dose it to the patient. If you ship them something and they don't have an inventory system or a way to document or track that, it causes a lot of problems. And here you can just see that each individual investigator is really perhaps building their own bespoke model based on the dosing regimen that they're thinking of. But I see this as a great opportunity for our industry to come together and build a standard that everyone can latch onto. You know, things like this are non-IP related. This is just about building an infrastructure so that people and physicians and these discoverers can get the therapies to the patients that need them. So, exciting development. Last October, we had a cryopreservation, a symposium. 15 presentations from world leaders that are, building out their processes, whether it be at BMS or Novartis. We had great industry leadership there represented. Standards coordinating body kind of gave the keynote at the end. And what they wanted to do was develop a call to action around standards for cryobiology. And so they identified 2 areas, one in secondary packaging, the other in the bioprocessing and some of the more, 80% of cryobiology is probably the same, but there's 20% that's a little bit different. So if we could really educate people and the stakeholders on that key 80%, the key considerations, and also provide them some secondary packaging to put in whatever materials they would like, these are 2 great ways that we can help the industry go a little faster. If anybody wants to participate, it's a wide open group, and currently I think we have about 50 or 60 industry representatives on that working group. So as I wrap up, just a little bit about Azenta. We are helping startups scale, and we do it in a lot of different ways, but here, just briefly, genomics and multi-omics portfolio. We do sample sourcing, we do automation, and have GMP storage facilities around the world. We also do some of the other items here listed at the bottom. So key takeaways today. Think about plant-to-patient mentality going forward as a startup, with the new cold chain infrastructure and training as a requirement. Address and understand the temperature control, stability, and tracking for these new advanced therapies. Learn from your care centers about equipment and ability and willingness to partner early on. I've heard from many end users, many hospital settings that, the manufacturers will go to them and ask them, hey, we want access to your patients, and here's our great therapy, and you need to buy this equipment. And they're like, whoa, hold the phone, time out. So I would just say work with them early on and select packaging which achieves the dosing potency as well as simple adoption in the clinical space. With that, I'll wrap it up. Thank you.
Thank you, David. Please join us here. Our next speaker in the panel is Philippos. All right, Philippos, the floor is yours. Go ahead, please.
Great, thank you. Thank you very much for the opportunity to present remotely. Couple of words about me. I'm a mechanical engineer by training with a PhD in mechanical engineering right across the Hudson River at Stephens Institute of Technology in Hoboken, New Jersey, and a couple of years as a postdoc and research scientist at the MIT Media Lab. Biological Lattice Industries is a startup manufacturer of architected biomaterial scaffolds, and we hope to contribute in the regenerative medicine paradigm. We were incorporated 2 years ago in the US during my postdoc, and we have operations in Greece with an R&D and lab setup, backed by venture capital funds like the E14 Fund and a group of angel investors. For mechanical engineers, pretty uncommon for a biotech startup, and I hope I can give a little bit of engineering flavor in this field of in all these talks that we hear about regenerative medicine, and we are currently closing our seed round. So I will assume that not everyone has a background in biomedical engineering or experience in cell culture labs, although I spent 3 years maintaining a BL2 lab facility as a mechanical engineer and learning how to culture cells. So usually everything starts with a bio of cells, and cells are in suspension. They have these circular shapes, and it's obviously everyone is moving them on and plating them in 6, 12, 24, 96 well plates, and the goal is to culture expand them because we need to grow them in number. And as you can see, they adopt different shell shapes, which I believe is a very important topic also that I heard about myocytes, and I got really happy that myocyte shape is something really important for controlling their function and their phenotype. So yet we still use those two-dimensional glass surfaces, but they are increasingly insufficient, as the largest manufacturer also admits. And it's a hot topic right now, also due to the FDA Modernization Act that passed US Senate in September. We have also the FDA Modernization Act number 2 that passed in February, in January. And the goal is to have and move and change the way we do cell culture, trying to have environments that are closer to the human relevant scenario. And at Biological Lattice Industries, we are a group of engineers where we, instead of using chemical processes and gene editing to control cell function in two-dimensional petri dishes, we like to imagine a world where we have machines that design materials, and based on the architecture and the structural cues that we can give to cells that are seated on top of these scaffolds, we can tune single cell shapes, and by this way we can control downstream specific cell functions that are of interest. So basically it's like reversing this dogma of biology and manipulating very reproducibly the microenvironment in order to induce downstream changes to the genome. This field is known as mechanobiology, with very interesting discoveries over the years, but largely has an untapped potential, commercialization potential specifically. So it's very well known that life sciences suffered from reproducibility issues. We see the numbers here from these papers, and a large portion comes from biological reagents, reference materials, but also from laboratory protocols. And in the early days, and this is an example of of this issue, is like this company that after many years of funding, and also this represents this classical approach that if we have access to unlimited capital, we can solve any problem we want. This is an example of being able to actually tackle this issue of reproducibility and consistency when it comes down playing with materials and cells. And so it is very well known also that regenerative medicine suffers from these 3 issues. That's why there are all these interesting companies around trying to solve these 3 issues of cost, scalability, and reproducibility. So at Biological Lattice Industries, we believe that if we take out the regulatory lexicon and think about in terms of the primary mode of action, we believe that the key is that if we plan to design biomaterial therapies, if we do it for a mechanically driven PMOA, we go through a much, convenient, I would say, pathway. Because it's important to notice that, for example, in this paper from Science Translational Medicine and also from FDA's website, we see that you can have, if the primary mode of action of a specific, molecule, in this case, an injectable colloid, comes from its physical properties, this would be considered as a medical device, and it would not go through the drug and biologics regulatory pathway. And the difference in costs are tremendous, as you can see from the numbers in the slides. So we are trying to design and manufacture architected biomaterial therapies that harness the innate regenerative capacity through programmable mechanical driven mode of actions that come purely by the architecture of these scaffolds. Our first product corresponds to the 3D cell culture market. A biomedical engineer, a cell biologist, investigator in the life sciences, if he wants to culture cells in 3D, it has a wide range of tools in his palette. And if we classify them according to geometrical and dimensional features, these products range from nanofiber membranes up to three-dimensional printed scaffolds with conventional 3D printers. So you have the nanoscale on the left side, and on the right side you have 0.5 millimeter lattices in fiber diameters, and in the middle and in the nanoscale you have those hydrogel matrices, natural hydrogels that you can use and embed cells and study cells in 3D. So we come here and we fill in this missing dimensional scale window that is close to the single cell level, from 2 to 50 microns. And we do that by using, by creating robots that allows us to print those scaffolds that have precisely defined architectures down to single to 10 microns. And we do that by using a technique that is inspired by electrospinning. You get those fibers, very thin fibers, by stretching polymer melts. No chemical solvents are involved in the process, like in solution electrospinning, but also because you are working with polymer melts, you have very viscous materials, and by applying this electrical field from the needle tip to a grounded collector plate that is mounted in a robotic stage, you can get, precisely writing of fibers layer by layer. And the process is slow, but and very complex, because there are many parameters involved in the process in order to control and have these precisely defined scaffolds. So we have been developing a lot of automation, and this has allowed us also to decrease the cost of the machine, so we can scale out and we produce them at scale. The two products that we plan to market within the next year are two scaffold architectures that are also protected IP. They have protected IP for use with mesenchymal stromal cells. Most of the work comes from Stevens Institute of Technology, me as a PhD there, working with MSCs and trying to manipulate, the stemness of MSCs by changing their shell shape. And, we have published some work where using fibroblasts as a cell model system, where we show how we can manipulate very, very homogeneously and reproducibly cell shapes down to the focal adhesion level using our scaffolds and using two-dimensional, petri dishes and random nanofiber meshes as controls. And we have established also a machine learning framework to classify and quantify this relationship, how, from the scaffold architecture, we can very precisely tune single cell shapes. There is IP with MSCs where we show by purely changing the architecture of the scaffold, we can manipulate the self-renewal process without any bioactive molecules. Just by tuning the early cell shape phenotype, this has downstream effects on how the cells, self-replicate, and if they lose their stemness, the stemness phenotype. So there is one architecture that allows us to very efficiently culture expand MSCs and very homogeneously without losing their phenotype. So we thought that it's a very interesting opportunity to use those scaffolds, not only as research tools, but also as delivery vehicles of MSCs at trauma sites. And in total, we have a scalable manufacturing process. We are using medical-grade polymer melts that being used in the medical practice, surgical practice, without any chemical solvents involved. We have absolute control of the porous microarchitecture of the scaffold, and also the geometrical feature sizes of each fibers are down to the single cell level. There is a huge opportunity, we believe, and for regenerative medicine. And in the pipeline, we're developing a technology platform that will allow us also to use scaffolds as implantable medical devices. Specifically, we are looking at soft tissue reconstruction, and specifically patients that need to undergo lumpectomy. We are moving more in the breast cancer field, and specifically women, we are moving more from breast mastectomy surgeries and the oncoplastic breast conserving surgery to partial breast mastectomy and oncoplastic breast conserving surgery in one step. So we would like to eliminate autologous fat transfer and be able to have an acellular biomaterial product that implanted and with patient-specific shape and give it to the breast surgeon, who can also initiate implanting and then over time initiate the endogenous cells to proliferate on the scaffold and start depositing good extracellular matrix. So the way we are doing it, we've been creating a very modular technology platform where everything we would like to do starts from the left side. So we know a shape that corresponds, that needs to be filled with a specific microscale architecture. And from this microscale architecture, we are running different search optimization algorithms in order to give us the optimal architecture that corresponds to the mechanical properties of the host tissue. So basically, the goal here is to create a perfect mechanical analog of the host tissue that has the trauma. And in this way, we are trying to eliminate the foreign body reaction mechanism that comes into place whenever you put something foreign inside the body. And everything this happens in a closed loop in silico. And as soon as you arrive at the optimized scaffold architecture, you can start sending it to the fabrication module, which are the robots that can print the scaffold. Thank you very much, and for the opportunity to present. And please reach out if you think there are any synergies using scaffolds for creating new therapies.
So I guess I'll jump right into presenting. I am Ross Youngs, CEO of Biosortia. And what we're doing is we're basically looking at nature, which has small molecules that work throughout biology that have been virtually inaccessible to research. They have created the vast majority of drugs on the shelves, but we've got a new way to get directly at those molecules. That is going to be the most powerful small molecule drug discovery platform that's ever existed. And what makes it so powerful is it will really enhance dramatically machine learning, AI, synthetic chemistry, and synthetic biology. So when you look at what has been successful, quite frankly, about 75% of the small molecule drugs that are on the shelf today have a direct or indirect connection to biology, to plants, to microbes, to animals. And even when you look at synthetic chemistry, synthetic biology, and the newest approaches today, that's been going on for decades, and it really has not ultimately increased the amount of discovery of new molecules or created opportunities to reduce cost in drug discovery at all. So part of what has to happen in the future is we've got to look closer at what's going on with the small molecules in biology in order to have meaningful data. Ultimately, that is where industrial scale microbiome mining comes in. So when we look at nature, it works under a set of certain circumstances for specific results. So certainly you've got the code, the DNA, you've got the coded metabolic pathways, but they have to work in the real world of biology. And in doing so, it's very complex. It's not simple. The molecules that get created by nature are complex and they hit targets. They also are perfect for being a target and also a marker. Reality is 90% of all the drugs on the shelf are small molecules. And also when you look at a microbiome, whether it's a human gut microbiome or any microbiome, it overlaps drugs on the shelf remarkably. So the code, in order to be successful, it must ultimately represent biology. And just executing code does not guarantee that it's going to represent biology. So there is a firewall related to that code. On one side of the firewall, you've got data that's accessible. And ultimately from that data, you want to be able to create a metabolic pathway to create a small molecule that exists in nature. Right now, unless you have some idea of what that small molecule is on the other side of the firewall, you're not producing natural molecules. You might be producing a lot of molecules, but will they work in nature? Not really. On the other side of the firewall, you have the small molecules and you have the activity. Being able to break down this firewall and have these small molecules will accelerate, quite frankly, AI, machine learning, and synthetic approaches. Let's take a second to talk about computing power, because right now we put a lot of weight in what computers can do and what they're going to do in the future. So let's take the fastest computer in the world as of last year, which was the Fugaku, and it had a 1 exaflop calculating speed. This computer takes 160,000 square foot. Let's compare it to what E. coli can calculate just with mutations in 24 hours. Now, this is giving the computer calculating power a tremendous edge because a number of different ways to calculate this showed numbers substantially higher. But using what I would describe as the minimum equality in computing power, you would need to have the state of South Carolina filled with supercomputers to keep up with the computing power of E. coli's mutations in a 24-hour period. That's remarkable, but we've got to remember E. coli is just one species. We've got 4.2 billion years of evolution going on. Nature has made the small molecules that work. If nature wanted a simple small molecule that worked, it would have designed it, it would have kept it. That's one of the challenges we have today. So when you talk about what works in biology, it really is what plants, animals, and microbes, what biology has created. Those numbers are relatively small when you talk about what we found and what the possibility is. It may be the least common denominator in biology. When you talk about synthetic chemistry, when you talk about synthetic biology, and you talk about the possibilities of small molecules, it actually approaches the number of atoms in the universe. And if you're talking about synthetic biology, you're likely approaching infinity. So having the guide of nature is critical to make these other technologies functional and working. Let's also look at it in this way. When you talk about what nature does, it creates a complex small molecule, not simple small molecules. When we look at what is going on in synthetic biology, you can go find libraries that possess more than 1 trillion synthetic biologically created small molecules. They may fit the lock, they may even hold into the lock, but do they work? Well, research is showing that these molecules are more of a diversion of where we're going rather than where we need to go, because the complexity in nature actually forces us to look for molecules that actually will work. And one of the key opportunities that's occurring is improvement in ultimately delivering the small molecule right to where it's needed in biology. That advancement's going to make small molecules from nature even more important in the future. So I'd already talked a little bit about biology's making the molecules that matter. It has the biodiversity, it has the signaling, the chemical diversity, and the high potency. Natural molecules absolutely work. And with the presence of biology on Earth for nearly 4.2 billion years, again, nature has created the molecules that work. When you look at biology, small molecules actually even make up DNA. Small molecules can slice and dice DNA. Small molecules can pass through the blood-brain barrier and pass through cell walls. And when you look in biology, the overlap of small molecules that work in biology is across biology. What works in a human can work in a plant, can work in the microbes. As far as small molecule signaling, nature has used those things, and obviously there are molecules to be found outside of a specific area of biology. And that's where industrial scale microbiome mining comes in, because in biology, microbiomes are pretty consistent. The human microbiome overlaps the aquatic microbiome by a minimum of 73%. So what it boils down to, can we have the technologies to collect microbes so you don't have to culture them? Because less than 1% of the microbes on in the planet can grow in a culture. So the goal is, yes, of course, we've got this at scale, we've got it proven. We look for inspiration from the disease and the microbiome, look for metabolic pathways, look for molecules using our capability to get molecules, ultimately process those molecules so they can be screened by researchers, and ultimately create value in the proposition of moving a molecule forward. So when we're talking about getting it at scale, we've got to realize that typically a lab benchtop is about a 100 milliliter sample. And in a 100 milliliter sample, if you look at the signaling typical in cellular signaling, it's in picomolar, nanomolar concentrations. You can't even read that if you know the molecule at those rates. So what you've got to do is amplify it. So instead of starting with 100 milliliters, we start with 200 million times that. At 200 million times that, when you talk about getting small molecules from unculturable microbes, we're talking about something that's 1 million times more efficient than the next nearest competitor for obtaining these kind of small molecules. That's a major reason why science pivoted to the indirect approaches, because they couldn't get nature's molecules. We're here to say you can get nature's molecules, and these as inspirations to accelerating all other technologies is critical. So this is proven, this is scaled. We have collaborators, we're ready to go at full scale. Our model is to find lots of collaborators because they know what they're doing and we know what we're doing. In 1 hour of running our technology, we can take a 350 molecular weight molecule from a small molecule, from a microbiome, and amplify that to 50 grams. So think about that. That gives people, for the first time, the opportunity to really research molecules at scale, and that's critical. So our business model is to support those 400 research teams to drive value in a variety of ways. So we want to work with those that have needs or trying to find new inspiration for drug discovery across a wide spectrum, not only therapeutics, but targets, markers, agrochemical, and even sustainability. Our whole goal is to amplify the ability of machine learning and AI, which is lacking critical data in order to make decisions, and advance omics. we need in our stable of partners, we need partners, we need collaborators, and we need funding sources. And we're working right now extensively on a number of grant pathways. But what we would ask is that if you have an opportunity to want to learn more, if you want to learn how we can work together, give us a call and let's start a conversation. Look forward to hearing from you. Thank you.
Thank you, Russ. And our fourth panelist, Paul, as soon as we can have Paul joining us, floor is yours.
My name's Paul Marshall. I'm one of the founders of Rapid Fluidics based in Newcastle upon Tyne in the north of England. I'm going to introduce you today to our work in rapid prototyped microfluidic systems. So the development of diagnostic systems has understandably seen a huge rise in recent years. You know, the average man on the street now knows the term PCR, even if they don't know what it means. They know that lab testing can give better results than a lateral flow test, but it's less convenient. They're also coming around to the idea that lab tests don't need to be carried out in a lab, but they can be carried out at the point of need, similar to lateral flow tests. This itself is driving the need to develop easy-use systems, so almost using the Nespresso approach to single-use diagnostics. So as these devices get smaller to reach that point of need, they need to incorporate microfluidics, and rapid fluidics have a technique to produce fully functional prototypes in a matter of hours rather than weeks. And this came about to provide a solution to a genuine unmet need. So I previously led the engineering for a company developing a portable point-of-care molecular diagnostic system. We struggled to make prototype microfluidic cartridges quickly. Neither in-house fabrication or outsourced manufacturing was good enough or quick enough. So through a PhD at Newcastle University, we've developed a method of relatively cheaply 3D printing microfluidic prototypes, offering a 24-hour turnaround of bespoke designs. And this allows our customers to try multiple iterations of early stage designs in a very short space of time, reducing the time and cost of product development. So depending on how much you know about microfluidics, you may or may not know that there's various traditional methods for producing and sealing them. You've got things like CNC machining, hot embossing, and obviously injection molding when you volume. And all of these require secondary bonding, using adhesives, using solvent diffusion bonding, laser, ultrasonic welding, for instance. And they all have their different advantages. So rapid fluidics, we obviously focus on 3D printed microfluidics. So we can produce open channels very simply using our high resolution printers, which could then be enclosed through any of the conventional methods that were listed. We can even produce fully enclosed channels down to around 500 micron diameter. But to get down to near 100 microns, we avoid the time, expense and risk of all of these other techniques by simply 3D printing a lid directly onto the microfluidic layer. So this has got many advantages, the key one being time, but also the lid itself can include additional features. So more channels with vias linking the layers, or external connectors can simply be incorporated from mounting features from manifolds, barb tube fittings, threaded holes. We use SLA and DLP 3D printers and a range of photocuring resins suitable for each application. Our biocompatible resins comply with quality requirements for Class 1, 2A, 2B, and 3 of FDA, ISO 13485 and 10993 for the manufacturing medical devices. We can get dimensional accuracy of about 5% with a print resolution down to 20 microns, surface finish RA value of 0.5 micron on the horizontal and 1 micron on the vertical for the square channel surfaces. So the microfluidic market is fairly small, but there are several companies around the world providing prototyping and production services, each with their own processing niche. While they all have their respective strengths, Rapid Fluidics is unique in offering a very short turnaround. We use slightly modified commercially available 3D printers with much lower overheads to cover. And we know that representatives from the industry are in agreement that offering prototyping as an outsourced service is a growth industry, de-risking product development for our clients. In fact, from our market research, 95% of our customer base are SMEs, favoring low CapEx outsourced prototyping. And the great thing is, because all the other service providers are substantially different, it opens up huge potential for collaborations. So I've got a couple of case studies to go through for you. So we've worked very closely with Chromec in Sedgefield in County Durham since we started in 2020. Chromec's background is actually radiation detection, working with medical, power and defense industries. And through this connection, they're working with DARPA on the system for detection of airborne pathogens, which uses microfluidics to analyze samples. Early on in the development, to translate a particular process to lab on a chip, they had to make small volumes of 4 different liquids using a chain of proprietary components. They simply couldn't do this. So we discussed this process one afternoon in the lab, sketched it on a whiteboard, modeled it in 3D CAD, 3D printed it that evening to be tested on the lab bench the following morning. In this case, it didn't work, but the next one did. We also sent the design out for quotation from other suppliers. Nothing was available within 7 to 8 weeks and at a considerable cost. So Jamie Marsay, their head of biotech, described it as a game changer. After all, if you're in the situation of developing a diagnostic system, trying to detect a virus that's threatening to wipe out half the population of the planet, would you rather test it in 8 weeks or tomorrow? So another case study is a project we carried out for CPI, which is the Centre for Process and Innovation, also in Sedgefield. One of their research groups was optimizing H filtration for particle diffusion between 2 streams of fluid. They had a range of 30 microfluidic chips designed to test, each with unique geometric characteristics. They also needed to be able to include Lure-Lock connections for ease of use and designed to fit under specific imaging equipment. As with Chromec, we were able to produce these individual designs relatively quickly with a full range of 30 unique parts delivered within a week. We also work with academic groups. During the pandemic, a research group at Southampton University weren't able to access their fabrication labs, so we made a batch of flow cells for them. Following this, they realized that using the biology researchers to make plastic parts wasn't the most efficient use of time and money, so we continue to support them. So as well as the more normal customer requests for batches, just 1 off, 5 off, 20 off, we've recently been approached by several companies about inquiring about larger batch productions, volumes up to tens of thousands. Ultimately, if we can make 1, if we can make 10, we can make 100 for data generation, we can make 1,000 for preclinical trials and so on. To allow Rapid Fluidics to scale up to meet this potential market, we're engaging in processes to improve efficiency by automating our manufacturing, developing our own specific and exact materials. We started down this route with an Innovate UK Smart Grant, along with angel investment backing it. We've moved into larger facilities that you can see here. We've just taken on a 3rd engineer. And the next step is to source more people, upgrade our manufacturing equipment, and then with the scaled up production capabilities, we can expand into ever larger markets. So thank you very much for your time, and feel free to get in touch and ask questions later.
Thank you, Paul. And again, thank you all to our panelists.
30 seconds to introduce yourself and some of your history in the area.
So it all started with a phone call. Somebody explained cell and gene therapy to me, and I got excited about it. And we founded, I guess almost a quarter century ago, a company called PCT, which became the first contract manufacturer for cell and gene therapy industry. Today, that was sold. Today, it's part of a conglomerate, a global company called Minaris Regenerative Medicine. So I was one of the co-founders. This is a multidiscipline team, so you always find co-founders. And I was the business guy. So I was the chief financial officer, the development officer, construction officer, coffee maker, et cetera, as you start with in a startup. Eventually, we had several hundred people.
I think you
so that's my background.
I think you described yourself as the CJO, the Chief Janitorial Officer, because you had to clean up all the crap out there, right?
That too. So it's I encourage anyone who thinks has an entrepreneurial spirit to do it, but realize that the job in the initial stages, irrespective of the title, is very vertical. So you can be the chief of something and also the chief, garbage collector.
Exactly. So kind of at the beginning, what was like the original promise when sort of cell and gene therapy became mainstream? What was the promise that it was trying to solve for us?
So this is a relatively new industry, and the initial concept was, today it's more of a reality, that manipulated cells, can become a product to treat patients. So today, even still today, we treat patients with cancer or the autoimmune diseases, in some cases with chemotherapy, which has horrible side effects. So hopefully the next generation, maybe within our lifetime, certainly within your lifetime, your children's lifetime, they're going to look back at that and say, so when you had cancer, you got chemotherapy, that must have been awful. It had terrible side effects, and maybe it wasn't all that good. So the idea of cell and gene therapy is that we take cells that are deficient in some way from a patient. In the current technology, we extract it, we manipulate it, we knock out the bad cells and we reinfuse the corrected cells, maybe with a missing gene. And those will, in effect, to use a non-technical term, they will take, and it will fix the problem inside the patient's body. The promise is fantastic and, in fact, miraculous. The industry has been at it for at least 25 years. There are a few products that have been approved, probably about 5 to the major diseases. We read about it, but we haven't seen massive beneficial results because the big diseases have not yet been conquered. Solid tumors, the cardiovascular diseases, and so on. But there are certain rare diseases for which there is now a cell and gene therapy treatment.
So I guess originally, sort of like the first mainstream company that you would think of in this category would be like a Spark Therapeutics. And of course, they've had their trials and tribulations over time. But I guess how have the expectations of what the technology can bring, how has that changed over time?
So there was a bit of what I would call now in hindsight, we can all be very smart in hindsight, of irrational exuberance. If we can just do this and this, look, it works in mice. And that promise has been around for maybe 25 years. And a massive breakthrough has always been 5 years away. We're still more or less at that point, although there have been some breakthroughs. So, and the industry has been financed. About $20 billion went in 2021, about $12 billion went in 2022. This is just new money. So there's some modulation of expectations, and there are quite a few companies that are close to getting approval. But the key word is close. And there really hasn't been a major breakthrough that would make the front pages of the publications or that would be on the evening news.
So when you think back to like the origination of Spark, so that comes out of UPenn, and we were actually introduced to the company as could we be their outsourced accounting provider. So, okay, sounds good. How many people do you have? How much money do you have? None. So we presented a solution for their outsourced accounting, assuming they had almost nothing. So we present that to the board, and the board says, so we're going to try to go public tomorrow and try to raise like $500 million, even though we have no people and no money yet. So I said, whoa, that's pretty far-fetched and really way out in front of themselves. But it was sort of the promise of what the technology could bring. That was the exciting part about it, right? So the capital markets were super enthusiastic at the beginning of these companies.
There was this enormous exuberance that this thing will displace existing forms of treatment. It will be a better way to treat, and it will be permanent. So that once you fix the cells, you fix the disease. It hasn't quite happened. I don't think today you could go public for $500 million valuation with no people and no accounting system. So I think that's what's different today. But there's still quite a bit of activity. Let me just paint a kind of a global picture for you. There's probably about 1,500 companies around the world that are developing cell and gene therapies. About half the action is in the United States, a quarter is in Europe, and a quarter is the rest of the world, it being Japan, Australia, South Korea. There are about 1,800 clinical trials underway. So if you think about that, and if you try to put that in global perspective, there's only about 10 at most 15 what we call big pharma companies. So you can foresee that there will be massive consolidation here from the 1,800 down to something a lot less, and there has been some consolidation. And the challenge of these companies and the reason for what's happening at the grand level is that these therapies are very, very expensive to develop. And the know-how today to manipulate cells is still at the manual stage, which means that cells are collected from a patient, they're transported to a very expensive, what we call manufacturing facility, which is clean rooms with operators in bodysuits, and those individuals, highly skilled, manipulate the cells, get the result they're looking for, and then that manipulated set of cells or bags find their way back to where the patients are for infusion. That's a very unwieldy way of doing it, very expensive, and because humans are involved, it's error prone. So there are quite a few companies that are working on automating this process to take the human element out of it, which is expensive and error prone. And it requires specialized skills. So the biggest challenge in the industry today is the manufacturing challenge, the cost of manufacturing, and a corollary to that is the availability of skilled staff. Virtually everyone we talk to, we're in the consulting business, advisory business. From time to time, in fact, with high regularity, ask us, do you know someone who? Because we're looking. So there's enormous sort of pent-up demand for qualified staff. So that's where the industry is. It's very exciting at the scientific level. It's very exciting as these clinical trials move to their various stages to approval. We're actually running a clinical trial, actually a couple of trials on a contract basis for approval in Europe and the United States. Incidentally, the business is very global. COVID has had one benefit, which is people have gotten used to Zoom and Teams. And the systems themselves work a lot better than they did 3 or 4 years ago. So no one thinks for a second about the challenge of getting on a video call. It's just almost on autopilot. Click, click, and you're on. And because these things require highly specialized skills, we reach for whoever we can find around the world. English is the common language. It's unusual for us to have a conference call with maybe 15 or 20 people, and not everyone, virtually everyone, have some accent that is other than the local accent. So it's an exciting field. If I was in engineering school today, I went to Brooklyn Poly, which at that time was across the street from NYT and NYIT, and I'd love to go back today, know what I know, and just sit in the class because there's so many exciting things happening. And I would pick one of the bio fields, bioengineering, biomechanics, because this is, to me, it's the cutting edge of where we are as a society.
Cool. So we have all this cost and process to manipulate the cells, and obviously that's translated into the pricing that is that physicians and hospitals are charging to patients, and that's come under a lot of scrutiny. So maybe just talk a little bit about, what pricing may look like, what it looks like now, and what it may look like a little bit over time.
So let me start with a horror story, because then we'll finish on the upside. So clinical trials are being run in the United States and Europe. In the United States, the reimbursement is much more liberal than it is in Europe. It is an actual case of a child that was enrolled in a clinical trial in Europe, was treated successfully. The product or the therapy was approved, but the Europeans are not reimbursing it because it costs $500,000 or $1 million. And so this child, the one that was treated, will survive. His brother, which has the same genetic deficiency, will not survive. So that's a terrible situation. So let's talk about cost for a moment. Like any experimental product at this stage, it's very expensive. So to bring just think about what has to be done. A patient has to be conditioned, cells need to be extracted, they need to be transported, they need to be manipulated. And by the way, transported in a very secure way we use the same services that you use for organ transplant. So this, you don't FedEx this stuff, you hand carry it. So if you visualize that a box, FedEx box costs, I don't know, $50 to move. Well, a cell product costs 20 times that to move. Yeah?
So that guy on the bicycle going down the street here is not carrying cells?
No. It's very specialized, and it goes by air and all of that. So the cost can easily be $400,000 to $500,000 real cost between the transportation, the patient conditioning, the hospital cost, the manipulation of the cells, and then the reinfusion and the follow-up. The highest single component is the labor at the manufacturing facility or at the laboratory. That's where the highest cost is. As I mentioned earlier, there are some companies that are working on automation. At this point, these things are highly customized, so it's hard to automate something that's highly customized. I'll just give you a small example. Many of these products arrive in bags, in plastic bags. Well, robots have a terrible time with plastic bags because they're too soft. They don't know how to access it. So they need to we will need to convert this to some kind of a hard container in order for robots to be able to manipulate it. Just a small example. It would probably cost $1 billion to figure this out. Yeah? So that's the challenge. In the United States, some of these therapies have been approved for reimbursement at $500,000, et cetera. And because they are for relatively rare diseases, the FDA has said we're approving it. We're only going to treat a few patients, a few dozen patients during the course of a year. But once there's a breakthrough, and let's say we have a treatment for diabetes, just to make a big condition or a big situation, we're looking at billions of dollars. So something has to happen to reduce those costs. One of the more obvious things to do, and we're actually working on this, is to bring the manufacturing capabilities, the clean rooms and the operators to where the patients are, that is at the major hospital centers, so that we call it distributed manufacturing, so that the cells don't have to travel halfway or all the way across the country, some cases across the continent, to be manipulated. That has its own challenges because it's a very specialized environment, and hospitals are hesitant to install something this specialized for a specific application unless it's a major indication where it makes sense economically for them to treat patients. So there's all these different moving parts, and those of you that may be engineering strategists would want to think about this. What do I do with this very high cost? Where are the cost elements? And what could we take out? Clearly, you can't have therapies approved that cost $1 million where you have 1 million patients. So potentially that's just out of any kind of range that can be reasonably reimbursed. The Europeans have a different view about this. My own personal view, they're much more fatalistic. And they simply say, well, can't afford it. The system can't afford it. The country can't afford it. Sorry, you'll get the standard course of treatment, and we're very sorry. In the United States, we don't have that attitude. We still treat patients. And anyone that goes to a hospital, irrespective of their station in life, expects and generally gets state-of-the-art treatment. So that's the difference. Asia has its own calculus.
So we just have a couple minutes left, and so a number of the folks in the room are probably budding entrepreneurs and thinking about taking a technology, let's say like a cell or gene therapy type technology. What are sort of the characteristics that they need to be able to present in order to maybe attract the attention of potential investors into this field?
So if you were to identify something pretty narrow, and I wouldn't go for something big, something narrow, please keep in mind that you need a team of highly specialized experts on the science side, on the clinical side, maybe on reimbursement, maybe on regulatory. So I would encourage you to to establish what I would call a salon, that is a discussion group, to talk to these folks. You don't need them full time, but you do need them. And assemble a team that has very high credibility, because the investors or the financiers will look at that and say, if he or she has been able to put that together with that team, then this thing has credibility. So that's the way I would do it. I would research who is the best of the best from around the world, and then assemble that team to move it forward.
You always hear investors say, without the team, it doesn't actually matter how good the technology is. If this team cannot execute the technology, can't invest in it. So it's got to be the team first. Obviously, the science has got to be unique, and obviously you'd like to see proofs of concept and things like that. But obviously, at the very early stage, you're dealing with promises.
Yeah, the investors are going to talk to your team and say, what do you think, and what do you think, and what do you think? And those answers have to be positive, aligned, and there has to be a track record.
Any questions here from the audience?
I'd like to hear more of your thoughts about this issue of pursuing therapeutics and manufacturing of therapeutics for rare diseases, and the contrast with diseases such as heart disease, diabetes. Let's exclude cancer for now. But if you could talk more about market opportunities in those two areas, I'd like to hear more about that.
Sure.
If you could quickly paraphrase the question, because the remote audience couldn't hear the question.
Yeah, so sort of contrasting, working on more niche therapies as opposed to more of the larger mainstream categories.
So quickly, the answer is that the science is opportunistic. So we work on what we can work on, and university professors and departments are not looking at the size of the disease. They're looking at the interesting aspects of a molecule or of a cell. Right? So it's driven by that. So by the nature of the way biology is built, rare diseases tend to be more frequently addressed than the big ones. Not to say that it's excluded, it's just that whatever is in front of us, right? So that's what's happening here. Everyone knows that there will be a breakthrough when we can address cardiovascular disease or the autoimmune diseases, or diabetes being one of them. But science is not focused on the market. Science is focused on discovery, and that's really what's driving it.
It's hard for investors to think about investing in some of these, super large programs when you think about the cost of doing, thousands of people in clinical trials with, periods of monitoring for, decades. The costs of those things are just so enormous. There's interplay of who can even invest in those companies, right? So venture capital can't even really help support those types of things because it's too big. It really has to be through big pharma, or government is really the only choices for the largest of the large programs. So a lot of people think of these niche programs as opportunities where I can raise an amount of money that I can raise, and I can accomplish an objective sometime before my grandchildren have to start worrying about the program.
So now that you've taken one company straight to public without going through the venture capital raises, being in that sphere of trying to develop cardiovascular therapies and having encountered the skittishness of VCs wanting to invest a large amount of money, which is what it takes to do cardiovascular clinical trials, what do you think about just going public?
So the question, So the question was, taking your company public without having to go through the traditional rounds of venture capital. So part of the answer to that question would be, you have to have a strategy and a long-term vision to be a public company. It's not just to get the money. You have to have the process, especially in something like cardiovascular, will be a long process. So what are you going to do for the years where you may not be, where you're still waiting for your top line data results to come out? And then in the meantime, you have all the burdens of a public company. So the capital markets were really terrific over the last couple of years, not so much the last 18 months or so, but prior to that, you saw very early stage companies going public, single product, preclinical, maybe a phase one data. Now those companies are struggling because they don't have great data. Their teams were never fully developed, and they didn't raise enough money to really finish the program. And now the capital markets are relatively dormant, and what do? So those companies are really struggling now, and some of them will become sort of the fallen angels, where they just won't be able to raise the money and they're going to have to stop the programs. So it's really kind of unfortunate. And part of it gets back to, I think, the strategy of should you have been a public company to begin with? And at the really early stage, without opportunities to do many things as a public company, it's really hard to be kind of a one horse saloon, if you would. So, George, any other thoughts from your perspective?
I always like to put dollars on it. Just to get started, you need about $500,000 a year just to be a public company, maybe $1 million, even with an efficient operation like Eisner Amper. So that's to begin with. The second thing is that you got to have more than one shot on the goal, right? So if you have a single product, you're betting everything, it becomes a very binary outcome. And that's very risky in a public setting. So if you have something that may be a platform technology that enables other things, then perhaps you have a window towards a public offering, because you're going to have ups and downs, but at least you can say, look, I've tried it on this, this, and this, and here's 15 other things that I'm working on. So that would be perhaps a way to thread a way to a public setting. But in the beginning, the experience has been venture capital, some private equity, but venture capital really to get going.
That said, there's been a number of companies who have said, I have no other options. I've tried X, Y, and Z, and I have not been able to raise money. And if I can do this through a SPAC transaction or a reverse merger or a public offering or something, and I can get some money to get to my next inflection point, I'm going to do it, because otherwise, my heart and soul is at risk here. So I'm going to do it anyhow. Those companies tend not to be terribly successful, but scientists oftentimes have a unfettered enthusiasm for their technology and are just going to go forward however, whenever.
Thank you so much, John, for hosting what a wonderful fireside chat. Thank you, George, for being with us. Maybe next year we invest in a fireplace in here, make it a true fireside chat. Or do it the cheap way. Absolutely, yeah.
Hi, good afternoon. I'm Scott Rodeo from the Hospital for Special Surgery. I'm an orthopedic surgeon. My practice is sports medicine, shoulder and knee problems and things like that. But I also serve as vice chair of orthopedic research at our hospital and run our soft tissue research laboratory, as well as our Center for Regenerative Medicine. So I'd like to talk to you today a little bit about the current status of this whole area of regenerative medicine in orthopedics, some of the great potential, but also some of the current pitfalls here. Here's some of our research is supported by the NIH and some various foundations, some of which relate to our laboratory work here. So jump right in. What is regenerative medicine? Well, you'll hear the term orthobiologics. This generally refers to the use of quote-unquote biologic augmentation techniques to improve tissue repair. So we can use different products derived from blood. You may have heard of PRP, platelet-rich plasma, but there are a number of other blood formulations kind of listed here, not to get into a lot of the details, but a lot of these blood formulations work via inhibiting different anti-inflammatory mediators. We'll talk more about that as we go through it here. We can use cell therapy. You'll hear the term stem cells a lot, and I'll make some comments about why that may not be the best term to use, but we can obtain cells from bone marrow, from fat, adipose tissue, and even from perinatal products, placenta, amnion, even umbilical cord bloods. We can use gene therapy techniques to modify these cells. Another area of biologics is using purified cytokines and small peptides, and there's a lot of research going on in this area. Currently, we have peptides, cytokines that can help bone formation. That's BMP2 and PGF, platelet derived growth factor. So these are available as purified small peptides to augment bone biology. There are different scaffold materials that are useful to support tissue healing, and even the ability to use systemic drugs, parathyroid hormone, even things like vitamin D, testosterone, and a number of other medicines such as doxycycline, the commonly used antibiotic, that can have positive effects on soft tissue. Losartan, for example, is a commonly used antihypertensive that actually can have an effect on connective tissue. So we're learning a lot about these different ways to kind of leverage the biology of these drugs to improve soft tissue repair. So I would submit to you that this area has tremendous potential, but the clinical use of biologics has greatly outpaced the current scientific and, frankly, clinical evidence. And there's a lot of false claims for what are often, frankly, unproven therapies. This is a billboard picture from Chicago. There's, frankly, a fair amount of indiscriminate use in this field. There's aggressive marketing. But that's kind of the current state of where we are. If you follow the news, you'll see the FDA cracking down more and more on some of these sites that are promoting these unproven therapies and using these inappropriately or indiscriminately. So that's the current landscape, but this is a huge market and a large growing area. And our efforts at HSS have been to really study this area rigorously, understanding that there is great interest, there's great potential. There's a lot of marketing, so our job is to really collect rigorous data and try to do this the right way. So to jump in, what does this all mean? So our overall message is that techniques and methods to affect the biologic events in tissue repair certainly have tremendous potential, but I'd submit to you are not fully ready for prime time. I use these materials myself judiciously in my practice, some of these different blood-based formulas and cell therapy approaches. Certainly animal studies suggest tremendous promise, but there are some limitations, and certainly indiscriminate use of what are sometimes unproven therapies may in fact hinder progress in this field. The first question you have to ask when we're treating any tissue with these agents is, what are we trying to treat? So we're first in orthopedics, we have bone, we have cartilage, meniscus, tendon, ligament, muscle, a lot of quite varied tissues. So what is the goal we're treating? Do we want to increase the blood supply in a tissue? Do we want to increase cells to migrate into a tissue? Perhaps we want to decrease inflammation or accelerate remodeling of the healing matrix materials. Another goal would be to attract either local or even distant cells to aid healing. Maybe we want to decrease the degradation of the fibrin clot, the scaffold that we need for early healing. Maybe we want to just affect the production of various proteins that cause pain. All of these are worthy goals, but they're all very different. And we need to, before we jump in and use these agents, we need to kind of identify first, what is it we're trying to treat? Identification of the desired target may allow us to ultimately match a specific PRP or cell formulation to that particular tissue. We'll see that one size does not fit all. Now, this certainly requires identification of the underlying cellular and molecular mechanisms of tissue degeneration, tissue healing, really understanding what we're treating. For example, take tendon problems. Some of these are very chronic, some are much more acute. Those are probably very different biologies. Arthritis, sometimes it's real inflammation, sometimes there's not so much inflammation. Again, what we call arthritis is probably 25 different conditions. They're quite heterogeneous. At the same time, we need to rigorously define the composition and the biologic activity of these different formulations, because only then can we match the specific agents, the specific drug or treatment, with the desired biologic target. Again, as I mentioned, one size does not fit all. Ideally, we need to tailor these biologic therapies to the underlying pathology, different anatomic sites, again, an acute process versus a chronic process. The presence of systemic metabolic abnormalities such as diabetes or renal disease or frankly age can affect the biologic response to these materials. The local mechanical environment, our orthopedic tissues, tendons and ligaments, cartilage and bone, they all see load. We walk on these joints. That mechanical forces has a real effect on the biology. We need further understanding of how mechanical load interacts with biology. Certainly the same either PRP or cell therapy preparation would not be expected to have the same positive effect on, again, diverse types of tissue, acute versus chronic process, sex probably matters, age matters. All these are important factors that we need to take into account and really gain further information so we can have a much more refined, responsible use of these agents. So what are the cell therapy options? What do we have? So we can derive cells from bone marrow. Often we aspirate bone marrow from the hip, the iliac crest, but we can get bone marrow from different areas. This picture at the bottom, it shows taking it from inside the knee during surgery. The majority of cells in bone marrow actually are hematopoietic stem cells, cells that form the blood elements. And that's not the cells we need. We want the so-called mesenchymal stromal cells. Understand that only like 0.01% of cells in bone marrow are truly the kind of stem cells that we want to use for orthopedic tissues. We can derive cells from adipose tissue, from a lipoaspiration. There are different techniques available. So one is a so-called microfragmented fat, taking fat and using emulsification, mechanical, to kind of break up the fat. Beyond that, we can derive the stromal vascular fraction, this requires enzymatic digestion of the fat to isolate and then concentrate the specific cell population that we want. Because fat has a very heterogeneous population of cells listed here. Adipose cells, there are red blood cells, there are different white blood cells like lymphocytes and monocytes. Enzymatic digestion can help us to isolate this stromal vascular fraction. However, at this time, FDA guidelines require us we need an FDA approval to use enzymatic digestion. This needs to be done under an IND, an investigational new drug application. This can only be used in a clinical trial. Similarly, even further along in using adipose tissue is culturing the cells. So we'll see that for any of these things, bone marrow or adipose tissue, we can't take these from the patient out to the laboratory and do cell culture expansion. That can only be done under an FDA trial. So these techniques can be used in other countries, but we're a little bit limited in this country by our regulatory environment. So if you want to use adipose tissue right now, the only real currently available technique for your average clinician is microfragmented fat, which has some value, but again, certainly needs further work and further development. Furthermore, there are cells derived from these so-called perinatal products, so amniotic membrane-derived products and even umbilical cord bloods. These contain a number of anti-inflammatory cytokines and immune-modulating proteins. These have been marketed as quote-unquote stem cell sources. There are vanishingly few, if any, cells in these materials, so they shouldn't be termed stem cells, and that's really inappropriate marketing. Again, they do contain a number of cytokines and anti-inflammatory mediators. It can be helpful. Very little controlled data out there. Again, these can be, the cells or these materials have been used derived from umbilical cord bloods, placental tissue. Importantly, as of 2 years ago, May 2021, these formulations can now only be used in an FDA-approved clinical trial. There are basic criteria, in particular, the reliance on live cells in these materials. As I mentioned, there are very few live cells, but they're marketed as containing live cells, and thus the FDA requires these can only be used now under a clinical trial. So these are really largely off the market, quite honestly. I submit to you that we currently have very limited ability to use stem cells. Again, cell therapy, tremendous potential, but there's a lot of unknowns. In particular, these minimally manipulated cell preparations that are currently used, as I showed you there, bone marrow or adipose, those need to be carefully distinguished from culture expanded cells that are produced in the laboratory. Those are very different formulations. And in fact, the term quote unquote stem cells should really not even be used when discussing current techniques for cell-based therapy. A better term that's out there now in our field is connective tissue progenitor. This is a population of cells that exist in many tissues that can in fact proliferate, and they can generate some new tissue, much more limited repertoire than a true stem cell. A true stem cell is a very specialized cell by any cellular, molecular, or functional criteria. So a connective tissue progenitor cell, what we're talking about, these cells can be measured by culture. It's called count colony forming units. We culture the cells, and each connective tissue progenitor cell is a founding cell for a colony. A colony, little purple dots you see there. A colony is a group of 30 or more cells. And so I would submit to you that you've heard the term MSC, mesenchymal stromal cell. Some have called this mesenchymal stem cell. That term should only be used to refer to culture expanded cells that meet the specific criteria from the ISCT, the International Society for Cell Therapy. They promulgated criteria in 2006 to define culture expanded stromal cells. Well, that's not what we use. These freshly harvested tissues, we can't take these and culture them. So we should not even use this term MSC or frankly stem cell. You'll hear it used a lot. There's a lot of questions and confusion about nomenclature, and some of this is the aggressive marketing, but we should not use the term MSC for these cell populations that are currently used by clinicians. Moreover, I'd submit to you we currently have essentially zero ability to use true stem cells. Again, the number of true stem cells by any criteria in these currently used formulations is vanishingly small. As mentioned, the classic definition of this mesenchymal stromal cells based on these on culture expanded cells, based on the definitions from the International Society for Cell and Gene Therapy, which again is very, very different than cell populations that are contained in freshly harvested tissues. So again, this term stem cell or MSC should only be used to refer to a culture expanded population of cells that meet these formal criteria, and clearly should not be used to describe these heterogeneous populations of native cells that really have undefined properties. Moreover, there is very little evidence to suggest engraftment of injected cells. When we inject cells, we like to think that cell turns into a bone cell or a tendon cell or a cartilage cell. Well, that probably doesn't happen. Rather, these cells act via a paracrine mechanism. What that means is the cells we inject produce a number of signaling molecules, anti-inflammatory molecules, immune modulating molecules, important molecules to modify the local injury environment. They may attract local cells. So they have an action, but these cells do not intercalate into the healing tissue. We know that in culture, stem cells will undergo multi-lineage differentiation. That is, a stem cell can turn into a fat cell, a bone cell, a cartilage cell, a tendon cell. That happens in cell culture. That we don't know that occurs after injection in the body. Understand that the current regulatory environment in the U.S., as mentioned, prohibits us taking our material out of the operating room and doing cell sorting and culture expansion. That level of manipulation is not currently allowed. It can only be done under an investigational new drug application through the FDA. So again, very important to distinguish these cell populations that are freshly harvested in the clinic or OR. We have to distinguish that from these culture expanded cell populations. As mentioned, we can't perform this ex vivo manipulation with culture expansion. Culture expanded populations, they have real potential. That's a highly selected and fundamentally different cell population. That's different from the mixed cells, the population that we use that contribute to the generation of these cultured cells. So again, very different here. The currently used heterogeneous cell populations are really, frankly, poorly characterized. The basic identity and certainly the biologic activity of these cells is really not well known. Clearly, this field, we recognize we need markers of cell purity, of potency of a cell therapy formulation, markers of biologic activity, and only then can we start to characterize these very heterogeneous formulations. All this foregoing suggests that true tissue regeneration via cell therapy is frankly going to be a bit unpredictable. A critical limitation is the heterogeneity in these populations. Frankly, we have a very rudimentary understanding of these currently used therapies, whether it's PRP from blood or bone marrow cells or adipose-derived cells. We clearly need reproducible methods to measure and classify these products. Again, as I mentioned, this heterogeneity points to the real need to identify sentinel markers of activity, purity, potency, etc. Ultimately, such characterization could allow us to move this field forward by allowing the ability to remove competing or inhibiting cells, allowing more targeted therapies, which may be very helpful for us. So in any of these heterogeneous cell mixtures, there's probably good cells and bad cells. There may be cells that inhibit healing. We need to identify how we can remove those inhibiting cells and expand or concentrate the good guys. This is an example of a criteria put forth by one of our co-authors in recent years, these minimal information for studies evaluating biologics, just demonstrating all the things, the data we should record. Where are the cells from? How are they processed? What area were they injected into? What markers were checked? Until we have this data, it's hard to make any real conclusions. On the right is one of several editorials that I put together on this whole area, and here I discussed the need for minimum reporting standards in this whole area, and kind of summarized here that the adoption of minimal reporting standards that facilitate the characterization of these therapies hopefully bring us closer to ultimately this personalized medicine approach for our patients. This, one size does not fit all. We need to personalize or choose the optimal treatment for different conditions. Another major point regarding all these biologic materials is that they have been demonstrated to be symptom modifying. That is, they can improve symptoms of pain or swelling, but very little data to suggest they are truly structure modifying. That is, that they regenerate tissue. So whether it's blood products or cell-based therapies, we know they can improve symptoms. As mentioned, they work via a paracrine mechanism by producing these signaling molecules that affect the local environment with, again, very little evidence that the cells themselves actually can differentiate into a specific cell or become intercalated into the healing tissue. Rather, they act by producing all these signaling molecules. Very little evidence to suggest that they affect tissue microstructure, composition, or strength of the tissue, the material properties. Again, they produce these various signaling molecules. These signaling molecules are numerous and complex. There are cytokines and growth factors. There are chemokines, proteins that attract cells, a number of anti-inflammatory proteins that will be helpful for symptoms. There are proteins that modulate the local immune cell response that are critical in the response to injury. So these are very, very complex materials and need to be further characterized. The biologic effect of these cells appears again to be due to the production of these various mediators that modify the local environment. So again, this MSC, mesenchymal stem cell, well, in fact, it's been suggested this should be renamed a medicinal signaling cell in recognition that these cells produce these various modulating proteins. Arnold Kaplan is a giant in this field. In 1991, coined the term MSC, mesenchymal stem cell. And then in the mid-2010s, he and the field recognized that it's really these cells act via this signaling, and he encouraged changing the name to the MSC to a medicinal signaling cell. Recognize these cells are little pharmacies, if you will, that produce these number of factors. And so these cells may function by modifying the local injury environment, again, rather than directly contributing to healing. So again, they're symptom modifying, but very little evidence to suggest that they are truly structure modifying, that they regenerate tissue. Frankly, what we're learning now is cell therapy, these exogenous cells we inject, whether it's from adipose, bone marrow, maybe perinatal tissues, they probably act by stimulating the intrinsic stem cells that exist in many tissues. In the walls of blood vessels exist pericytes, small cells in many tissues that are progenitor cells for that particular organ or tissue. These cells are quiescent. Our challenge is how do we stimulate those cells? Well, the factors produced by cells we inject may stimulate these intrinsic stem cells that exist in many tissues. My own view on the future of this area, I think the heterogeneity in the starting material for a cell culture suggests that some elements of this population may in fact be desirable for a specific tissue, while other cells present may not have the potential for generation of a desired tissue. So again, as mentioned, there may be cells that compete with or inhibit the desirable clones in a given population. Ultimately, the development of methods to reproducibly characterize a population of cells could really allow the ability to remove these competing or inhibiting cells and improve what we're doing. And so we can use in the future, hopefully, cell sorting techniques to select a desired subset of cells based on some criteria, which would eventually allow a much more precise match of the cell therapy formulation to the underlying pathology or tissue that's being treated. And this would eventually, again, matching the cell population or even just PRP to the desired biologic target could facilitate this precision medicine approach. Again, the precision medicine concept is the right medicine for the right patient, at a low level, 1.5 to 3 times. In contrast, so-called Buffy code formulations includes both plasma and cellular layer, and the difference here is there's a much higher platelet concentration. They also contain white blood cells. And I'm going to leave it at that. PRP is very complex, and you get into much more nuances as far as characterizing PRP, but suffice it to say there are different formulations, and we're still learning much more about the basic biology of PRP. I thought I'd kind of end by reviewing the biologics, or the role of biologics for knee osteoarthritis as a way to kind of illustrate sort of the current status of the field and some of the current concepts and some of the both potential and pitfalls, if you will. So what about PRP for OA? I mean, the most common thing we see in orthopedics, osteoarthritis, and knee and other joints, certainly. And there is actually good data now to support the use of PRP to manage symptoms of knee osteoarthritis. Most studies seem to support use of a leukocyte-reduced formulation. That is a PRP that has a low white blood cell formulation. The reason being, white blood cells contain some inflammatory mediators. So most data, not all, but most data suggest using a leukocyte-reduced, a white blood cell reduced formulation is helpful for treating knee arthritis. But again, it's a very complex area. We have a rudimentary understanding of currently used therapies. There's tremendous heterogeneity in PRP composition. You think about it, if you put in a drug, this is a known composition, known concentration. That's very different than these agents that are really unknown composition and concentration. We certainly need a much more detailed understanding of PRP, and this whole idea of leukocyte rich or white blood cell rich or poor. We need a much more refined analysis. We need to know the types of white blood cells, and frankly, the ratio of white blood cells to platelets is probably more relevant. So again, you'll hear individuals say you have to use this one or that one. I think we should not be dogmatic because we need much more data here. And osteoarthritis itself is very heterogeneous. So not only are our treatments heterogeneous, but the patient and their disease is heterogeneous. So what is the data for knee OA? Well, this is a several years ago, a meta-analysis that basically concluded that PRP does show more benefit in pain relief than other injections. HA is hyaluronic acid. You may be familiar with so-called gel injections that have been used for osteoarthritis, which have some efficacy. Most studies demonstrate PRP to be more efficacious, so there's some early data there. They concluded here in this particular meta-analysis that for short-term follow-up, PRP injection is more effective in pain relief and function improvement than either gel or certainly placebo. But again, most of these studies show kind of short-term pain relief, 6 months to 12 months. Another meta-analysis here, including 18 studies, mean follow-up just about a year, 11 months here. The improvement was higher in the PRP group when compared to the gel group, again. And here, again, the leukocyte poor, the white blood cell poor PRP had better data or better results than the white blood cell rich formulation. So there's some data out there. More recently, we found that combining platelet-rich plasma with the gel can be helpful. This particular study from China looked at 122 patients where patients received either the gel, PRP, or the combination. And essentially what they found is that the combination of PRP plus the gel was superior in pain relief to either the gel alone or the PRP alone. They did it's rigorously done. They have various PROMs as patient-reported outcome measures of pain and function. They looked at synovial hyperplasia, essentially inflammation in the joint, and that was improved by the combined injection. They demonstrated reduced levels of various inflammatory mediators in the joint fluid. So there's some data to support the combination here. There are other generations of PRP being developed. One is something called autologous conditioned serum, which actually takes blood and heats it and incubates it overnight with these little glass beads, which what that does, it stimulates production of something called IL-1RA, interleukin-1 receptor antagonist. IL-1 is a very potent inflammatory mediator. The receptor antagonist, IL-1RA, is a potent anti-inflammatory cytokine. So this material kind of concentrates this potent anti-inflammatory agent. Now, this has been used overseas. This is not currently FDA approved. There is some data, though, from this is studied 13 to 14 years ago using this particular material, demonstrating that it can be effective in treating symptoms of knee OA. And so they found positive results here, but not currently available in the U.S. on a regular basis. Something called autologous protein solution, yet another formulation, again, using blood. But here they use a cell separator to separate out the cellular components, and then it's filtered through these polyacrylamide beads, which again concentrates in particular this IL-1 receptor antagonist, same as that prior slide there. So again, a lot of the principle here is concentrating these anti-inflammatory mediators. And there's some early data, this is a study from Europe, patients that are randomized to either saline or this autologous protein solution, and they found improvements out to 3 years after a single injection. So there's some early data to suggest benefit here. This is currently being evaluated by the FDA in this country in a clinical trial. What about cell therapy to finish up, or stem cells and cell therapy? Well, there's pretty extensive data from animal models to support the potential of cell therapy approaches, but not a lot of clinical data as of yet. This is a study from Japan where patients were treated with cell therapy. They all failed a prior steroid or gel injection. And these patients received either culture expanded cells, which we can't do here, but they can in Japan. So these are culture expanded cells, or non-cultured cells. Injected into the joint. These patients quite simply had osteoarthritis. They looked at the patients at 6 months, and both groups report improvements in standardized outcome measures. But in fact, the improvements were better in those patients who received the culture expanded cells. So again, culture expansion really can add something. Again, we can't do that yet in this country, but that really has the potential to produce a cell population that is concentrated, isolated cells that can be beneficial. Again, they concluded that the cultured cells outperform the non-cultured cells. As I mentioned, there's very little data to suggest that these materials are structure modifying, that is, that they can regenerate tissue. Well, this is a nice study from Spain some years ago, a randomized trial where they used, again, culture expanded cells. We can't do that, but in Spain they can. So they use culture expanded bone marrow cells compared to a group of patients with just the gel injections. Those patients who received cells had improvements in pain and their function, but importantly, based on MRI scan, there's an improvement in cartilage quality in those patients treated with cells. So this suggests not only do they improve symptoms, but in fact, there's some improvement in structure with quantitative MRI scans demonstrating that there was some effect actually on the cartilage here. Again, culture expanded cells. So as the FDA, if the regulatory environment changes here, we'll be able to use some of these techniques. In contrast, a study a colleague of mine down at Mayo Clinic in Florida, this is a placebo-controlled trial. This is just using bone marrow aspirate, uncultured cells. So, but it's an interesting study because patients were randomized. They all had bilateral knee pain. So one knee got bone marrow aspirate, essentially bone marrow cells, and the other knee got a saline placebo. And what they found, again, these cells are not culture expanded, no difference. So the knee with saline placebo improved just as much as did the knee injected with cells. And we know there's a strong placebo effect with injection. So no real difference between knees. When they went ahead and looked at these patients further, they used an MRI scan to see, okay, patients got better. Was there any change in the cartilage using these quantitative, sophisticated MRI techniques? Well, in fact, these quantitative MRI techniques showed no changes after treatment. So there was no regenerative benefit with this single bone marrow cell injection. So the mechanism of action that led to pain relief certainly cannot be concluded to be due to cartilage regeneration. So for me, the bottom line for knee osteoarthritis, where is the field today? Many of us will use a leukocyte pore, a white blood cell pore PRP for management of symptoms. Consider combining PRP with hyaluronic acid is very reasonable. This ACS, autologous conditioned serum, again, not yet available in the United States. I know there are some practitioners doing this off-label, but it's not FDA approved. So the autologous protein solution, again, not yet available in this country, being evaluated now by the FDA. Again, a lot of these materials concentrate this IL-1 receptor antagonist, which has great potential. So as we learn better ways to isolate cells that produce this material, then we'll have some potential here. I do not routinely use cell therapy approaches for the knee. It's not practical to do that in the clinic. We'll do this sometimes in the operating room, but doing a bone marrow aspirate in the clinic or a lipo aspiration, a liposuction, that's not trivial. It can be done, but you need sterile setup and sedation and things like that. So, and again, we cannot use culture expanded cells at this time outside of clinical trials. Just some work that we're doing here at HSS to give you an example of how we're studying this area. This is a study we published several years ago where the goal is to correlate the responses to PRP and culture, to correlate that with the composition of the PRP and, importantly, the clinical outcomes in our patients. So what we've done is we developed a co-culture system of cells, macrophages, important immune cells, plus fibroblasts, kind of essentially culture condition that kind of simulates a joint environment. Then we add this particular cytokine, TNF-alpha, which is a pro-inflammatory cytokine. It kind of induces an inflammatory environment. And so now this inflammatory culture, we add PRP to this culture, and we look at gene expression to see how PRP might modulate that inflammatory environment. Now, the PRP samples came from 51 patients that were receiving PRP injections for knee osteoarthritis. And what we did is we identified responders and non-responders. So of these 51 patients, some did well, some didn't really get better. So we identified those that responded and those that didn't. And we used PRP from all of these. What we found was PRP from those patients who were responders, who had a good outcome, their PRP had a more potent inhibitory effect on these particular molecules, CCL5. And their PRP also produced higher IL-7 levels. The particular molecules aren't important, but the point here is that PRP from patients that were responders, that had a good response, actually had a more potent positive effect in this particular cell culture models. This is just an example of how we can use these co-culture models to understand, kind of characterize a PRP formulation. This particular CCL5 that we found inhibited, that molecule has an active role in recruiting inflammatory cells. So the ability to inhibit that makes sense. It's clinically relevant. So just one example of the type of tests we can start to do to characterize these formulations. So in summary, I think biologic approaches for treatment of these different conditions are very promising, certainly require further investigation. Current data does support the use of a leukocyte pore PRP for management of knee OA. Again, cell-based therapies are currently rarely used, really only in conjunction with surgery on any regular basis. Unfortunately, there's still a lot of, frankly, indiscriminate use of unproven therapies. A lot of this is due to the allure of these biologics, aggressive marketing, certainly. Practitioners, frankly, in many specialties advertise these cell therapies for numerous, frankly, often implausible conditions. This area, again, needs further rigorous study. Frankly, often see exorbitant charges for these unproven therapies, and the aggressive marketing has contributed to that. Patients sometimes told they can avoid surgery, which sometimes may delay use of more standard and proven therapies. I think we as orthopedic surgeons need to exercise leadership in this area because we are treating a lot of these patients with a lot of these different pathologies. I'll stop there. I think clearly we need a rigorous, evidence-based based approach, and that's what we've been trying to do at HSS with our laboratory and clinical studies. Thank you very much.
Thank you, Scott. And our first speaker for the afternoon session is Dr. Randy Stout. Randy, if you could come up to the podium, please. Randy is the Associate Professor of the Department of Biomedical Sciences and the Director of NYIT Center for Biomedical Innovation at the New York Tech Imaging Center at our NYIT College of Osteopathic Medicine. Randy's talk is titled Super Resolution Microscopy for Discovery at the Frontier of Cell Biology. Without any further ado, I'll pass it on to Randy. Thank you, Randy.
Thank you very much for allowing me to speak about some of the stuff we're doing, and especially focusing today's talk will be on the NYIT Imaging Center and describing a little bit about what's coming with regard to our Biomedical Research Imaging Innovation Center that is being built and established. So I'm going to be talking about how we're using microscopy and how that interfaces with the world of engineering and innovation. So why would we use microscopy? And it's to understand the spatial relationships between biological samples, materials, things like that, and even just having a list of parts of what makes up the list of chemicals, the list of molecules that makes up an organ, a cell, a material that we're trying to use as a prosthetic, it's not enough. We need to know how those things are arranged within that material. So that's where the NYIT Imaging Center comes in, and that's going to be included into this larger project, the Biomedical Research Imaging Innovation Center. And it's really exciting to be able to, a lot of the things we've been talking about today are going to be expanded. It's going to be access to the researchers across the entire institution and our collaborators. We're going to have a place where we can do this type of work. We can do the research that will further these engineering and biomedical research projects. And so along those lines, as a collaborative imaging center, we have too many projects that I can talk about today that we're collaborating both within NYIT, across different colleges, and even outside institutions. So we've really focused on optical light microscopy. And these two movies that you're seeing are both super resolution imaging techniques. And so I'll talk a little bit more why we use super resolution imaging and why we're focusing on that. But we have a neuron here, and you can actually see the individual dendritic spines. This is a neuron where I got the sample from Dr. Ramos and imaged it on one of our super resolution confocal microscopes. And it's not just about getting pretty three-dimensional images, but more so about quantifying how these structures change in different disease models. Below that is a live cell. So this is a living cell where we're doing super resolution imaging. And the little spheres are less than a micron across, and you wouldn't be able to see any structure in a normal microscope. This is a live cell movie of gap junction connection between two cells that we took in the super resolution microscope in the Ryland building. So moving on to kind of show the different tools that we've been able to, over the past 3 years, add to NYIT's research capabilities. There's too many things to talk about, so I'm going to have to focus on one in particular that we're doing some really exciting work with. It happens to be the one that's been at NYIT since 2019. Moving forward, we're going to just continue to build in this area of optical imaging and be able to do more and more things to be able to find out what's happening in our cells that we're trying to study and engineer different new tools to help regeneration and help the human condition. So this, what are we talking about when we say super resolution? In optical microscopy, so not electron microscopy or other types of scanning electron microscopy or something like that, if you just magnify the light signal coming in to your detector more, you just get a larger blurry image past a certain point. And this is based on a number of limiting factors, But it really boils down to the fact that depending on the wavelength, so for blue or green light, it's about 250 nanometers where you can separate two different features in your image. And that's what we're trying to do. We're trying to understand the shape and spatial relationships between things. And that 250 nanometer distance is larger than the size of even molecular assemblies of proteins and stuff like that that are so important for how our cells, our heart, our brain cells work. So a number of different techniques have been developed to break past that resolution barrier to be able to resolve features at an even smaller scale. And one of the ones that won the Nobel Prize in 2014 was with Eric Betzig and William Warner, was the localization microscopy. And it was a specific type that we use in the lab in Ryland all the time. And it was extended. The way it produces higher resolution is that instead of just magnifying more and just getting overlapping signal that you can't resolve, instead it spreads the signal out over time into single blinking events. And then you can know that this is an individual blinking event, even though it's blurred. You can find the centroid and then map out and create an image with 10 times the resolution of a standard microscope. And we're using this and combining it with newer three-dimensional imaging methods that allows calculation of the distance in the z direction above or below the focal plane. And then you get this very different type of data out of this that is leading to a point cloud of information about the location of structures. And once we got this microscope in, we were able to start doing research right away. This is an example of a normal microscope image. And this looks nice because it's nice. It's a continuous image, but molecules are discrete things in the cells. And what we're seeing here is a blood vessel in the brain. And we're looking at the molecules that are around it. This technique, this STORM super resolution microscope technique, and combined with some other types of optical microscopy, allowed us to develop a hypothesis about what would happen if we removed with a knockout or we mutated a particular channel that's really important for how our brain functions, and especially the blood-brain barrier. And what we're seeing here is that we have increased microhemorrhages in this mouse model with mutations. And we used imaging to quantify this relationship between the proteins and how their arrangement has changed, and use that to generate a hypothesis about what would happen when we looked at the blood-brain barrier integrity in this example. And then we quantified that with imaging as well. So this type of microscopy, localization microscopy, as I mentioned, leads to a very different type of data that is point cloud based. It's not on a framework of voxels or pixel array. Instead, it's just floating points in space. And in order to be able to quantify and gain insights from the distances between your signal and this type of imaging, we use different techniques, different quantitative algorithms for this. And a lot of it actually comes from looking at astronomy and that type of research, and K nearest neighbor and data analysis and point clouds and separating features. I just wanted to show that how different this type of imaging looks when you're actually doing it. This is a video screen capture of me imaging a spherical structure in 3 different color channels, but the colors you're seeing here represent the distance in the location in Z space. And so this is a 3-dimensional, basically instantaneous holographic image, and we're building up these points in this point cloud to understand this mostly spherical structure, what its shape is. And then we're starting to see a different color channel. So this is a different molecule, different labeling is starting to build out. And we can see at 20 nanometer resolution, getting down to close to the size of many proteins, the relationship between these two structures in this sample. And then because of the nature of this, and it's We're looking at the relationship between points in space. It's very conducive to work running these algorithms on graphic processing units. And a lot of the algorithms for segmentation of three-dimensional images and these frontier of computational research is really good for analyzing and gaining insights from this type of data, microscope data. And so here I'm working in a virtual reality program that we're actually partnering with to further the development and create new algorithms for analysis in this program, and analyzing, this is between two brain cells and looking at the connections between the cells. So how do we use this in actual research? For example, here, working with Dr. Kai Oyama, we looked at the effect of the molecular arrangement in rat cardiac myocytes when these rats were either lacking thyroid hormone or resupplied with thyroid hormone. And you can see the dramatic and very well quantified effect, significant effect of the loss of thyroid hormone and then the replacement. So what we're doing at the molecular scale is we're bringing these molecules back into the arrangement that allows the contraction of these heart cells in the proper way that they should. And this is what we're using this at a molecular scale, being able to quantify the changes that occur. So I'm going to shift gears a little bit and talk about an exciting area that is very important for this particular type of microscopy, where we're looking at extracellular vesicles. And these are small lipid, basically packets of information that can be found in the blood of all animals and interstitial fluid. All organs basically generate these very small packets of lipids and phospholipids and proteins. And well, there's many unknowns in this area, and it's a really hot area of research. But what is important from this illustration that I'd like you to take away from this is we can look at these structures with electron microscopy, and we can isolate them and run biochemistry, sequencing, mass spec. But the really strong value of these structures for liquid biopsies, diagnostics, is the fact that we have the structure, but we have different proteins and different RNA, or even in some cases DNA, contained within these. And they're together in space. They're one packet of essentially very rich information. So if we can extract these, we have to be able to look at which ones are going together, which proteins are with which RNA, for example. And that allows us to decide and detect which organ and even which cell type within that organ these extracellular vesicles came from. And the type of imaging, this localization microscopy, because of the size of these, makes it one of the best tools to understand where these are coming from and how they're changing in different diseases. In addition to that, you know that these are, one form of these were used for some of the COVID-19 vaccines, but also they can be loaded up and engineered to deliver other substances other than RNA, for example. And all of the different methods that are emerging for the isolation and characterization of these different extracellular vesicles, it's going to be so important. It's going to be an area where we're going to be able to drive the research forward and develop new intellectual property and things that we can use to treat diseases and promote regeneration. So in my own lab, we are looking at, I study gap junctions and in the context of the brain mostly. And so we're looking and detecting new things and finding out that these are actually present in blood-derived, blood-borne, brain-derived extracellular vesicles, but they have a characteristic different size and they sort differently than some of the other proteins and markers that these extracellular vesicles are known to have. So this is an area of ongoing research, and we're generating hypotheses about how a different route by which these extracellular vesicles can be generated. We're working with a software startup company in the UK that's allowing us to develop high throughput methods for detection and characterization of these vesicles. And then in turn, they're working with outside companies to be able to find applications for these extracellular vesicles. Moving forward, I'm a cell biologist and studying how these are generated with the idea that potentially the brain cells such as astrocytes or tanocytes in the hypothalamus are producing these extracellular vesicles. And this happens to be an area where we can actually see these vesicles forming and see their association with a marker for extracellular vesicles. And this is a place in the brain that controls everything from metabolism, sleep-wake cycle, hunger, different things, just many different functions with homeostatic functions in the body are characterized or controlled by this part of the brain. And we see, and it's also a place where the blood-brain barrier is open. So it's a really interesting thing to see which extracellular vesicles are coming from this. But this is just a general, very powerful biomarker that we can use and combine with other imaging techniques to be able to understand how these are forming and how they change in these different conditions, such as autism spectrum disorder, neurodegenerative diseases, Parkinson's disease, traumatic brain injury. We know that these extracellular vesicles change in these conditions. And then coming back to what I talked about where we apply a therapy and try to quantify the effect of that therapy, we can access a window into the human brain using the blood-borne extracellular vesicle samples. And that combines with up through kind of the research stack with the exciting idea that we're going to be getting a functional MRI at NYT that is going to be able to look at brain activity and the changes of brain activity, and we can then see how that changes in tandem with these extracellular vesicles as one example. Taking it even further and trying to design therapies and see and gain insights into how those therapies are changing the extracellular vesicles or brain activity, we're currently working with medical students to develop exercise therapy modules and training modules. And this is just one example of something that we're building in virtual reality from scratch to be able to not only expand access to these exercise therapy programs that are being developed by faculty in health sciences and stuff like that, but here we have my daughters and we're boxing against a medical student who's in Connecticut. He's holding up the mitts that I sculpted there, those mitts, and he can clone himself out in virtual reality, which is something you can't do in real life. So this is a specific example of what you can't do in real life, but what you're not seeing is we're quantifying every movement that's being made here. And we can then take that back and see how it changes during training sessions. So the bottom line here is that imaging and the technology that we've been able to bring in and really reach out and collaborate, train, work together on new methods in microscopy forms an important link in the entire workflow or stack of innovation, from detection of changes in different disease models all the way up to engineering behavioral or other types of therapies for this. And so I really want to thank everyone who came today and thank the different funding and the great support that I've had from leadership.
Thank you, Randy, for that wonderful talk and letting us know all the great science that's going on in your center. Next, along the theme of today's event, our next speaker is Dr. Daniel Grant. He's a professor of orthopedic surgery, molecular medicine, urogynecology, and I'm sorry, these terms are too big for electrical engineers. And he's the Associate Director of Research at the Northwell Health. And without any further ado, Dr. Grant, floor is yours.
Thank you very much, and thank you to the organizers for the opportunity to present here today. I also want to thank my previous two speakers who kind of set the stage for me, so I appreciate that. Okay, my disclosures, I do have stock options at the above companies. And so let me start with autologous chondrocyte transplantation, which was something I developed in the 80s It was our first attempt at an orthopedic regenerative medicine approach. It was basically chondrocytes that were expanded mitotically, and then we had little or no biomaterials at that time, but we used periosteal flap to hold the cells in place. Now, that really spawned the idea or shifted our paradigm regarding whether cartilage will heal or not. Up until that time, it was considered absolutely impossible, but we showed that we could, in fact, heal cartilage. And now, fast forward to today, we've now, the third, some people would argue the fourth generation of ACI is now MACI, or matrix-assisted ACI, and it has, the chondrocytes are now grown on a collagen scaffold to allow for ease of implantation at a defect site. So you can imagine you can kind of roll one of these up like a paper clip, or like a roll up a sleeve of paper and then insert it through a micro portal through an arthroscopic approach. So that's really revolutionized how we can approach cartilage repair, one of the reasons. So now we enter the era of the orthopedic biosurgeon. So he's applying now these ideas of orthobiologics to the field of sports medicine, and with the idea that we can teach the body a better way to heal itself, and we feel that it has the ability to revolutionize how we treat disease. So in the clinic now, currently, as Dr. Rodeo mentioned, there's PRP, platelet-rich plasma. We have bone marrow aspirate concentrate, lipoaspirate, where we have the adipose-derived stem cells, and of course, umbilical cord, which is now has to be done under any umbilical or amniotic materials must be done under an IND because of FDA cracking down on some of these orthobiologics. And so we also, of course, have bone graft materials that are both osteoinductive and conductive. So I think the key question is, and as Scott mentioned earlier, is what exactly are we treating? Is it the ongoing degenerative process of osteoarthritis, or are we really looking to heal a focal cartilage defect? And each of these would have a different strategy or treatment algorithm for how to approach that. But what's interesting about the utilization of these orthobiologics by sports docs is a recent survey that came out that showed that a large portion of them a large proportion of the sports docs are incorporating at least one of these orthobiologics in their practice, probably PRP, due to a lot of times patient really clamoring for those procedures. And then a high proportion are showing increased use of their orthobiologics, and a quarter of them almost advertise the use of that, so marketing, et cetera. And of those, as Dr. Rodeo mentioned, PRP with low leukocyte pore is the most commonly used for is the most commonly used, and it's mostly used for osteoarthritis. But BMAC, bone marrow aspirate concentrate, is used in focal cartilage defects. So interestingly, the users of these strategies or these technologies firmly believe in their efficacy. So even though, as Dr. Rodeo mentioned, there's a very unknown or at least not a lot of consensus in certain areas, they still believe in their, based on positive meta-analyses. So one of the things, and as has already been mentioned, is this proliferation of these stem cell clinics all over the country, and of course, it's causing a lot of issues with whether these are truly stem cell, and to what extent are they really purporting to treat certain conditions. But what we need is, as also has been mentioned, we need to provide some evidence-based approaches to its utilization. So standards and that we can attribute to whether these cells are really working or not, and then identifying critical outcome measures that we can, and also the ability, for instance, to define various subtypes of OA, because OA is osteoarthritis is a long-term degenerative disease that's been hard to stratify patient cohorts into exactly the same level of degeneration. So that has also led to a lot of conflicting analyses about whether it works or not. So it's, as been also mentioned, it's a very hot topic, these orthobiologics. There's been over 4,000 publications in the last 3 years. All of our subspecialty groups, whether it's Arthroscopy Association, Sports Medicine, PMNR, they all, Academy just had a whole day devoted to this. And as a result, there's been the formation nationally of something called the Biologic Association. And we have formed, along with HSS right here in the city, our partners, these 7 groups across the country have joined forces to form what we call the BARB, the Bio Association Registry and Biorepository. And the goal is everyone will basically take PRP, whole blood, analyze that. We all have the same analysis equipment, so a Horiba unit for doing that. We're going to take the blood, we're going to analyze the platelet counts, then we're going to take a small portion of the PRP for ourselves, freeze it down, and then follow these patients over 2 years to see which patients are responders versus non-responders, and then be able to do a multiplex analysis of all those PRPs. Currently, we have 150 patient samples in the repository, and at Cedars-Sinai in LA, our colleagues there will be doing the multiplex analysis. So we have also, right here in the city, formed the Center for Regenerative Orthobiologic Medicine. My colleague Ken Zaslov is the head clinically. I run the scientific portion at Feinstein Institute. And across Northwell Health System, so 24 hospitals, a number of regions are all participating in our own in-house version of the BARB. So with regards to some of these orthobiologics, there have been pertinent advances in treatments, and here you see a number of things that have been, used for these things. Knee injections, rotator cuff's been very prominent, spine fusions or unions, bone union fractures that are at risk for a non-union, and then of course meniscus repair and cartilage repair. So let's just delve in for a minute to the field of osteoarthritis. We have a situation where we have these players, as Scott also mentioned, TNF, IL-1 are aggressive and negative cytokines. They're inflammatory. It results in eventual chondrocyte or the cartilage cell death, and eventually something called bone on bone, where we have a lot of other problems, including pain. So one of the things that we in our lab looked at was this concept of, well, how do you, and this is something that's led to clinical trials across our health system, and that is the concept of, well, we know PRP has some positive effects. What about if we add something that's been shown to also have a positive effect, if there would be synergy, that is hyaluronic acid injections, which are also approved for that. So, and our hypotheses were that we would see an increase in bone marrow, stromal cell and chondrocyte proliferation, as well as having an anti-inflammatory effect on those tissues and cells. So here you see a panel, and with regards to chondrocyte aggrecan expression, you can see that we saw with 5% unexpanded or I forget what it's called, but PRP that hasn't been lysed, if you will, to release all the growth factors, showed a very high rate of chondrocyte gene expression, aggrecan expression, as well as a decrease in a model of osteoarthritis of MMP9, that's matrix metalloprotease 9, which is a very negative, and matrix metalloprotease, it dissolves or degrades the cartilage matrix. So we see that has a strong effect. Also, another player in osteoarthritis, MMP13, was also significantly decreased at 1 week with our combination of PRP and hyaluronic acid. So that led actually to our health system incorporating that as a clinical trial too, and that's an ongoing thing currently. But the idea is that we showed a mechanism of action in the laboratory which could be translated to a clinical scenario. So as was also mentioned by our previous speaker, Dr. Stout, there's definitely this, and also to some extent Dr. Rodeo, here you see a time-lapse photo, micrograph of a endothelial cell and a bone marrow stromal cell exchanging a vesicle over a period of time. And we know now that those exosomes are important, and we heard about that before, whether they're AVs, extracellular vesicles, or exosomes, because you can see there's a rather a broad, well, not so broad, but certainly a spectrum of sizes for these. We've been looking at them in a number of different stem cells, most recently also synovial fluid stem cells. And again, the cargoes that these contain are important, and the therapeutic potential for osteoarthritis is significant. It's less immunogenic than even so in other words, you can isolate these exosomes independently of the cells and deliver them without any potential immune response from the host. And then we know that we can also manipulate these exosomes in culture by either preconditioning them in a situation where it might be inflammatory, so that they would produce more anti-inflammatory things. And we know that they have a number of different pathways that they can knock down: IL-1, IL-6, TNF, et cetera. And it also polarization of macrophages from an M1 to an M2 phenotype, which are very anti-inflammatory. And then we also know that it has effects in the subchondral bone. And if we think about osteoarthritis in general as a generalized joint disease, it's not just the cartilage. Although I showed that slide, it also includes bone, subchondral bone marrow, the synovial fluid, the synovial membrane, the meniscus, a host of other, and even in the fat pad within the joint act as a homeostatic organ system. So we have to really think about not just one pathway or another. But so we took some exosomes that we isolated from MSCs, and you can see in that panel on the panel A is normal control, and you can see there's also it's stained with safranin O and fast green. The safranin O is that red stain, which is very stoichiometric for aggrecan or proteoglycan matrix, so important for mechanical properties of that tissue. And then if we, in B and C, these are different doses of in a normal condition, and you can see you can even get more incorporation of matrix by stimulating them even on their own with just exosomes. Now, in panel D, you start to see a condition where we've introduced interleukin-1 and some other enzymes that mimic an osteoarthritic condition, and you can start to see more fast green. So in other words, we're actually degrading the tissue, and the aggrecan is leaching out. And you can see that with increasing doses in E and F, we can actually restore some of that or be anti-inflammatory for counteracting an osteoarthritic condition. And you can see in the other panels on the right, in G, you see that we're able to get increased collagen and aggrecan synthesis. So you have a degradation process where it's being depleted, but you're having an increase in synthesis. And then you can also show that you can see in panel H that we also show a simultaneous decrease in the MMP 3 and 13 negative so we're decreasing the enzyme degradation and increasing synthesis, so pushing the swing, if you will, toward a positive homeostasis. So we've shown here again in both a control and an OA condition that we can rescue aggrecan, collagen, and COMP, which is cartilage oligomeric matrix protein, an important maintainer transcription factor for protein rather for maintaining the cartilage matrix, very important, and you can see that in an OA condition, we can rescue that with a treatment of exosomes. And the same thing we see in reverse with regards to decreasing MMP3, 9, and 13, these key players in osteoarthritis with treatment of exosomes. So here's an example of, now we're moving into an in vivo model where we have we transect the anterior cruciate ligament, destabilize the medial meniscus, and you can see from that you start to get in panel C, and A and C, you start to see really decreased cartilage matrix. Remember, it's the red stain. You can see here, you start to see degeneration of the in an osteoarthritic model. And then here, with weekly injections, you can actually maintain and combat the effects of osteoarthritis. So now looking at some immunohistochemistry, you can see also that whoops, sorry, let me go back. You can see that the collagen 2 stain for immunohistochemistry is reduced in OA, but with exosome treatment, we can see that after 10 weeks, we have restitution or maintenance of the collagen 2. Same thing, MMP13 is almost absent normally in articular cartilage, but in OA, it's prominent, but it's decreased again in our exosome-treated animals. So now, as I mentioned, we've also moved into this concept of synovial fluid-derived stem cells, which are commonly obtained from effusions from patients who have had injuries, either trauma or from osteoarthritis. And you can see here that for instance, in an OA model, you see much less expression of aggrecan, collagen 2, and comp reduced, but you can recover it somewhat with exosomes. And the same thing here, all the negative cytokines are reduced in an OA model with use of exosomes. So we have a proposed mechanism of action for this, where we feel that the TXNP gene, which is inhibitory, is upregulated and results in autophagy, which results in chondroprotection of that. That's one mechanism that we're exploring for mechanism of action for how these exosomes work. Now, of course, you can imagine that there's a multitude of microRNAs, proteins, et cetera, in those and growth factors in those exosomes. So how do you translate that to the clinic is going to be very much a challenge, because it's how do you ensure, like, for instance, PRP, you take blood in the morning, and then from the same patient at night, you have a different, it's an orthobiologic, it could be completely different. The same thing with these exosomes. How do you know that you're isolating them from a specific cell population, consistently making the same cargo in each one? So that's some of the things that we're working on in our lab to try to do that. So these exosomes have a significant therapeutic potential, certainly for the treatment of OA. They're less invasive potential therapy for OA, such as knee replacement. Future directions include, we need to do some comparative studies from different sources, whether they're stem cells or other cells. Recently, we have found that they're present extensively in PRP, so we're going to be looking at that. What's the role of non-coding RNAs in alleviating OA? Because that's also present in these exosomes, non-coding RNAs. And then large animal studies are required for more, which have more comparable knee anatomy to the human, as well as more weight to do that. And then, of course, we've also been working on various biomaterials for delivery of these. And we also found that, for instance, you can lyophilize them onto a scaffold and then get them to work even a week later at room temperature, or even at 4 degrees. So we're exploring all aspects of that. So with that, I think I'll pause and take any questions if there are any. Thank you. Yes.
Just a perfect comment. Yeah, the lyophilization and maintenance of activity is really fortuitous, I would say. It's really exciting that you can lyophilize these and they retain activity better than even freezing them.
Yeah, and that actually has great applications for as an off-the-shelf availability for when docs want to maybe inject in their patients or in the OR or in the office even. So,
what's the most promising technology or techniques for
isolating
specific subsets that you want to target?
So we're still in the process of like figuring. Well, we're doing RNA-seq on a lot of our recent samples so that we can figure out what and then, as you know, RNA-seq with the analysis of all the different crosstalk and everything is complicated. So we want to target one or two specific microRNAs to figure out if they're effective or not. Because at the end of the day, that's really what is going to get approved or not. But as far as isolation goes, we've done everything from 24-hour, 100,000 G things to recently more membrane-based and so forth. I think that's still also a work in progress as far as what art and science have to, the merger. Sometimes it's more art than science, but we're and even like isolate, like when you have, I didn't show any, but when you get in and out, we have a particle analyzer, if you will, which is you put the samples in and then it gives you characterization curves like the diameter that's most and what the spread is, and it's still kind of primitive in my opinion. Like how do you isolate that? I guess one of the things would be some kind of specific antibodies for something that you could pull out. I just don't know at this point, but we're grappling with it. Okay, any other questions? Okay, with that, thank you. Thank you.
Thank you. So our panel moderator today is Dr. Michael Nizik. He's the director of the Entrepreneurship and Technology Innovation Center, ETIC, here at New York Tech, and the Cyber Defense, ETIC and Cyber Defense Education. Michael has been a force of change here in the College of Engineering. I hope if you haven't looked at the presentations in the back of this room, in the entrance during the networking hour, you do that. It's doing amazing work in terms of innovation in multiple areas that are overlapping the topics of today's conference. And without any further ado, I ask Mike to invite his panelists on the stage here. Michael? Thank you.
Thank you so much, Dr. Beheshti. Thank you. I better see my students clapping. That was. So hello, everybody. It's a pleasure to be here and be your moderator for this panel. There are going to be people up here, so we're going to call them now. First off, we have Miss Dr. Carmela Stevens. Carmela from Carter, Duluca and Farrell, LLP. Sorry about that. Carmela, why don't you have a seat? Thank you. We have James Liu, the founder and managing director of EOS Bioinnovation. James, pleasure to have you. Pleasure to meet you, too. Then we have Charles Goulding, Esquire, and CPA, the president of R&D Tax Savers. Charles. And then finally, we have John Pennant, CPA, and partner in charge of the National Technology and Life Sciences Group at Eisner. All right, great. Let's have an applause for our guests, our panel. This is wonderful. So as I'll kind of open a panel, I just wanted to, you heard about some of the things that we do at New York Tech and the Entrepreneurship and Technology Innovation Center, or ETIC. I just want some of the students who have worked on these innovative projects, kind of raise your hands, there they are. Oh, they're on one spot. That's amazing. So for everybody, and especially for the students, from all New York Tech students, this group of professionals up here today, it's representative of a couple of things. It's representative of innovation and technology, and not just building new tech, but protecting it and licensing it and everything that we're going to have to learn about. We don't just build things. We don't just invent things, right? We also have to somehow make it a reality so that the rest of the world can enjoy them, right? So today's panel, we're going to focus on that. And I'd also like to just, on a personal level, these events, if you've liked what you've been hearing, they don't happen very easily. It takes a lot of work, and it takes support of our sponsors and folks like you have up here today. There's a lot of the companies here are sponsoring this event, so we're really grateful to have this. Thank you again. One more round of applause for our okay. So that being said, I think what we have is a series of presentations, if I'm not mistaken. So we have Carmela, you're going to go first, I think, if that's okay. And Carmela's going to talk about some of the work they're doing at Carter, DeLuca, and Farrell. Yes? Okay. Yes. Please welcome Miss Dr. Carmela Stevens.
So I'd like to, at the onset, just thank the organizers for providing me with the opportunity to talk this afternoon. And I'm going to be talking about bioprinting, and which is a very exciting field right now. And my talk is going to focus on intellectual property issues that come up with patenting of bioprinting. So, what is bioprinting? Bioprinting is the process of creating human tissue and organs using a 3D printer. To just step back and talk about 3D printing in general, there's been a upswing in the use of 3D printing for very many different applications. And, you know, when it first started, it was the printers were expensive and the printing materials were expensive. But through the development and innovation, the prices have come down. And so there are many, many applications outside of bioprinting where 3D printers are being used. And those include in the classroom. So little engineers are being trained to design and print different things in the classroom. Manufacturing, there's lots of applications. There's building of structures, of building materials, clothes, shoes. There's lots of applications for 3D printing for those types of products. And an advantage is that the products can actually be designed for each individual. So there's perfect fits. Art and jewelry, there's just a wide range of different applications for 3D printing. So getting back to bioprinting, bioprinting can be used to print tissues and organs that can have many different uses. They can be used in research. They can be used for drug development and testing, and in the future for organ transplants. So what I think is exciting is that these different biotissues can be used in drug development and testing. This can be a cost-effective and ethical means to identify and test drugs without the use of animals in the lab. For organ transplants, it can be very useful because you can start with cells that are actually derived from the subject that is in need of the transplant, and so you eliminate the chance of organ rejection. This is a very simplified set of slides that just goes through the bioprinting process. So you start with a computer model or scanned image of the tissue or organ, which is loaded into the printer to create a blueprint of the object. The living cells are then mixed with a gel or a matrix or growth factors, whatever's needed to create bioink for the printer. And then the living cells or combinations to be used will be depend on the type of organ or tissue that's to be printed. And then the printer deposits the bioink in a thin layer through printing nozzles onto a platform to create the final product. Following the printing of the tissue or organ, additional steps may be needed to solidify and incubate the printed tissue or organ. So patent protection is a key determinant impacting the progress and development of 3D printing technologies. So with patent protection, more investments and resources will be committed to enhance the state of these technologies. And of course, without patent rights, the progress of 3D printing may be hindered. So it's going to be unlikely that people will want to put make the investment in time and money to develop the 3D printing processes if there's no chance of patent protection at the end of the process. So it's important. So now just getting into the specifics of patent protection, and I'm going to be talking about patent eligibility specifically. So the United States Patent Code allows, and this is 35 U.S.C. Section 101, allows for patents on any new, useful process, machine, manufacture, or composition of matter. So you can get claims on composition of matter. However, human organisms and products of nature are not patentable eligible, but variations of the naturally occurring organisms may be. So this just means that you can't patent cells, you can't patent proteins or DNA because those exist in nature. So if somehow you modify the cells or the protein or the DNA, then that can become patentable subject matter. So just focusing on 3D printing. So what is patentable subject matter? So certainly the process of creating the bioprinted organ or tissue would be patentable. So you're not, in that case, you're not focusing on what you're making, but rather the process that you're using for creating the bioprinted organ. The printers or the printed components used for the bioprinting, certainly the printers would be patentable, and the components, I'll get to that in a slide ahead about whether that would be patentable. So this is just an example of a patent that issued to a process, and it's a very broad claim. So it's a method of producing a tissue construct comprising depositing cell aggregates from a dispensing device and allowing the cell aggregates to fuse. So this is an example of a process claim that actually went through the patent office, and the claim was allowed and the patent issued. So what about the pre-printing materials? Those might face a patent challenge. So that would be the cells or the matrices or any factors that you might be adding into the printer to form the printed organ. So the question becomes, those inks or those printing materials, do they exist in nature, or have they been manipulated before they're used in the bioprinting? So matrix materials may not be materials that exist in nature, and so that may not be maybe not be an issue. If the cells that you're going to be using to form the organ have been engineered in any way, so perhaps they were engineered to express recombinant proteins. In that instance, those cells would be patentable. If you're starting with stem cells that you've isolated out of nature, those cells probably would not be patentable. But if you, again, engineer them in some way to change them from their natural state, those cells could be patentable. So then the question is the organ, the actual organ or tissue that you produce, is that patentable? So certainly attempts to patent a human liver would be denied patentability based on 35 U.S.C. 101 because that exists in nature. What about patenting a lab-created liver that functions and resembles a human liver? So what it boils down to, as far as whether that would be patentable, is whether the tissue or the organ has markedly different characteristics from the natural state of the tissue or the organ. For example, scaffolding live stem cells on biomaterials such as polymers, this wouldn't be something that would naturally exist in nature. And so that product would be patentable. An organ that functions better than its natural counterpart. So if somehow in engineering the bio organ, it works better, maybe it produces growth factors or proteins or enzymes, that type of organ would be patentable. So there's a number of issues that might come up. One of them is one consideration producing bioorgans is, does it go against the National Organ Transplant Act, which prohibits human organs for sale or purchase. And I think that it's generally agreed that that would not be an issue with the bioprinted organs because it's not a natural product. And then the final question is, what about infringement? So what about the patient that actually receives the organ? So let's say that there is a patent that covers the process and covers the actual bioprinted organ, and a patient is transplanted with the organ. Does that patient infringe the patents? And in general, this would not be the case. And so it's the manufacturer that is liable for infringement. And that's in any medical device, that's usually who is responsible for the infringement and would have to pay the damages. So there's no concern on the part of the patient for taking the organ, for getting the organ or the tissue.
Thank you for Carmela. Okay, next up we have Jimmy from EOS.
My name is Jimmy Liu. I am the co-founder of a company, or actually a venture fund called EOS Bioinnovation. We actually got started last year with funding to invest in regenerative medicine projects. And so I guess you've heard today some folks talking about their own technologies that they're bringing onto the market. You've heard about, I guess, efforts by the state and city of New York to help the overall biotech ecosystem here. You heard from Carmela. It's very important to have an attorney behind the companies that you start, because you don't want to get into infringement issues before. And I think John mentioned being able to provide the accounting advice that you need at the very beginning. So I'll give you quickly the sort of the venture perspective. Any startup that wants to get going needs money to get started, pay bills, pay for development of your programs. And so sometimes you get non-dilutive funding from the NIH and get started. Other times you really need to get the outside funding to support your payroll, to support your programs. And that's the business that we're in. But we're focusing on regenerative medicine. So for any startup, I mean, we are kind of a startup too, first time fund. You have to think about, okay, is this particular business that you want to get into the right time to get into that particular space? And so we really felt that regenerative medicine was the right time to get in based on recent developments and recent approvals in the marketplace. So by background, I've been in the venture investing business for 25 years now. Started in technology. I helped with the founding of the venture arm of a company called Acer Computers. They're Taiwan-based, and so they're looking at ways to expand their footprint and looking at new technologies. So they started this venture arm internally. Then spun out and created a $260 million venture fund to focus on technology investments both in the U.S. and in China. And then in 2010, I joined a firm called W.I. Harper and kind of pivoted into the field of healthcare. Now, the chairman at that time asked me whether I was willing to go ahead and head the healthcare team at that time, because the person that was the chair of that particular area had left for a much larger venture firm called OrbiMed. And so when our chairman asked me whether I could help the team look at investments in the healthcare space, he said, I need somebody with experience and gray hair to lead the team. I said, I have gray hair, but I don't know if I have the relative experience. But he said, that's okay. You know, you've got young folks, really smart people on your team. You can learn from them, and they can learn from you. And that's how I got started in the healthcare space, and led investments and sat on the boards of healthcare services companies, also diagnostic medical device companies, and more recently when I was at W.I. Harper, focused on AI-based drug discovery companies. So who are we? Basically, we focus on regenerative medicine, as I mentioned, and we have a pretty broad definition of regenerative medicine. Basically, gene and cell therapy is one of our areas of focus, as well as tissue engineering and biomaterials, and also small molecules that help with the endogenous repair mechanism of the body, as well as those compounds and molecules that help to arrest or slow down or reverse the degeneration processes. And so basically, we got the idea to start this particular venture from a friend of mine, Brock Reeve. Brock was the executive director of the Harvard Stem Cell Institute. He had been there for 16 years, and he and I talked about whether it was the right time to start a venture like this. And in the succeeding slides, I'll sort of go into a little bit of a detail why it was something that we felt was the right timing. And if you're in venture investing, you have to invest at the right time. There are lots of investments that I've made in the past that may have been great if it was 10 years later, but they were started too early and the marketplace was not ready for them, and so therefore they didn't work out. But we felt that this was a good idea based on his relationships at Harvard. He had been there for 16 years and knew basically most of the faculty members that were associated with the Harvard Stem Cell Institute. It's about 350 faculty members throughout the Harvard University ecosystem. And you can see some of these hospitals that Harvard Stem Cell Institute is affiliated with the Harvard University, as well as some of these other institutes, including the Wyss Institute and the Broad Institute. And we also look at projects coming out of the School of Engineering, the Paulson School of Engineering and Applied Science. And so we thought it was the right timing, and then the relationships that Brock brought from Harvard was very important to get started. And then also, you need investors. So for those students here who may not understand venture as well, folks invest in us in order for us to go ahead and invest in promising companies that will ultimately be purchased or go public to return money back to our investors. And so a good friend of mine who was a general partner, a former general partner of Kleiner Perkins, China, who had an extensive background in life sciences. He and I worked together for 12 years, co-investing in companies, working on projects together. He was very interested in this project and agreed to anchor our investment, meaning being a significant investor in our fund. So why is it the right timing? I think earlier today we talked about, this is still a new area, a budding area. And so, but these headlines, you can probably tell, there have been advances in recent years that give this particular field a lot of promise. And so, dealing with Alzheimer's, with diabetes, with, vision, some of these are headlines in the New York Times that give us hope that this is a good area into investing in early stage projects. And we do get in very early in terms of the time that we get involved with companies, and I will mention more of that later. This is just another slide to tell you about the sort of the acceleration of this particular field. And obviously in the biotech space, you have to necessarily get the approvals by the FDA before you can put your product into the market. And so this just gives you some idea about the growth and the acceleration of some of these approvals. And then the lifeblood, as I mentioned earlier, is having your portfolio company exit through an M&A event or a mergers and acquisition event. And this slide kind of gives you some idea about what is happening with respect to some of these biotech companies that have actually partnered or collaborated with large pharmaceuticals. Big pharma, you know, they develop their own drugs, but then they also are looking for opportunities where they can actually buy biotech companies that have put a lot of work in there, and then be able to then buy them and then continue the programs with more resources to bring clinical programs into the market. Again, this gives you some idea about the Harvard Stem Cell Institute and some of the companies that came out of the institute. And then this slide gives you some sense as to where we may actually fit in the overall ecosystem from the Harvard Stem Cell Institute. Under the sort of new co-ready IP is where we actually have a nice sweet spot where we focus on companies, and even companies that have not been formed yet. Because our feeling is that we understand the space well enough, working with really proven investigators and scientists, understanding the science behind their work, to be able to work with them, maybe fund them initially with $250K to reach a proof of concept, and then help form the company with them, and then be their partners and essentially co-founders with them to help them grow, help introduce them to the relevant lawyers, accountants, scientific advisors to be on their scientific advisory board, and also board members, and even recruiting a possible CEO to start the company with them. But as I told my partner Brock, who spent 16 years at Harvard, Harvard doesn't have a monopoly on all the good deals, okay? We have to always look for promising projects all around, and so I'm hoping that I can meet many of the folks here to see if there are really exciting projects that we can continue to, have discussions over. And so we're always looking at building relationships with top research and academic institutions throughout the country and even abroad too. So here's just a snapshot of some of the projects we're working on. And so even though I showed you a lot of progress in the gene and cell therapy area, but we are looking at a project relating to cartilage repair, and this was something that was discussed in the last couple of sessions in this conference. And we also are looking at projects focusing on CNS and also wound healing, wound repair, and drug delivery. Quickly, this is the three people that I mentioned. James is the chair of the company. He was the one that provided the initial funding for us, and then Brock and his Harvard relationships. So we pride ourselves in helping companies all the way through, even though we focus on early stage companies. We feel that our overall skill sets and our experience, plus the network around us, can help companies go from the very beginning to an exit and even beyond an exit. So this is just a summary slide in terms of having the right, being in the space at the right time, having the right team to deliver it, and also being able to understand understand sufficiently the overall space that we're working in, and then being able to provide the right returns for our investors, but at the same time, the successes will translate to patients, better outcomes for patients, and also helping with their health span and lifespan. Thank you. Thank you, Jim.
Thank you so much, Jim. Okay. Next, we have up Charles from R&D Tax Savers.
Yes, thank you. I'm glad actually to have the young people in front of me here that have completed your projects, because I'm going to talk about some opportunities that might surprise you. We're in a different world. You had an excellent presentation from the New York City Economic Development individual in the back there. He's waving his hand. And he's exactly right. If you go back 5 or 6 years ago at our firm, R&D Tax Savers, where we specialize in helping companies get tax incentives and cash rebates, we've always done a lot of life science work across the whole panoply of life science, pharmaceutical companies, labs, medical device companies. But up until 5 or 6 years ago, we weren't working in our backyard. We were working in Boston. We were working in California. We were working in New Jersey, where we probably have about 100 clients. Why? All our brilliant clinical people, and this was touched on, are working in the leading hospitals that we have. They've got great ideas, but they couldn't create a business because they're busy doing their clinical services. They had an hour or two a day, and they wanted to walk across the street like they could in Boston or they could in California and walk into a wet lab and work on their project. But you couldn't do that in New York City because we had inadequate wet labs. And you're getting a presentation, I think, after this about one of the firms that's filling that gap. But that's been the big change. And look at the amount of capital he talked about that's coming into the area. But with that, I'm going to get started with the presentation. First of all, I'll talk about tax credits because I know you're scientists and engineers. A tax credit is a dollar-for-dollar reduction in your taxes. It's the best thing in the tax world. The only other incentive is enhanced deduction, which is only worth your tax rate times a deduction. But tax people, we like tax credits. Now, you can get a tax credit for your current year. You can go back 3 years, but you can carry them forward for 20 years, which is very important in the bio area because a lot of biotechnology business ideas take a long time. They take more than 4 years, 6 years, 8 years. So you want to generate your credits. You want to work with an advisor who can help you generate your credits and then carry them forward so that when that payday comes, you're all ready. If you don't do that, the trouble is you work all these years, 7 or 8 years. You finally hit a home run. Now you're confronting a big tax bill. You didn't generate the R&D credits you could have generated if you knew about it and generated them from the onset. And the good news on the R&D credits, R&D credits were enacted in 1981. I did my first R&D credit study in 81, so I can tell you with a straight face, nobody's done this longer in the United States than I have. But the problem was before, they were temporary. They would always sunset. And you didn't know whether you had a credit or you didn't have a credit. But now, as of 2016, they're permanent. So now you know you can get your credit prospectively year in, year out. All right, so I already told you they're permanent. There used to be some problems with minimum tax. That's off the table. Now I want to talk about the startup payroll credit. Why? Because you can take it as a credit or you can get it in cash. Okay, up until December 31, 22, it was $250,000 per year for 5 years. Now, as of the beginning of this year, January 1, 2023, it's $500,000 per year. So you can get $500,000 back in cash rebates. In order to get those cash rebates, you have to have employees. And that's one thing about a lot of these tech businesses. They require smart people that are highly paid. And your rebate is in the form of cash payroll taxes. Go ahead, please. I just covered this. The litmus for the startup credit is you have to be less than 5 years old. The 5 years begins from the year of first sale. There's a lot of biotech companies that they may be in business 10 years, but they may not have their first sale until after 8 years or 7 years. So for 5 years, you can now get, prospective 5 years, you can get $2,500,000 in cash from the government if you structure it right. Go ahead. This just gives you some of the technical pieces of it, but mainly the startup situations we work on are tech companies that have a lot of engineers, designers, scientists on staff. Go ahead, Melissa. All right, this may be hard to read, but this will give you some of the examples of the startups. AI is a hot topic now, but when we worked on this leading AI company is before, the chatbot everybody's talking about before. And if you go through, you'll see a mixture of all kinds of companies. There's a building foundation company in there. Carmela mentioned using 3D printing, which is increasingly used for the process cement. Lots of software companies, a health software company. Point being, any kind of company can get the startup R&D tax credit if they're innovating, but it's particularly good for life science companies. All right, now this one I want to talk about because I'm amazed at how few times it's utilized by New York startups. New York State has a new $1,500,000 tax credits, $500,000 a year for 3 years. Business has to be less than 5 years old, and you have to fall into one of those boxes. I think if you look at that list of companies and you study it later, just about every company that had a product out on that in the exhibit area or all the companies presenting here today are going to fall into one of those boxes. Let's do the math. Feds are going to give you 2.5 over 5 years. New York State's going to give you $1.5 million. What do we get as possible cash from those two programs? $4 million. So although I think Jimmy gave you excellent advice, you do have to time everything in life. But sometimes you just got to go for it. And if you have a good idea and you've got potential of $4 million to help you, think about it. Go ahead. These are the high growth areas that we work on. Health software. I'm glad Carmela presented today on 3D printing. We have about, last week, 50 companies, 50 clients that use 3D printing. We happen to be the staff writers for Fabaloo. If you ever go on Fabaloo, it's one of the leading 3D printing websites. Why do we write a lot about 3D printing? Because if somebody's buying a 3D printer, they're either developing a new product or improving an existing product, which is perfect for the R&D credit. We love 3D printers. We'll go back for a second. We do a lot of dental lab work, all kinds of life sciences, telemedicine, big area for us, analytics, kinds of stuff you're aware of. Go ahead. Now, this is the meat of it. How do you get an R&D credit? You have to have technical people doing one of the things listed in category 1. They have to either be developing a new and improved product or a new and improved process or new and improved software. Well, everybody's doing something on software here, whether it be AI. You had discussions of virtual reality in one of the previous presentations. You have migration to the cloud, new ERP systems being put in. So we see software every year, everywhere. Now, an invention counts. Carmela is your invention expert. She's going to help you get a patent, and your patent costs are going to be part of your eligible R&D tax credit. Your project has to be technological in nature, which for the students here and the scientists here, by definition, your projects are technological in nature. In nature, it has to be an elimination of uncertainty, not uncertainty that you can't do it, uncertainty as to how you're going to do it. And you have to show a process of experimentation, which you guys are familiar with. It's a scientific method. You have to retain your notes, retain your lab notes, and document. How did you go about the process to develop your new product or your new process? What are the costs that are going to help you generate a credit? Employee wages. It's primarily the technical people's wages. Supplies. That's going to be items consumed in prototypes and making your new products. And then third-party consultant payments. That could be outside labs to do testing, but most frequently with our clients, the outside consulting payments are to software people. Most of the companies we work on do not have a deep IT staff, particularly the startups. So if they need some help in their software area, they use an outside integrator, and that outside integrator's cost could go into the credit. This again is a list of some of the major areas we work on. We do a lot of work in analytical laboratories. Because we represent many dentists with laboratories. We do a lot of intraoral camera work. We do a lot of mouse model work. We represent one of the biggest mouse model companies around. Again, give you an idea of where the tax credits are. Go ahead. Patents. We love to see patents. That's why we work very closely with George Losardos and with Carmelo, because if you know the standard for a patent, to get a U.S. patent, what you've done has to be innovative and non-obvious. That's a higher standard than the tax standard. So once you have a patent, by definition, you qualify for an R&D credit. And frankly, it can't be challenged by the U.S. government. Why? The IRS is a department of the federal government, but so is the U.S. Patent Office. So if the U.S. Patent Office has said, hey, what you did is innovative and non-obvious, you're done. IRS can't challenge. Their brother organization, sister organization, has already said what you've done is innovative. Go ahead. Key with an R&D credit study, just so you're aware of it, is you have to have documentation. That's what we do on behalf of our clients. There is an 80% equals 100% rule. So to the extent you've got some key people that are doing nothing but developing your new and improved products, you can actually put 100% of their salary into the credit calculation. I think the point here is our government wasn't nickel and diming when they come out with this credit. We talked about the 3D printing. We publish, this is done by our staff, a 3D printing article almost every day. This will show you some of our articles in the life science and biomedical space, so you get an idea of what's going on. That's some of our medical related publications, which you can look at any time. And our firm is, the market, I didn't even know this until I started the firm. The market calls us an interdisciplinary firm. Why? I'm sort of a traditional tax guy as an attorney and a CPA. We have other attorneys, we have other CPAs, but most of the people in our firm are engineers. Warren in the back is one of our medical engineers. Melissa is one of our biomedical engineers. We have other biomedical engineers, but we have electrical engineers, chemical engineers, environmentalists, we have a nuclear engineer. Why? We're working on this odd intersection of tax and engineering. We are members of, I call it SOPI, the Society of Physician Entrepreneurs. That's a great organization. They have a strong chapter here in New York City. That organization is focused on helping physicians who want to become entrepreneurs. Okay, and it's a good place if you do have an idea and you want to get an understanding of how do I make it my idea into a business. I'd suggest you join. So we're also the writers for Thomson Reuters, for Commerce Clearinghouse, and then for Fabaloo on 3D printing articles. That's it. All right. Thank you for having me.
That's great, Charles. Thank you. Charles, one question before John comes in. If a company has no revenue in the first several years, are you saying that the tax credits are applied only for future revenue, or do they benefit from it somehow?
Well, you don't trigger the beginning of the 5 years for the cash rebate. But if you have no, I'm presuming you're talking about a situation with no revenue, but you have expenses, you'd still generate a credit. And you can record the credit on your books, which is going to make your balance sheet look better. But then wait till you have the requisite number of employees and try and maximize the 5 years. You want to try and maximize the optimal 5 years you can for the $500,000 a year. Gotcha. The guys that we have that have gotten the big credits tend to be people that have had big cash funding rounds. Because you're not going to get $500,000 a year back in payroll taxes unless you have 100 engineers, 150 engineers. So those are not mom and pops. They're people that, a couple of our clients in that area, they've already sold out one company for big dollars to Google or Meta or somebody else, and then they come back in now and hire 50 more engineers.
Great. Thank you so much. Okay. Thank you so much. And now we have John Pennant from Eisner Amper. Welcome, John. You want to use this one? You got that one.
So who here wants to have for the last part of the presentation to talk about accounting? All right. One person there, liar. Liar. Nobody wants to talk about accounting. So we're a big accounting firm. We do all these things. Cool. We help companies at early stage life sciences companies, outsource work, tax work, help them with technologies to help enable everything. Cool. Okay. Outsourcing is a big thing. I'm going to skip over here and get to something that maybe will be more interesting, I hope, for everybody, which is, how do we get to a position where Jimmy's going to help us, right? So you guys got an idea. You're trying to pull something together here. How am I going to get some funding for this, right? So how am I going to pull this together? So first thing you have to think about is, like with any pitch that you're going to put together, is what is the problem you're going to try to solve? Okay. understand what else is being done to do that. So if someone comes in, and I'll also from the perspective of, I'm an angel investor in Mid-Atlantic BioAngels, so part of my talk will be talking more from the perspective of an angel investor than an accountant, because nobody wants to talk about accounting. Rat bastards, all of you. So you're trying to figure out what the problems you're trying to solve, right? So somebody comes in and says, I'll ask a question. Who's your competition for what you're trying to solve? If you say no one's doing what I'm doing, you can immediately leave because you're wrong. 100% wrong. It may not even be somebody in a biology department or engineering department. It could be a philosophy company, it could be a communications company, it could be Walmart. Somebody is working on that problem and is probably way ahead of you. If you don't know everybody who's working on the same thing that you are already behind the 8 ball. Okay? So then you think about, we talked a little bit about this earlier, right? So if you're going to try to ask someone for an investment, okay, what are you going to do with that money? So this is my accounting background, right? So if you can't explain to me exactly how much money you need, it's an estimate, I know, and what you're going to do with that, and what you're going to accomplish at the end, I cannot invest in you, right? So if you say, I'm going to work on this thing, we're going to be able to get FDA approval. Okay. How long is it going to take you? Do we need 20 million, 200 million? Do we need 10 years, 5 years, 100 years? What do we need? If you don't know the answer to that question, you're not ready to have the conversation with an investor. Doesn't mean you know the exact right answer, but you have to talk to some people. What is a phase 2 clinical trial going to look like? I don't know. Talk to somebody from an FDA consulting practice. Yeah, those are typically, 200 people trials. Got to monitor them for a year. You can put a cost estimate on that and you say, I need $20 million. You have a basis for your decision. As an investor, I may not agree with that. I may challenge you. I may say we need to raise twice that or whatever, but at least you've done your homework and you've put yourself in some sort of a parameter. So I know what I'm looking at. If I'm an angel investor and you say I'm going to need $2 billion to do a CNS trial of 5,000 patients, which I have to monitor for 7 years, I can't invest in that. I'm an angel investor. When I give you a tiny little bit of money, you need billions. Forget it. Wrong person. Okay. So really understanding, you have to have command and control of your budget. You have to understand what your budget's going to look like and what you're going to accomplish with that. Okay. So that's pretty cool. So you also think about, whenever you hear people talk about pitches, you think about team and TAM, right? We talked about team earlier. Great science is terrific. If you don't have a team that can execute the plan, doesn't matter. I can't invest in you. You got to find people to supplement your skill sets. Whatever those skill sets are, you have to surround yourself with people who can help round out all those things so that I, as an investor, have confidence that this team can execute the program. Okay, really important. We're looking at an investment now in our group. We love the technology. We love their marketing person. We hate the CEO. We are not going to invest in this company if that guy stays. We have to either convince him to step aside or we're not going to invest. He cannot, we believe he cannot execute the plan. So a little bit of that is sort of self-awareness, self-coaching, professional coaching, whatever, but really important. Okay. And then you think about TAM, right? Your total addressable market. So is the problem you're trying to solve a big problem or a little problem? Goes to how much money you're going to need, right? So if you're solving a tiny little problem and you say, I'm going to need hundreds of millions of dollars to get there, how am I ever going to get my money back? Right? So there has to be a relationship as to how it all kind of comes together. Okay. So then you start thinking, okay, so I've got an idea. I've got my budget in mind. I know my market. I've done some market research. I got some folks from the MBA programs to help me pull some things together. I kind of have an idea of what I'm looking for now. And my I have a relative who works at Goldman Sachs, so I'm going to go talk to Goldman Sachs about this. I need to raise $500,000 to advance a project towards proof of concept. I'm going to talk to my buddy, my family's friend at Goldman Sachs. Okay, they do $500 million deals, not $500,000 deals. So you have to know who your investors are. You have to do a little research. Has to be relevant. So if you're talking to, Qualcomm Ventures about your biotech project, you're talking to the wrong people. They don't understand it. You have to be focused, right size, right stage of development, right industry, something that's helpful. It's even more helpful if you talk to them when you don't need the money. So especially as a really early stage company, if you can say to them, listen, I'm working on this really cool idea. I'd like to introduce you to it. And I'd like to keep in touch with you over the next few months to tell you how we're doing against that. Start the relationship early. So if your family friend knows somebody at that earlier stage VC that might be relevant, that's really helpful to say, you already found this. You already found the right investor. So if you can have the conversation, build a relationship early on, show that you've actually accomplished what you've said you've done, especially as an early stage entrepreneur, you've now started to build some credibility, since as an earlier stage person or a student level person, you haven't had successes before, right? So you need to build your credibility up. Okay? So having sort of your command of your budget, know your target investment audience, building those relationships are a couple things that are really, really important. Just a couple of things on the lessons learned here, right? So one of the things you want to do is as early on as you can is to take care of your organizational structure. So set up your entity the way you want to. So get some advice from counsel or from accountants or whatever, whether you want to be a corporation or an LLC or whatever. Whoever you want to give shares to, your partners, your co-founders, whatever, stock options, do it early, document it, put it in your vault. So I was subpoenaed 3 times over a 4-year period for a company, really innovative company. Ultimately, they sold for $600 million. The founder had his personal tax accountant come in and be the financial person. He was clearly there. The financial person says, I was promised 10% of the company. Not a single piece of paper proved that. The founder said, after $600 million, I'm taking it all. You get none. 4 years in court. Financial person wound up with 0. Not documented. And if it was documented, it would have been terrible tax consequences. So if you're going to do something, do it right, document it, put it in the vault. You don't need to do a whole lot of stuff on like accounting and books and processes and all those types of things, but have books. So early stage due diligence is not going to be like an audit, like you're going through the SEC. You got to have books. If you have people on payroll, pay your payroll taxes. Do all those kind of real simple, basic things. Keep it nice and simple, but follow the rules. Have a deal room ready. So set up a Dropbox, some sort of a system. There's really good ones out there. Put all your stuff in there. Have it organized neatly, properly, with the right dates, with the signatures. Keep all that stuff there. So if somebody asks you, you have it. It's clear. They can go look at it. You don't have to go scramble and try to find it. So simple but an easy way to do that. So funding agreements. So I'll give you another example of a really bad lesson learned. So I had a client who had done a preferred stock offering. They were in a biosimilars business, was going to try to build out a plant. So he raised some money on a preferred stock offering. I asked him how the deal terms were. He said, straightforward, simple preferred stock. Couldn't have been an easier deal. I said, awesome. So I read through the deal. So it says here, you have to pay them back. No, I don't have to pay them back. It says right here, you have to pay them back. And not or, and you also have to give them an equivalent number of common shares. So it's a double down. So you have to pay them back their cash and you have to give them shares, both. So you've done the worst deal in like the history of all deals. That's not what I asked for. Well, you probably never read it, did you? So take care of business there. And so the final thing I'll just sort of mention here is know what you're asking for, right? So it gets back to that budget piece a little bit there. What are you going to accomplish with that money? Have a really clear message in hand so you can say, with this money, I'm going to accomplish preclinical activities X, Y, and Z. And when I'm done, I'll be able to do this. So your money will get us to this point. You want to have a really clear definition as to what that ask is. Okay. So I will stop there. I'm not sure if I went over or under, but that's kind of off script here. So.
I think we're right on time. I do think we have a few minutes, not long, but a few minutes for any questions that we might have. So, first of all, a round of applause. Thank you. Thank you so much. So do we have any questions in the audience? Yeah. Randy.
Go to the student maybe first. I'm assuming. Sorry.
Yeah, if you want to go first.
Okay. So I have a question for Charles. When you were discussing the benefits of a patent on the eligibility and attainment of the tax credits, when normally, oftentimes I would say, we sign over the invention and the patent to the institution as a faculty member, an employee. Does that negate any kind of benefit, or if you're the founder or have some prominent role in the company, do you retain some benefit of having those patents?
Yeah, it's really based on facts and circumstances. If in the typical situation I think you're describing, you're going to have some ownership of some underlying technology. It's the process or other knowledge assets. I mean, there is a requirement that in order to get the credit, you have to have some interest in the technology, but it's not extensive, but it has to be, significant.
Okay, was there one more question? I forget. Yes, go ahead, sir.
This question is for Dr. Pamela. You mentioned, sorry?
Go ahead.
Oh, you mentioned that you need a patent specifically for 3D printers when it comes to like the cells or like the product the cells are going to be used to make. However, it seems like for this kind of technology, you need to keep the cells alive somehow. So instead of patenting the cells in the products, why can't you just patent the device is keeping the cells alive and producing them?
That would be something that we can patent. So if the device is needed to keep them alive, that would be, perhaps that device would be patentable.
Okay, all right.
I'm not sure that I heard the beginning of your question, but you don't necessarily have to have a patent on the cells that you're going to be using in the organ, let's say, unless there's somebody else has a patent on it that you might gets complicated. Somebody could have a patent on those cells that you want to use, and then you'd be infringing those patents. So that's something you'd have to take into consideration. Does that?
A little bit, because my understanding from your presentation, in order to have a patent on cells, it has to differ from the way it is presented in nature.
Oh, yes, that's correct. Yep.
Therefore, from what I understand, it's kind of complicated to make cells that differ one way or another from the way they are presented in nature, and to be used. So instead of patenting the cell itself, if you patent the way the cell is kept alive, then you have control over who has access to these kinds of cells. Or am I misunderstanding?
No, so you could patent, I think I did answer the question, that you can patent the device that's being used to keep the cells alive.
Okay.
And so if someone else is using that device,
to keep their cells alive, they would be infringing your patent.
The device or the process?
Or the, yeah,
Okay.
the device.
So if you have
or
a device or a
process.
process to keep it alive, then you can protect that, but not the cell itself, is what she's saying. Okay?
What I should have said to you in that university tech transfer situation, you've got a license to use technology.
You could.
That license is all you need to get an R&D.
Oh, okay. Thank you.
Okay. All right, that was wonderful. One more round of applause for Carmela, James, Charles, and John. Thank you so much. There's something special here for all of you. I'm very happy to present this to you from the College of Engineering. We have Dr. Kat Donaldson from Launch Labs. Kat, hey, how are you? Nice to see you. So she's going to be talking about introduction to Alexandria and New York City life science ecosystem engagement, right?
Yep.
Okay, I got it right.
Hi, everyone. Really nice to meet you. Thanks so much for having me here, and looking forward to giving you a very quick introduction to Alexandria and our role in the New York life science ecosystem, and particularly highlighting a few of our platforms, but especially Launch Labs. I am Kat Donaldson. I'm the site head for Alexandria's Launch Labs platform here in New York. We have two. I'm the site head for the one on main campus. And I'm also a principal for our venture investments team. And I'll talk a little bit about how those two kind of intersect and work together. Okay, so for those of you that are not familiar with Alexandria, overall our mission is really focused on creating, growing, nurturing, facilitating these ecosystems of life science innovation across the country. So essentially we have ecosystems wherever you expect life science innovation to be happening. So here in New York, Boston, Research Triangle Park down in North Carolina. I think I actually have a slide on this. Yes. And then on the West Coast, we're in Seattle, San Francisco, and San Diego. And we own and operate over 40 million square foot of life science real estate space for life science, pharma, biotech companies. And I think the unique thing about Alexandria that perhaps a lot of people don't necessarily appreciate is really the whole spectrum and the breadth of our activities, but especially in terms of the real estate pipeline. We really cater to companies from very earliest stages with our Launch Labs platform that I'll go into in a bit more detail, up to, you know, a whole building of basically, you think of it in big pharma and major biotech, they're probably a tenant of ours in our space. So we're really kind of the go-to service provider in terms of real estate for life science companies. So as I talked about, our core business is real estate, but we have several other verticals as well. We have our venture investments pipeline too, which is not necessarily kind of tied to the real estate pipeline, but is definitely strategically synergistic. We also take corporate responsibility and thought leadership in our ecosystems very seriously. So we do a lot of engagement with ecosystems. I'll talk about that too, but things like this, really actively engaging with all of the stakeholders in each of our regions and kind of all of the soft support that goes along with the hardware to, you know, as you can imagine, like really grow a life science and biotech ecosystem and region. Okay, so we're in New York, so I'm going to focus on New York City, and that's also, with our teams are very regional too, so myself and my colleagues here in New York are very focused on New York City and all of you guys that make up the ecosystem here. So this is a summary of our sites in New York currently. Our main campus, as I've mentioned, is down next up between Bellevue and NYU, so it's on 29th and 1st. You may have seen it. It's on the water, very nice kind of midtown, all the way on the East River. We also have, so we have my kind of Launch Lab site is based at that location. We have a second Launch Lab site, which is up on Columbia University's campus in their Alaska building in their biomedical campus. And then we have additional sites, one near Grand Central and one soon to be two actually in Long Island City, and growing footprint in New York. So these locations are strategically positioned in New York to be easily accessible to the academic institutions. We really appreciate the amazing breadth and diversity and depth of innovation that's coming straight out of the academic labs, and that's definitely, we take very seriously, that's definitely a serious pipeline and feeder for biotech companies here in New York, very much, you know, grassroots kind of ecosystem still compared to some of our others. So we take very seriously our role kind of engaging with all of you that are based at academic institutions. So yeah, this summarizes that, but also, you know, I think Alexandria being kind of the go-to. We've been described as the expression the tide that raises all ships, something like that. So we really work very closely with all of the stakeholders in the ecosystem, not just the academic institutions, but we have a phenomenal investor network too that we're very close to. Obviously, our tenant base is kind of the companies in the ecosystem, and then all of the entrepreneurship groups, programs, accelerator programs, all of these. We really see ourselves in our role here as being like at the nexus of all of that. So we're kind of the go-to. When you want to start a company, we're a good like one-stop shop for kind of everything that you might need when you're just like, what do I do? Who should I talk to? You should talk to us. Okay, so I want to delve into a bit more detail on some of our platforms. As I mentioned, we have this kind of unique combination of hardware and software. So I'll go into some of the platforms that I felt would be most relevant to this audience today. And most of interest, but for sure, if there's anything else you want to hear more about, just grab me later or reach out on LinkedIn. Happy to discuss. But Launch Labs is probably the most relevant of our platforms. So Launch Labs is essentially kind of that entry-level space platform in our real estate pipeline. So it's turnkey, plug-and-play lab and office space designed to be kind of the very first lab space that companies move into. Oftentimes when they're spinning straight out of an academic lab, Launch Labs will be their landing site. It's designed to be super flexible, modular, easy, a lot of shared equipment. You know, you can just like move straight in and get going day one and really focus on the important thing, which is working on your science and building your team and attracting great people to your company. So as I mentioned, we have two sites. This is actually the main campus, Alexandria's main campus. So Launch Labs Launch Labs currently takes up half of this floor, the 14th floor in one of our towers, but very exciting. We are expanding, and so we're soon going to be doubling in size at the end of this year, taking over the whole of that floor. So it'll be 30,000 square feet in Alexandria's main campus in Midtown. And our second site is, as I mentioned, based up at Columbia University's campus. In terms of kind of strategy, concept, facilities, they're basically equivalent. Obviously, the Launch Labs that's on Columbia's campus is kind of geared towards providing a pipeline for spin-out companies coming out of the Columbia labs, but by no means is that an exclusive relationship. There are companies there from other institutions for sure. So it's really just kind of looking to unlock another geography because we appreciate that New Yorkers don't like to change their commute in any way whatsoever. So we know that we have to just set up sites for people. So to date, across the two Launch Lab sites, we've had over 50 member companies come through our platform, which is pretty incredible here in New York. And actually, Launch Labs originated in New York. So the site that I oversee on main campus, it opened back in 2017. And it was kind of an experiment at the time, both for Alexandria to see like what the impact and appetite for a space like this would be, but also really for New York. Like Launch Labs was the first kind of turnkey space like this for early stage life science companies. And then obviously since it was such a roaring success that not only has Alexandria now opened Launch Labs across our regions, but there have been, other similar types of offerings opening up across New York as well. Although obviously we're the best one. So this is a snapshot of the companies that we have currently at the Launch Labs at the main campus at ACLS. So maybe some names that you're familiar with, but generally speaking, these are ranging from pre-seed to very early Series A stage companies, teams of like one person up to realistically like a dozen, 15 people maybe max is what we can kind of fit into our space. And these are the companies that are up at Columbia University's Launch Labs. So again, you may recognize some, but there are a lot of Columbia representation in there, but also, Weill Cornell, Yale even. I'm not sure what others my counterpart would know in much more detail. Okay, so I've been through some of this already, but yep, we really we've designed the Launch Labs platform, and we're constantly iterating on the Launch Labs platform to make sure that it really is tailored to the needs of the companies that we're trying to serve. And essentially everything that we do is trying to make life as easy and straightforward as possible for the companies coming in. I think a nice unique thing about Alexandria, because we're not just looking to capture companies for kind of that snapshot of time where, we're just an incubator and you give us money and cash and we give you space for whatever time you need it and then you move on. We really see Launch Labs as like the entry to a very long-term relationship with companies. And we really hope that when we get excited about a company, bring them into Launch Labs, our hope and expectation is that the company will be hugely successful, grow the team, and we hope stay within the Alexandria family and Alexandria pipeline as they grow. So we can run through these, but yeah, space is designed to inspire. So we really put a lot of effort into making sure our spaces are like just fun, cool locations where people want to come to work. It's easy to attract really great talent to join your company and join your team, because that's not easy. We appreciate startup friendly philosophy. So this is this flexibility. So if you're growing your team and you need to add space tomorrow or next week or next month, we can do that super flexible, super modular, and super streamlined. It's not like a clunky, bulky process for the admission, for the growth, for the kind of graduation on the back end as well. And we're priced very competitively. As I said, we are not like this is not our core business, right? This isn't like our business model is not to generate revenue through Launch Labs. We really see this as like a strategic relationship that we're growing for the long run with biotech companies in New York. I like to talk about this, really focused on kind of that growth pipeline, which means that ultimately all of our interests are very much aligned. So we all just want like the best chances of success and growth for companies that are coming through Launch Labs. So we provide a lot of kind of the soft support that goes into growing companies to kind of access to our network and obviously our investment side too, which I'll talk about in a second. Well, that's what we've got here. Yeah, so we have really nice kind of facilities and operations. We take a lot of pride in our operational excellence. We have kind of everything you would imagine for the most cutting edge and current cell and molecular biology in terms of shared facilities, and even on kind of the non-lab side. We have, nice conference rooms, spaces for board meetings, that kind of thing. Especially the Launch Labs that's on main campus, you are plugged into a broader campus too. So you'll be like in the elevator with the CEO of Bristol Myers Squibb or Pfizer, just like hanging out, or you'll go down to the cafe and be getting lunch with people from all these larger biotech companies too, which creates a really nice environment. And this is just a summary of, I think we may have more than is here because we're constantly updating and kind of getting new equipment in, but this is kind of a snapshot of the type of shared equipment that we provide. And access to our network as well. So obviously, Alexandria, it's like we have a phenomenal network. We've been doing this. We've been like growing. We've been at the heart of the life science industry for decades now across the country. As I said, you think of it, but all the thought leaders are kind of engaged with us in some way, whether they're a current former tenant of ours or an investment. So we do a lot in terms of kind of organizing thought leadership events that our members are invited to and invited to participate in, like big fancy kind of summit things that we do on a regular basis, but really intimate things too. So we'll bring like the New York founders together for like a nice intimate breakfast roundtable to just talk about like whatever's top of mind and keeping them awake at night. So really like fostering this nice community around Launch Labs as well. Let me see, I'll skip this, it's kind of the same point. And then moving on to access to capital. So not only through Alexandria Venture Investments do we provide access to capital for our Launch Labs companies, but we also have a fantastic investment network. So that's kind of one of the first conversations we have with Launch Labs companies when they start fundraising is who do you want to meet? Who do you want to talk to? What kind of investor are you looking for? How can we help in that respect? And how can we make those introductions and really facilitate that process? So moving on from Launch Labs, it kind of is an overlapping Venn diagram, but moving on to our venture investment side, our venture investments are focused in life science innovation. Primarily, the bulk of our portfolio is in kind of therapeutics, diagnostics, that side of life sciences, but increasingly so, we have a really exciting, growing portfolio in ag tech and climate tech now too. So that's kind of run out of the North Carolina region, but it's growing and expanding, so no reason that it can't expand here in New York. We are, a lot of people don't appreciate, but we're actually the #1 bio investor in the country, certainly in New York, and we have been for many years. So we pop up on a lot of cap tables when you do searches of kind of who's most active, who's in the most deals, it's us. And I think that it speaks to our great network. So all of the kind of institutional blue chip VCs that you might want to talk to when you're raising funds for your company, most likely we have a connection with them. We've most likely been in a syndicate with them. So that's why we're able to kind of get access to a lot of those folks. Okay, I feel like these slides it's been a while since I handed these in, so each slide is a surprise. But yes, okay. So just to finish off, I also wanted to do a shameless plug because we are introducing this year a really exciting opportunity. Some of you may have even gone to previously, we've done like an innovation showcase every summer that we host at the Alexandria Center. We're iterating on that and kind of to reflect the maturity of New York as a biotech ecosystem. We're kind of pivoting that to focus really much more on specifically early stage ventures that are emerging from New York or looking to move into New York to take advantage of this ecosystem and kind of really establish operations and get going. And we're launching or reimagining, let's say, an innovation prize, a Launch Labs Innovation Prize valued at $100K. It's a combination of cash and rent credit for Launch Labs for companies that either kind of spinning out of academic institutions here in New York and looking to get operations going. Doesn't have to be tomorrow in Launch Labs, but like let's say in the next 6 to 12 months, like you can see a line of sight to getting a team going in Launch Labs, or companies from outside New York, as I say, that want to get set up in New York. So applications are open now. Please apply if you have a company that you feel like would be relevant, is doing something really paradigm shifting, innovative, exciting in biotech. It's kind of myself and a few colleagues that are reviewing the applications currently. And then in June, we have our summit event where we'll invite a kind of sub-selection of the applicants to do like a little Shark Tank with a panel of judges. And then we'll give the prize out at that event. So yeah, we'd love to see some great applications from you guys. And yeah, the application is online, should be easy to find. But again, if you have any trouble finding it, just email me or find me on LinkedIn and I can send you the link. I think that is it. Any questions?
The membership fee. Does that include all of the equipment and all that? Is that what it is, or is it monthly? Like, is it annual? Like, how does that work?
So it's a monthly fee, and it includes access to everything. It's just an all-in-one, and then you have access to, all the shared equipment, all of the facilities, and everything that I was talking about in terms of our network and our support.
Yeah, they've got a biotech lab.
Yeah.
Wonderful. Dr. Kat Dallison, everybody.
College, it's all about heritage, culture, tradition. And sure, it's all quite elegant and noble. It's a bit too uptight for us. We don't have an elite crest or secret society. As for a Latin slogan, quod su non realiter sumus. Because for us, we're less about flaunting history and more about reinventing the future. Both for the world and, more importantly, for people like you. Self-starters, doers, makers, healers, innovators. Since we started, we've been less concerned with prestige, more into, well, letting you do your thing. which is how the startups start and the experiments lead to a cure. When the blueprints turn into green cities and the world's problems become challenges you'll solve. Seriously though, we've gotten really good at making people great. And so we define ourselves not by who we are, but who we can help you become. Because our legacy is making yours.
Hi, I'm Kahil. Welcome to Full STEM Ahead Long Island. Our lives are networked in so many ways. Computing and cybersecurity are important for us and businesses too. Virtually every company needs problem solvers in these fields. Today, I'm at New York Institute of Technology in Old Westbury. New York Tech is home to the Entrepreneurship and Technology Innovation Center, which is helping students and companies prepare for the cyber challenges of today and tomorrow. Let's go inside.
Silicon Valley is actually forming within Long Island, New York City metropolitan area. And this is because of a number of very, very obvious factors. Our population density, many universities are providing skilled workers, and having a large concentration of medium and small sized companies provide for proving ground for these skilled workers.
ETIC is one of those proving grounds. In addition to cybersecurity, these innovators are working on various projects: a multi-battery monitoring system, an augmented reality app, clothing that doubles as air filters, and a more humane riding crop.
This is the place that students here come to learn about technology, start their own business, building technologies that solve industrial problems, solve global problems, and then they learn to protect their technologies by learning about intellectual property protection.
What kind of skills are helpful when students come to the ETIC?
I mean, if a student comes in as a freshman and they already have some STEM skills from other high school courses they took, it's always helpful, but it's not required.
ETIC attracted a major cybersecurity partner, CyberSafe Solutions.
In today's world, there's a huge shortage of young, talented cybersecurity analysts. So working with the organization, getting people started in those skill sets early is really helpful. So we are what's called a managed security service provider. We feel that no organization can create perfect cybersecurity defenses. It's rather supplementing that protection program with the ability to detect an attack, expel it before it's a major problem.
I'm here with Seth Levine. Seth, what are you doing here?
Working on a research project using machine learning to improve smartphone authentication systems. Current methods are entering a PIN code, and putting in your password, even Face ID, those sorts of things. The problem with that is that if someone takes your phone, that intruder can have all the information off your phone. What's being developed now are these continuous authentication systems. So while you're using the phone, based off of how you walk with the phone, how you touch the phone, anything that you're doing with the phone's behavioral biometrics are being used to re-authenticate you while you're using it to keep the phone safer. So the part that I was working on was basically when that authentication system should kick in. So I created a machine learning pipeline to figure out when there were different sort of change of possession events. So that would be if someone grabbed the phone from you, if you gave the phone to someone, or if you put the phone down.
Are you from Long Island?
Yeah, I grew up in Great Neck. I went to Great Neck South High School. A couple years ago, I got really interested in machine learning, and then I came back to get my master's in computer science, and I was able to focus on computer science and machine learning.
Assisting the researchers is Mike Campisi, a graduate assistant looking to combine his experience as a baseball player and his love for science.
Well, the new computing power nowadays really allows the whole analytics department to change the way they look at the game of baseball. Baseball is almost a new game than it was 20 years ago. And to see that data and computers are really driving this change, it's really interesting, and I'm hoping that it's something that I can get into in the future.
Tech trains the next generation of cyber warriors. Let's go into the front line of digital warfare with Atlas Cybersecurity. We were just at New York Tech, and I understand Atlas Cybersecurity and New York Tech work together?
As a cybersecurity services company here on Long Island, it's really important to build strong relationships with some of the great academic institutions here so we can help get students into our center, get them trained up, and get them ready for the workforce.
So what kinds of threats does your company handle?
What we try to defend against every day here at Atlas is what we call advanced persistent threats. These are nation-state caliber actors. They can be government actors, private actors. They're organized, funded, and objective oriented, whether it be profit motivated, politically motivated, or activist motivated.
I imagine for a large company or a municipality, getting hacked is pretty serious.
A hack not only has dollars and cents cost to doing business, loss of service, business interruption, there are also a lot of legal costs and fees associated with that, breach notification costs in the case of a data breach. We've seen averages in the corporate world of a data breach costing $3.5 million.
Wow.
I think that 60% of small businesses go out of business within 6 months of a successful breach.
I'm with Barry Dinkin. How did you get into this cybersecurity industry?
I started out in corporate law. At the firm, I was the one handling all the cybersecurity compliance related things for the clients that needed them. And we frequently get, you wrote a great policy, you set out what you need to do, but now we need to do it. One of the really great things about cybersecurity is there isn't one path. People envision that you have to be a computer science major, you have to be a Mark Zuckerberg or someone like that. We have friends who were people who were primarily in HR, now are incredibly successful. And if you're at the first in it, whatever your background might be or whatever your particular skill set might be, there's a role for you.
I'm here with Frank Trezza. What kind of mentality do you have to bend against hackers?
Well, as a defender, you also need to know how to think like your opponent, the hacker. I'm actually a certified ethical hacker. That means I know the same tactics that the bad guys use, but I use them for good.
How did you get into this field?
My older brother Richard was really into IT when I was little, and I wanted to follow in his footsteps.
Are you from Long Island?
Yes, I'm from Long Island, born and raised. I thought maybe I'd have to move somewhere else, but since cybersecurity is growing so fast, luckily there are a lot of places on Long Island where I can put my talents to use.
I'm here with Sukhdeep. How did you get into this field?
The journey has been, long and diverse. I was trying to major in biology in college as well.
Are there any transferable skills that you took from biology studies to what you do today?
There's this scientific way of thinking, correlating data you find, kind of like a crime scene. When something happens, you're kind of investigating it. But, instead of human biology, it's more of computer biology.
Where do you see the biggest risks are in terms of cybersecurity as we move forward?
I think a lot of people agree that the biggest risk are people. We ourselves forego our own security for convenience. Having your car's doors open just by walking towards it, opening a phone with the touch of a button. We like to have one password for everything. It's convenient, it's easy.
There are so many opportunities and interesting people in computing and cybersecurity. To learn more, visit listemhub.org. Now, let's get the hack out of here.
So it is my pleasure to introduce our next speaker. Our next speaker is Dr. Richard McFarland, PhD, MD, and he's the Chief Regulatory Officer at ARMI. The title of Dr. McFarland's presentation is Translating Discoveries into Medical Products. Dr. McFarland?
I've got a PhD, MD, which is not the normal way you hear it. That's because I'm a masochist. You start a PhD and decide you're going to do an MD halfway through the PhD and then do an MD right after. So I appreciate the introduction. I appreciate the opportunity to be here. ARMI stands for the Advanced Regenerative Manufacturing Institute, which is a public-private partnership in Manchester, New Hampshire, funded from the public part from the Department of Defense. We're almost 6 years old. And the purpose of that was to work on manufacturing of cells, tissues, and organs. So we'll talk about that in the talk a little bit. I'm also president of the Standards Coordinating Body. Dave Lewandowski this morning had a nice plug for the Standards Coordinating Body, which is another nonprofit that I volunteer as president of, that works on doing standards, and I'm also on the advisory board or something called True Trials. So Dr. Rodeo, if you heard his keynote, talked a little bit about stem cell clinics and not having stem cell clinics. So True Trials is just getting started, and their goals are to list in a publicly available website trials that are under IND or IDE that have FDA approval for regenerative medicine products. So patients, investors, other people can go and say, these are the trials that FDA has allowed to go forward. And the question is, well, why do you need that? And the reason is the fact that you have an IND or an IDE is a trade secret, and FDA won't tell you. And then the NIH has a thing called clinicaltrials.gov, which started about 20 years ago for the National Institutes of Health funded trials. They wanted people who had NIH and then expanded to federal funds at all to list those because they wanted people to know what they were funding. But they also wanted to have company-sponsored trials there. So they said, well, you can go and have other trials as well. But they don't vet it very well. So some of those stem cell people have trials, but they're listed on clinicaltrials.gov. They say they have clinical trials. That's where they are. So I've got my hands in lots of things, all about making this field better. I suppose that's recovery from having spent almost 20 years at the FDA, working in the part of the Center for Biologics primarily, both as a reviewer and a policy person, trying to move these fields forward. And here's one of the problems. Discovery science and discovery engineering, it's just too long to put science and engineering because it takes up the whole slide. So discovery science and engineering, it certainly includes engineering, and product development are different disciplines. So this is discovery science. You have a discovery, you publish it, then you think about what are you going to do? I'll publish at the end, but no, you also have to get the next grant application because you have to fund it. That gives you to another discovery. They both lend you to hypotheses. You go back to publications. You go back to hypotheses. That is a wonderful thing to do. It's extremely important in academics. It doesn't usually get you to a product. So we had some sessions on patent, tax, funding. If you're working about a product that is a therapeutic product, it needs to be more linear. It doesn't mean it doesn't go completely linear, but you need to put your academic discovery generation, oh, it's a new idea, I'll spend another year doing that, hat, and figure out how to do that without doing product development, and realize which one you're doing. Both are good. Some people can do both at the same time. Most people need to concentrate on one or the other. This big hexagon at the other end of that slide, and I will say, if you wonder why there are all these hexagons, that's why. It's supposed to represent liver triads and cells on top of them, and the colors you get when you stain things for a normal light microscope, not high-resolution light microscopes, but those blurry ones that we saw earlier. There is a development tool called, oops, you know, it's not a talk if you don't move a slide forward once or twice. So, I did it again, I'm sorry. There you go. That development tool is called the Target Product Profile. So during lunch, Dr. Rodeo was talking about what he does in his clinical practice and how you move that forward. That's kind of what the Target Product Profile, you've got an idea. It might fix something that the clinicians are need to use. You figure out what that is. Quality is, what does it look like? So it's all based, this is an idea that came up about in the biomedical field about 2004 by FDA and some folks, some of the regulators in Europe, and said, let's write some guidances about it and tell people, start with the end product. So start a product label. You know, when you get the therapeutic and it's that, has a big flashy title and then about 3 pages of very small text that no one ever reads, that explains what the product is and what it's really good for and all the millions of things that might be an adverse event. The idea is to take that as you're thinking about, I'm going to make this idea, this discovery, and my hypothesis is it might be a therapeutic product. Go and look, develop a target product profile. There are lots of guidances to do that. We have some guidances that we've developed for our members at BioFab to help them think that through. But you have to think that through, and then you have to stay there. Now, the outer parts of that hexagon are a list of all the influences, or some subset of the influences that might influence the target product profile as it moves. This is a slide that is wanted to be there. This is the Jimmy slide because it's the valley of death, where you have academic funding for your great ideas. You don't have industry funding yet. How are you going to explain it? And one of the things to talk to people like Jimmy is, what is your product? We heard that in the last session, right? If you don't have an idea, if you can't know what you're going to do with this, you can't talk to the accountant and tell them what you're going to do, because it makes no sense. This is where BioFab lives in this area. It's also the area where people start to interact with the FDA if they're doing things according to Hoyle. It's a really scary place to be because sometimes money is, well, all the time money is pretty scarce. But you see, it's that space between fabrication in the laboratory, capacity to produce a prototype. This is a DoD funding, so they don't do clinical trials, but you can think about after producing a prototype as clinical trials toward the end of that stand. You need to figure out how to get through there. That's how you get your discoveries into medical products. It's really kind of difficult. But I'm going to tell you it is possible. Listening this morning was listening to some of my careers at FDA, because many of the products are things I worked with on the other side in FDA. One of the things to remember is FDA is a public health agency. It doesn't really care about your target product profile, and they don't care how much money Jimmy has given you. They don't care how fancy your building is. The investors care a lot about that, so you've got to think about both of those things in your brains and remember who your audience is. So they've got a public health, they've only got 2 ways to do it in very basic sense. Keep dangerous things off the market and put helpful things on the market. Believe it or not, they do like to say yes. I'll send you the slide, Dave. You don't have to take a picture. It all goes back to this horse named Jim in St. Louis a little over 120 years ago. So they used to treat diphtheria was their whole big thing. Those of you who are parents, or have younger siblings or cousins or whatever, little kids get 3 shots DPT, 2, 4, and 6 months. The D is diphtheria. Used to kill a lot of kids. 120 years ago, they treated that by giving vaccines for diphtheria were made by horses. So they infected horses, took the horse serum, injected it in the kids. A bunch of kids started to die in St. Louis of tetanus as a little cluster. They said, what this doesn't make sense, let's follow it up. And they followed it up to this horse. Well, this is probably not actually Jim. I like to think since St. Louis, Jim was a Clydesdale, retired, but the theory, the fable is that he's a retired milk wagon horse. In any event, he developed tetanus. They put him down, but they didn't recall the product. So they ended up giving kids tetanus. That was actually the first part of what happened to become the FDA in 1902. Okay. It's interstate commerce. It comes with the Constitution. I'm not a lawyer, so I'm not going to practice, although I played one at work for almost 20 years at FDA. Biologics Control Act was the first thing. Pure Food and Drug Act didn't come until 4 years later. What's usually called the baseline of the starting of the FDA started in 1938. 1962 was the first requirement that you had to prove it was effective. And then medical devices, surprisingly, were not regulated until 1976. Some of us in the audience remember 1976, many of us don't. It was a wild year, it was a bicentennial year. It's like we're going to regulate medical devices. Why is this important? Well, you have to interact with the FDA, so we're going to know what they're coming from as part of the way. Congress has passed laws and have definitions of biologics. This is the statutory definition of the biologic. It says it's a product. It means it's a virus, therapeutic serum, toxin, antitoxin, vaccine, blood component, derivative, allergenic product, or product that has arsenic in it, which is what they used to use to treat syphilis 100 and something years ago when they wrote the definition originally. Protein didn't get there until 2011, distinctly as a list of things you could do. And analogous product includes all the cell gene therapy, all the tissue engineering with cells. Those are all analogous products. That gives you a lot of flexibility in biologics. The other definitions, the drug definition has what the thing does. It's intended for use in diagnosis, cure, and mitigation. It doesn't list what it is, but it tells you what it does. Devices, it's everything except it hasn't its primary purpose biochemical action. So we heard about some devices, some scaffolds. They could be devices. You put lots of cytokines on them, or you elute drugs. They're a combination product. And if your primary activity, maybe you're a biologic or a drug and not really a scaffold. It's important to know all those things because you have to go through this. A multitude of hexagons. I teach a class at master's program up in New Hampshire. This is about 3 days worth of talk. I'm not going to do that. The point being is this little line in the middle that goes to the end, to post-market, that should be your target product profile. What do you want to do with these things, these great discoveries? And you have to know how to interact with the regulatory authority to give them data. Dave this morning talked about Emily Whitehead, the patient one on CAR-T. I was on the other end when she almost died during that trials at FDA. We had to make a decision in a few hours to go forward. But when the folks at Penn first came before Emily was treated, they were doing things for tumor infiltrating lymphocytes. They were having some activity, but wasn't enough immune activity to treat anything. We said, we don't really understand what you're doing. And he said, well, we got this idea about how to treat, make a lot of the T cells, they said all the T cells, respond to a tumor, but you guys will never say yes. And we said, well, have you ever asked us? No. And so they asked us, and we said, well, maybe, show us some more data. So data really rules, and we had a lot of these circles here in the early preclinical. Doesn't help if I do the slide there. A lot of iterations here on the early preclinical data with those folks as they started to explain what their technique was. And then a lot more here. And when Emily Whitehead was sick, it was right in here. She was going to die. They wanted to try something really radical. They called us, gave us the rationale for it, sent us some emails, and we said yes in a few hours, and she lived. If she didn't, we wouldn't have CAR T, which we wouldn't have a lot of the funding. That's the reality of it. It's doable. It isn't easy. It's doable. We're talking about manufacturing. We talked this morning about how you can't afford to do it in the fireside chat, right? You can't afford if it's going to be expensive. This is research and development. Any of you who have worked in a tissue culture lab, that looks pretty familiar. This is manufacturing, state of manufacturing for one of the skin products that was early on that was discussed in the break. It looks a whole lot alike except for the people in the bunny suits. That's not scalable. And frankly, master students, engineers, and PhDs, and certainly not doctors, are not very good manufacturing technicians because they don't tend to do everything the same way every single time. So we have to move to new manufacturing techniques, which is why Army and BioFab USA exist. So this concept is to make manufacturing scalable, modular, automated, and closed. Let's talk about doing automation in manufacturing. And as pointed out, there are some situations that people are working on that, but we need to move to things that are automated and closed. Otherwise, it's just a black box, and you can't reduce the cost that way. So we are putting process analytical technology and process control. That's where the engineers really live. All of these steps, these are the 7 major steps to get from a cell product to the clinic. We can work at all of those steps and work in automation reduction. We're working on 10 different kinds of therapeutics right now at the same time, doing process development, moving them to automation, and doing things that are modular, moving things in and out, and not custom building for a particular product. And the only way you can do that is to build good sensors. So the high resolution microscopy, something we need to think about doing. Need to think about measuring things other than oxygen and CO2. We're doing that with about 170 members across the country. And it's all based on, we call this concept the tissue foundry. And around that ring, we have a deep tissue characterization center where we do omics and physical testing on products in process development, go back and say, what do we need to measure? Do we have a sensor that measures that? Can we build that into the control system? We tend to base Tissue Foundry on moving fluidics rather than picking things up in a robot that emulates humans because we think it's more efficient. It can be completely closed. We do use robotics to move things, and we have 3D printers. We could put in HEPA filters. We got this great member has this great 6-axis robot that can be a 3D printer. It can pick and place other things all in a hood. It's very cool. It can be built into that. But we have to do that to know what the cells are doing. Got to get sensing, and it has to be feedback. That's the only way we're going to build the industry, but I think we can do it. So we need all those students, all the engineering students. We're hiring engineering students all the time. Engineering students who know a little bit about biology are even better. So thank you very much.
Thank you very much. All right. With that introduction, thank you, Dr. McFarland. So, it is my pleasure now to introduce our next speaker, and Dr. Brooke Grinlinger. She's the Chief Scientific Officer at the New York Academy of Sciences. The topic of Dr. Grinlinger's talk is Ethics of Human Genome Editing. Please come forward. Thank you.
Thank you so much, and good afternoon, everyone. You're in the home stretch, so stick with me for the next 15 minutes. So first, my thanks to the New York Institute of Technology for the very kind invitation to come and speak to you here today, and for curating such a compelling scientific program. So among the many types of regenerative medicine that you've heard spoken about today, I'm going to spend the next 15 minutes talking about human genome editing, and specifically the ethical conundrums that scientists and society at large are going to have to wrestle with as this type of therapeutic approach becomes an essential part of regenerative medicine. So technological developments that we've seen in the last decade, including the development of CRISPR-Cas9, gene editing technology that garnered the 2020 Nobel Prize in Chemistry for Jen Doudna and Emmanuel Charpentier here, has given us the incredibly precise ability to make changes in the DNA code within live cells. And that has enormous therapeutic potential for thousands upon thousands of genetic diseases and their treatment. And indeed, human genome editing is now being explored in more than 100 clinical trials for diseases spanning cancer, rare genetic inherited diseases, HIV, and more. But what we're starting to see are the ethical concerns and implications of human genome editing seep into public debate. And many of these ethical concerns were fairly theoretical in nature until late 2018, when Dr. He Jiankui, who you can see here on the right, announced that he'd used CRISPR-Cas9 technology to edit the genomes in human embryos that were destined for implantation into women, with the intention of making them resistant to HIV. And he went ahead and implanted those gene-edited embryos into women, resulting in the birth of twins and a third baby a year later. And that prompted international condemnation from the community over the very premature and potentially very risky use of genome editing in human embryos destined for implantation. And he was sentenced to prison for 3 years for that work. So science really must lead the way in finding a path through some of these ethical and social concerns that arise when we think about using genome editing to alter the human genome. And the New York Academy of Sciences is preparing to launch a new research and training program that is designed to explore these very concerns. The fellowship program will converge on 12 focus areas, and I'd love to give you a snapshot of this fellowship program today and ask you all to think about how you or your organizations might join us in this new initiative. So in light of that very shocking research that was announced by Dr. Jiankui back in 2018, cultural change is very, very sorely needed around the reporting of illegal, unregistered, unethical, or unsafe human genome editing research. And we've seen an increased focus on research integrity in recent years. And with that has come an increase in the number of national and international codes of conduct about how this research is conducted. But there's still no transparent global mechanism for the reporting of this type of illegal, unethical, unsafe research. And that includes there's no support or protection for individuals that might want to come forward and report this unethical behavior. So this prompts us to ask, how could the research community leverage existing entities for investigating and disciplining this type of experimental behavior. So we want to think about how can regulatory agencies, government ministries, academies of science and medicine like ours, along with players like funders, journals, professional scientific and medical societies, be part of providing this type of oversight. In our second focus area, we move to clinical trials for human genome editing. So while guidance principles have been well established for clinical trials transparency and the sharing of data, there's very distinctive and still underexplored challenges that are posed for clinical trials that involve human genome editing research. And while it's really important for researchers, trial participants, and regulators to have access to clinical trial information, this ideal of data transparency at one end of the spectrum has to be balanced with the countervailing considerations about how do we protect patient privacy when we're dealing with their very personal, sensitive genomic information. And if you think about in the context of rare disease research, clinical trials often involve very small numbers of patients, often pediatric patients, and that makes them potentially re-identifiable by giving others access to their personal genomic information. So fellows in our fellowship program will be exploring questions like which data should be shared in these clinical trials, when that sharing ought to take place, and who should be able to access this type of data. Now, focus area 3, we shift to diversity. Genome editing therapies are currently being evaluated in very small groups of patients, and those groups don't adequately represent the incredible diversity in the global patient population today. Nor do they necessarily reflect the real-world use of how a medical therapy is going to be delivered in the healthcare setting for any given patient. And these therapies could work very, very differently in patient populations that differ by age, race, ethnicity, the severity of their disease, what other medications or therapies they're receiving, or other comorbid conditions that they may have. So we really have to think about how could real-world evidence feasibly be collected in a way that reflects the use of these therapies in practice in the real world, and is also inclusive of the various populations of patients, providers, and healthcare delivery settings in which these therapies will be used. So informed consent for the participation in research is an ethical imperative. It's an opportunity for individuals to exercise autonomy. They have the opportunity to make a decision about their participation based on their understanding of the risks and benefits of that research and that therapy. But informed consent for studies involving human genome editing therapies have some significant challenges. First is just explaining the science itself to a patient. The risks are incompletely known as of today and could have really serious consequences. And then in the setting of implantation of gene-edited embryos, the treatments pose risks not only for that potential child, but could be transmitted to their descendants for generations to come. Unborn humans cannot be informed of the risks, both known and unknown, of human genome editing, and they cannot give their consent. So many would bring up the question about, is this an infringement on the human rights of an embryo, of the developing fetus, as well as future children, descendants for generations to come? And then patients who receive genome editing technologies in clinical trials, they will require continuous follow-up for periods, for example, for up to 15 years after that initial treatment. Not only to monitor for the persistent beneficial effects of the therapy, but to also keep a watchful eye out for any delayed serious adverse events that might compromise the safety of the treatment. Now, sadly, the need for follow-up, long-term follow-up, is nothing new. Some of you might recognize Jesse Gelsinger. He was the first person that was publicly identified as having died in a clinical trial for gene therapy back in 1999. And as recently as 2021, we saw the company Bluebird Bio suspend their clinical trials for a lentiglobin gene therapy for sickle cell disease because 2 of the patients developed cancer 5 years after receiving the initial treatment. So there's still no binding standard practice for the period of follow-up that's going to be required for individuals who receive these types of therapies. So we need to define parameters around how long do patients need to be followed up, and think about what's the reasonable expectation for a patient to participate in follow-up over their entire lifespan. Now, scientific research has become increasingly collaborative and international in nature, and current and potential human genome editing research will go well beyond national borders. So governance mechanisms are needed at both the national and the transnational levels. So of concern are differences in what might be permitted and what's prohibited in different countries around the world. And this difference could introduce something referred to as ethics dumping, where we might see research that's prohibited in one country with robust regulation will instead be performed in countries that have little or no oversight or regulation at all. And then a very similar challenge arises with the offer by rogue actors, rogue clinics that will be offering premature so-called therapeutic inventions. So we have to think about what domestic rules either are in place or need to be put in place regarding residents, patients who might choose to travel to another country to receive this type of therapy if it's illegal or unethical in their home country. Are there plans in place? If so, what are they? And if there aren't any, what are the plans to develop them? And while certain aspects of genome editing might not be subject to intellectual property controls, the underlying tools that have been developed to edit genes, for example, the CRISPR-Cas9 systems, they are eligible for patent licensing. So can we think about how either existing IP systems or perhaps we need completely new IP systems to help ensure equitable access to this type of technology? For example, could we insist that licensing costs vary by country based on the economic situation of that country and be proportional? And then there are numerous pioneering researchers and their academic institutions that have a stake in a handful of startup companies. I think you've probably seen some of these logos in Jimmy's presentation earlier today. And those companies have attracted millions upon millions of investment dollars to help them translate genome editing tools into potential new treatments. But we still see that there are ongoing copyright cases, lots of questions and contention over who owns the commercial rights to these technologies, who stands to gain from the potential huge financial windfalls when these therapies come to clinic, and then who gets to decide who and how these technologies are implemented in the future. So the stakes are clearly very, very high. And then, as we've already heard mentioned, the development of genome editing therapies is incredibly resource intensive and expensive. But I want you to ask yourself, what's a fair price to pay for a life-saving treatment, for a cure? What would you pay for your parent, your child, yourself to receive this cure? Well, according to a 2021 report in Drug Discovery Today, the price tag can range anywhere from $373,000 to more than $2 million US. And it's not obvious how these one-time treatments can be readily assimilated into current insurance reimbursement practices. So we would need to explore how could these therapies be developed, introduced, and distributed equitably, taking account into questions of access, equity, and cost? And then how could those models be adjusted from country to country to reflect the various levels of economic development, particularly as we see these therapies rolled out in low and middle income countries? And while genome editing is first being pursued to overcome genetic disease by removing very harmful genes, some point in the future, and it's not too far away, society is going to have to elect where will the moral line be drawn between the use of genome editing to prevent the transmission of very severe genetic conditions and the risk of common disease, or between the promise of improving human health and enhancing human capabilities far beyond what's currently possible today. So if we could use this technology to build in, for example, genetic resistance to HIV infection, or immunity to the flu, or induce super abilities in soldiers and athletes by enhancing their muscle strength, should we? We're going to have serious conversations around where's the line between prevention, therapy, and enhancement. Now, over the course of human history, we have sought to alter our bodies in different ways to restore health or to augment our abilities. And a lot of those enhancements have been quite commonplace and very socially acceptable. I'm sure you've all had a cup of coffee today to help you remain alert, or perhaps you're wearing contact lenses to help improve your eyesight. But would a gene-edited individual be considered a more desirable future parent, employee, spouse, compared to others? So how can we ensure that genome editing doesn't increase disadvantage, division, or discrimination in society? And we're seeing some of these questions already being explored through some of our cultural and literary fields. If anyone's seen the TV series Humans, the cast of which is pictured here, it focuses on some of the social, ethical, and psychological concerns around the invention of enhanced humans. So all of these ethical conundrums have led us to ask, how can we develop better models of education, engagement, and empowerment that will bring all of the stakeholders together around the social, ethical, legal implications of human genome editing? And how do we best make sure we include underrepresented groups in decision-making? So the New York Academy of Sciences response is to invest in human capital. And for more than 200 years, the nonprofit organization, the New York Academy of Sciences, has been engaging with scientists, healthcare workers, educators, policymakers, and the wider public to drive innovative solutions to society's challenges like this by advancing scientific research, education, and policy. We like to think of it as science for the public good. So recognizing that today we live in a world where science moves faster than moral understanding, we are launching a new 2-year fellowship program, a fellowship in bioethics, which is intended to prepare the next generation of practitioners and leaders in bioethics to support their conduct of original research, to address some of these ethical questions around human genome editing, and to engage the public and help them to understand and address some of these ethical concerns as we move towards implementation of these therapies in society. So through the fellowship aims, we really want to invest in the next generation of leaders in biotechnology, people who can communicate, navigate, and understand the ethics and ethical questions and conversations that need to be had around human genome editing and similar biotechnologies that we want to harness. So the ethical questions that I've posed in these 12 focus areas are exactly the types of questions that our fellows in bioethics will be exploring. So just a really quick snapshot to wrap up of what will this 2-year fellowship program involve. Over the 2 years, fellows will participate in an expert speaker series. They'll be supported to do individual research projects and be paired with an expert mentor. We'll place them in a variety of 6-week internship programs, internships in industry, academia, clinical, and policy settings to give them an opportunity to hone their skills in ethical analysis in real-world settings. They'll receive professional development training to help them hone their skills in leadership, communication, conflict resolution, and emotional intelligence. They'll help us to curate the Academy's biannual bioethics human genome editing symposium. They will, of course, receive a very generous annual stipend and conference travel grant, and have the opportunity to publish their original research in our flagship journal, the New York Academy of Sciences. So I invite all of you to join us at the Academy in this effort. We are still inviting sponsors, partners, internship providers, expert mentors to be part of this new initiative. And I'd be happy to chat to anyone who's got questions or wants to learn more at the networking reception this evening, or please feel free to reach out to me at the Academy via email. And thanks so much for your attention today.
Thank you, Brooke, for that thought-provoking presentation. So our last speaker of the day before our closing remarks is Dr. Asar Ilyas, the assistant professor, hopefully soon to be associate professor, in the Department of Electrical and Computing Engineering at New York Institute of Technology. The topic of Dr. Ilyas's presentation is nanobiotechnology and biomaterials in medicine. Dr. Ilyas, please come forward.
Thank you, Dr. Bahishti. So I know that it's been a long day for most of us, and respecting the limited time and audience with the diverse background, I'll try to be precise and as much generic as possible in my talk. Okay, so my research interests are in the areas of disease diagnostics, therapeutics, and tissue engineering. And today I'll talk about some of the projects which are related to cancer, HIV management, and some bioactive surface coatings and 3D printed structural biomaterials for bone implant systems. If we look at cancer, so over the 5 or 6 decades, cancer is still the second leading death-causing disease, and it has not decreased. What's the major reason that why cancer is still the leading death? The reason is late diagnosis. Most of the time, it's late diagnosis and, of course, limited infrastructure that is keeping the cancer death at so high rate, and how nanotechnology can help us early detection. So how integration of nanotechnology and biology together can help us to detect these fatal diseases at early stage. So generally, you know that body is made up of different organs that can be divided into tissues, which is dissociated into cells. And within a cell, you find RNA, DNA, different molecules. This is where most of the diseases originate. And at cell scale, this is the first expression site for most of the diseases. So nanotechnology helps us to provide tools and platforms that can be used to interact with these small molecules and directly investigate at the cell scale. So it means we can have microscale devices or nanoscale devices for direct investigation for interaction with these small molecules. So if we, first project which we talk about the cancer cells, circulating tumor cells, CTCs, they're very rare in blood sample. So and the detection and counting of these circulating tumor cells can be a very strong indicator, not only for the detection of the cancer, but also as a therapeutic monitoring effect. So different techniques have been used to detect and count the cancer cells. We have used solid-state micropores, and if you look at the solid-state micropores, how they work, that we have cells that go from the inlet to the outlet side, and as they try to pass through the micropore, the current, there's a drop in the current. We call it as a resistive pulse signal. So there is a pulse signal, and that's how you get the resistive pulse signal, which is like a fingerprint or specific pulse for each type of cell. So it's very, very sensitive because you are interacting with one cell at a time. But at the same time, throughput would be a challenge because if you're processing a large amount of blood and you're counting one cell at a time, in that case, throughput might be a challenge. So what we did, that instead of using one channel, we proposed as a proof of concept that multiple channels in parallel can be used to increase the throughput. And here you see that this is a device where PDMS gaskets were used to avoid leakage. And this is a small micropore. That is what is actually detecting and counting the number of cells. We optimize different parameters to make sure that we get the best results. And after optimization, you see that this is the specific pulse signal, which I call as the fingerprint or translocation profile for cancer cell. And this is very different from the other blood cell, which is white blood cell or red blood cell. And on the scatter plot, it is very clearly different. We checked the detection limit, means that how small number of cells can be detected. We verified that we can go as below as 10 cells per ml. And the reliability and the repeatability, that was also confirmed over 30 minutes. We let it run for 30 minutes and it was very reliable. Now, scatter plot clearly shows that more than 85 to 90% of cancer cells can be identified by using this technique. So here you see that we did not use any cell staining. We are not using any specific pathologist who is looking at the sample, so there's no human error involved. It's like database that tells that which type of cells we have by looking at the electrical fingerprinting, which is a translocation profile. One question that might come into your mind, that cancer cells are larger in size as compared to white blood cell or the red blood cell. Are we looking at the size-based discrimination? The answer is no. We are purely looking at the biomechanical discrimination, means same size but different mechanical properties. Cancer cells, they are more elastic because they had to diffuse through the surrounding tissue, enter into the bloodstream, and make a secondary colony. Due to that elasticity, we can actually differentiate between a cancer cell and normal cell. Bladder cancer cell, as you see here, that they can migrate very easily as compared to normal counterpart. They're almost same in size. And here you see the translocation profile, because they're very elastic, they can quickly pass through the same size. And that's how we can distinguish between a cancer cell and a normal cell. So later on, we try to make it on chip for point of care detection. So we made a PDMS device where we had the isolation chamber to isolate the cancer cells immunologically by using anti-EPCAM antibodies. And then after isolation, we release the cells. This is the isolation chamber. And after capturing the cells, we release them and let them pass through this micropore, which is the detection chamber. So because one cell at a time, it's very sensitive, and immunological detection means it would be very specific also. So this is the process that we have the device, we functionalize the surface, we let the cancer cells to be captured, we wash it so that only cancer cells are remaining in the isolation chamber, we release it and let it pass through our counting chamber or enumeration chamber where the electrical fingerprinting can be used to count the cells. So this is the actual device that I was talking about. We fabricated here in Old Westbury with my graduate students. And that was one device which gave us more than 90% accuracy for cancer detection. Now let's talk about another project that we're working on for HIV management. HIV is an infectious disease, and it's mostly in Africa, which means poor resource areas where we don't have the infrastructure. So it's very much needed that if we can develop something which can be used at home, which can be used remotely. Okay, so for this, we have tried to develop a device which is paper-based microchip, just like you do sugar test at home. So we are developing a microchip which you can use in a 3D printed chip and use your smartphone these days. And that smartphone will be able to count the CD4+ T cells, which are an indicator of HIV, and tell you that how many CD4 plus T cells are. If CD4 plus T cells are less than 500, it means that you are on the onset of HIV, you need ART, which is the therapy available. But these days, due to this late diagnosis and not available of infrastructure, most of the time when people go for ART therapy, the number of CD4 plus T cells, 1 out of 4 people who start this therapy, it's less than 100, which means, again, if something that can be used at home, it would be great. So what we have done, this is the technique that we have used PMMA, which is paper-based plexiglass, and by using laser cutter, we make a semicircle so that a finger prick of blood sample, it can, under the capillary action, it can go inside. Then we let it hybridize because the surface is already functionalized. We wash the surface, and after washing the surface, we put it into the housing assembly that we have with our PDMS lens that we have developed, that's innovative also. And that we also have an app that our computer science students from our college have developed. It's called BNB Diagnostics. And by using that app, we have so far achieved more than 90% accuracy and more than 95% specificity also. So this is, again, another overview that how that device works. And this is how the lens, this is the PDMS lens that is inserted in this device. And you can use your smartphone to count the CD4 plus T cells in your blood. If it's more than 500, the device says green, means no, don't worry. And if it's less than 500, the application tells you that you are on the onset of HIV or AIDS, so you need the ART treatment. Okay, so this is, again, more detailed, that PMMA with some double-sided adhesive tape and glass cover slip. That's how our microchip looks like. We have also made sure that because this needs to be shelf stable, so we have used trialos to make sure that they are stable at room temperature, and we can activate it after 5 weeks. And even after 5 weeks, when we activated and checked, still we got more than 90% specificity and 80% capture efficiency. So it means that after we functionalize, trialos is used to stabilize it, and then we use vacuum sealing to make sure that this microchip can be shipped to anywhere in the world and you can use it. And this is the lens that we have built by using PDMS, that by using temperature and the volume of the droplet, we can control that magnification. And again, as I mentioned, that this is the first time, I believe, we have made it in our lab by using PDMS. It's based on a concept which is polymer-based lenses. And this is the chip that we made in our lab, BNB. When chip goes in there, you can actually use this threading to focus with your phone camera, and that's how we have a built-in LED that sits here to give you a better image capturing. And then the software which we have, again, this is a software, if you see, BNB Diagnostics Lab. So this is the software that counts the number of cells, and this is a testing phase where you see that more than 90% of cells were easily captured. It's still a project in progress, and we plan to launch it once the counting efficiency is more than 95 to 97%. Okay, so that was all about my the cancer and HIV. So the second phase, which is about the bioactive surfaces. You know that titanium is the gold standard FDA-approved material that is used in bone implants, but the surface is bioinert. It's not bioactive. So we wanted to make the surface bioactive so that it can help the bone regeneration faster. And bioactive bioglass coatings have been used, but they can delaminate, means the interfacial adhesion is not very strong. And also, there is no antioxidant support, because when you have a huge bone loss, at that time, reactive oxygen species, high ROS activity comes in. So what we found out, first of all, we found out that ionic silicon can play a role of antioxidant. And as you can see that it can help SOD1, which is superoxide dismutase, built-in antioxidant support, and other osteogenic genes are also overexpressed. And in order to provide ionic silicon, we developed silicon oxynitrophosphide PECVD-based coatings. As you can see here, by that process, we coat it on the surface, and we see that we can control the surface energy, we can control the degradation rate, and that also controls the degradation rate, and that also controls the early mineralization tissue, which is, of course, very much required. And the cell attachment is also very strong on this surface, as you can see here. Several genes, which are osteogenic genes, they are overexpressed. We also tested it in vivo, and in vivo results showed that when we have SiO&P coatings, in that case, the healing was much, much better as compared to non-coated samples. This histology also shows no signs of inflammation, and it's not precipitated bone, but it's with angiogenic potential, which means that you see the blood vessels in that newly found bone. And the 3D printed part, in the 3D printed parts, we actually make many novel composite biomaterials. First of them was the methacrylated alginate, where leponite was used as ionic silicon delivery mechanism. Degradation was controlled by degree of cross-linking and leponite concentration. We saw that a much increased cell proliferation was observed when we had leponite included into GELMA material. And also, we were able to print cells into the composite material, which is alginate and collagen. And we saw that the effect of strontium, that we control the degradation and the swelling rate by increasing or decreasing the rate of incorporation of strontium into our composite scaffold. Here you see that the higher concentration of strontium become toxic. So 1 milligram per ml is the maximum that gives you better cell proliferation, but no cytotoxic effect. Cell viability was still good until 7 days if you are using until 1 milligram per ml. And similarly, early mineralization, you see after 21 days, there were early mineralization when you are using up to 1 milligram per ml. And in summary, we saw that cancer and HIV management, we can use nanotechnology to make small-scale devices that can be used at home in point-of-care settings. And we have looked at some bioactive surfaces and 3D printed scaffolds that can be used for bone implant systems. I want to thank my collaborators, Michael Hage, Dr. Asgar, and Dr. Komatsu from Stony Brook. And of course, the driving force behind me is the lab members. And thank you so much for having me. I tried to cover up the time. And if there is any question, I'll be happy to answer that.
Thank you, Dr. Elias, for your talk, and really appreciate you rushing through some of your presentation. Hopefully going to be around during the networking session, so hopefully if anybody has any question, could ask you. And by the way, this was another example of the amazing research that's going on at New York Tech by our faculty. We made it. I hope you enjoyed it as much as I did. It was just an amazing day with an amazing set of speakers. And now we have reached the time for our closing remarks. And it is my distinct pleasure to introduce to you Dr. Chad Boughton, who's the Vice President of Advanced Engineering and Director of the Neural Bypass and Brain-Computer Interface Laboratory at the Feinstein Institutes for Medical Research at Northwell Health. But also, Dr. Boughton is a member of the Dean's Executive Advisory Board at the College of Engineering here. After Dr. Boughton's remarks, we will adjourn and we will have our networking reception. So with that, I invite Dr. Boughton to the podium here.
I haven't felt this much pressure in a long time because I'm between everybody and the drinks outside. Okay, so yeah, and I'm not an MD, actually. I'm a bioengineer, but I'm going to talk a bit about regeneration, really in a couple of contexts, really about regenerating our fields, and regenerating ourselves even. And I was going to direct some of this to the students. Some of them have left, and I'm a little bit upset with them and a little bit jealous because their cells regenerate way, way faster than mine at this age. And every morning, my lower back reminds me of that. So I have the privilege of working with an incredible team. They are right here in the back, if you guys will raise your hands. I warned them I would do this. And there's something happened last week that will hopefully be announced very soon. But this, our team did an amazing job in doing something that's never been done in humans. We can't say all the details, but I'll allude to some of it today. And hopefully in a couple of months, you'll get to hear all about it. But again, just an incredible team, a privilege and an honor to work with everyone. And everything you're about to see would not be possible without this team. Cancer. So I started my career, and I thought I would talk about the journey a little bit and talk about regeneration. I started in cancer and worked on solid state sensing technology to find tumors, also do intralymphatic mapping. And the challenge really is that surgeons were passing over cancerous tissue and leaving it in the body when they did a tumor resection, for example. And people would have recurrence and so on, a major, major problem. And so when I came out of grad school, I went right into a big program to develop this technology, develop a lot of the all the algorithms and all of the sensing methods. And after many years, it became the gold standard. I found this out later. Dick Cheney it was used on him and a number of very famous people, and it saved lives. So it was really, really neat to be able to kind of be involved in this. But my point is, you never know where your career is going to take you and what you're going to be involved with. And so I think you just always have to keep an open mind and always think about regenerating. The definition of that term is to create new tissue or create something new, to replace lost or damaged tissue or something, right? Fill in the blank. And if we think about it, we have to regenerate new ideas every time we enter into a new problem or a new program, and we have to replace old ideas. I think we often think, we're going to generate ideas and create new ones and we're going to use, we forget we have to replace the old ideas a lot of times. We really have to do that. Einstein, for example, was ridiculed for, special theory of relativity initially, in fact, for years, until it finally and slowly became accepted. And part of the problem was because it completely displaced, Newtonian assumptions and the idea that time is absolute. People did not want to let go of that. So, I've had a lot of projects. I'm going to keep it short, but really kind of going into new areas like hematoma detection, IV infiltration. This ended up being licensed into other areas, and we had thought of this idea and developed it, but they ended up being used in lots of fields around the world, and I loved having that opportunity. Okay, but now I want to talk about the new stuff. So meet Kathy. Let's see, can we play? I don't have the keyboard. You guys, you want me just to run the mouse? Yeah, no problem. Just want to play a few seconds of this.
Kathy Hutchinson is among the first humans to have her brain directly wired to a computer. Even a wheelchair at some point. Ready to try it for real? In fact, Kathy has already driven a wheelchair. See if you can drive it right over to the door. They haven't let her ride in it yet for her own safety. But with monkeys adopting robot arms and a completely paralyzed person driving a chair, imagine where this could be headed. Fantastic. Very good.
So I was able to work with Kathy for several years and would fly out to Boston and spend long days with her. And this was the first time she was among the first 3 people in human history to receive a brain implant to help her with her condition of being paralyzed, and she couldn't speak at all. I had I guess last year I talked about this in the opening talk about my own personal experience where I sustained a traumatic brain injury as I was finishing up grad school, and I had a tiny taste of what it was like to lose speech. I was in speech therapy for many months, and headaches for years. a lot of issues, but I was lucky and got through it. But folks like Kathy are not as lucky. And so this project finally, not too many years into my career, finally kind of pulled me into neurotechnology, brain-computer interface technology. I fell in love with it, and I've been in this field ever since. But even within this field, we're always regenerating. Somebody brought up Elon Musk earlier today. He did not come up with brain-computer interfaces. He was not even close as being the first one to do this. In fact, he came many years after we did this. This was originally in about 2005 and then 2006 with Kathy. Let me go to the next one. And then a few years later, we thought, well, again, let's try to regenerate. Let's try to come up with new ideas and replace some of these older ones. We realized in talking to patients that yes, it's good to move a cursor or drive a wheelchair, very important. But half of the folks living with paralysis, actually slightly more than half, are tetraplegic. They're quadriplegic, can't use their hands, and can't feel in a high percentage of those cases. Can you imagine what that would be like? It disrupts your entire world, and you need help every time you want to feed yourself or do something like groom yourself. So we came up with this idea of having a one-directional, kind of a unidirectional, one-way neural bypass to take signals from the brain, decode those, link them directly to muscle activation. And so we had a young man enrolled into the study named Ian Burkhardt. He became the first human with a brain implant, first paralyzed person ever to move his own limb using this approach. So let me just play the video. He had one desire, pick up a cup by himself. He's a young man, so preferably filled with beer, he would always tell us. But just pick it up by himself. Bring it up to his lips. And he did it. He did it. And that was the first time anyone had done that. So then, not too long ago, and you know, part of the lab is here today and witnessed this moment, but we had another man who was paralyzed from a diving accident, and he was really wanting to just feed himself. So can't open his hand. We are helping him do that through stimulation. Picked up a granola bar and took a bite. So just feeding yourself. All these things that we take for granted if you're an able-bodied person. And then the latest, and now I'm alluding to something that's happening right now. The FDA has cleared a new study that we have launched and are recruiting for, and it involves, let me go back, play this video. Just very quickly, I'll jump ahead to the punchline. We are implanting 5 chips now in the brain, 2 in motor and 3 in sensory, to restore not only movement, but the sense of touch in someone who has lost both movement and touch. And we are feeding that into the computer. We're doing what's called a closed loop approach, where tactile thin film sensors pick up pressure information at the fingertips. That is fed back into the computer and then into the brain by stimulating what's called somatosensory or primary somatosensory, S1. The person will then feel or have an experience of percepts, tactile percepts. We've done this acutely now multiple times, and we have mapped the areas very accurately. The team has been working very hard on that for years now, and we're finally at this moment where we bring those together, and someone can reach out, not only pick something up, but they can feel it. And imagine being able to hold someone's hand, just that personal aspect of life. So that has now led to the last piece that I'll talk about, and that is, okay, a neural bypass is artificial, and it only works when it's turned on, and only when it's implanted. The longest was 6 years with Ian. Now there's a gentleman who's gone about 7, I think approaching 8 years, but no one's gone past 10 years or 15 or 20. So at the end of the day, would there be a way to regenerate tissue? In this case, neural pathways, the spinal cord itself, or even in the brain, say after a stroke. Kathy, I mentioned, had a pontine stroke. So could you regenerate these pathways? We are working on an approach, and Dr. Chandrasekharan from my lab, who's here today, actually is writing the papers. It was a paper split now into 2. And so it was a year-long study and showed some incredible results of anywhere from 400 to 1,100 or 1,200% increase, I think was the final number, in folks that were either paralyzed from a spinal cord injury, or in one case, a woman who was hit from the side in her car and had head paralysis or paresis in her hand. and she couldn't do fine dexterous movements like threading nuts on. That's actually a standard clinical task, and now she can do that, pass that test with flying colors and within the time. And so this all is done through, and I'll use the laser pointer, through stimulating through an electrode array on the spinal cord itself, and you don't have to have a spinal cord injury to actually benefit from this. It looks like it can be beneficial to even folks that have who have sustained a stroke. I've had family members myself that have experienced strokes, and my mother-in-law did as well and never really regained use of her hand. It was her dominant hand. She couldn't write. And it's something we think is very promising. So now, what does this mean? This means that could we bring this whole neural bypass idea together with this brain-spine interface? Could we create almost, in essence, a double bypass? This has also not been attempted. We have approval to do it. We hope that we'll be doing this very, very soon. The benefit would be not only the immediate advantage of movement and touch, in a kind of what we call an assistive mode, but then over time to have a rehabilitative effect. So have lasting, possibly, and I hate to even, I use this word very carefully, but possibly even permanent benefits that could last. Our year-long study showed, I think after even 4, 5, 6 months, participants came back and still seemed to have the gains that they had. And so we're super excited about this. It brings everything together. And we hope this will change really the entire field potentially for paralysis and even paresis. Now, some of this is for the students, but I think maybe we can all appreciate this. Life is unpredictable, and I guess just take all the advantages you have, and even the strange twists and turns that come in, and even the maybe tragic events that come in your life. Make the best of them. Always regenerate. So, no, you talk about reinventing yourself. Well, I like to say, how about regenerating yourself and your field and your colleagues, and stay positive. And like I said, don't be afraid of trying something new, and don't be afraid of failing. I've already commented on Einstein and what happened in his case, and and think about everything he faced in terms of the Nazi Party coming into power and World War II, and his colleagues. He had some colleagues that joined the Nazi Party. I mean, what can you imagine dealing with all of those things? And he had to leave his own country, and but yet he kept on, kind of, persevering. It's the word, I want to say, persevering. And, at the end of the day, follow your passion. Thank you.
Thank you, Chad, for another inspiring talk. Really appreciate it. For those of you who are fortunate enough to be with us last year, the opening keynote was by Chad, and it was equally inspiring. Wonderful, wonderful event. Thank you for sticking through it with us today. All right. Thank you, everybody.